robotic arm, that the robot's right arm would be lifted up. Then the subject see the robot right hands up as a real-time feedback back to the subject, and it is a closed loop thing. The fourth principle, evoked potential. Usually the evoked potential would take place in our brain when we focus on attention task. One very famous principle of this is P300. When we focus on counting some flashing item, doing some cognitive task. Counting is a cognitive task. Then there is a peak, especially up here in our brain. I will also talk about it more. Code-based evoked potential, we can use coding to generate some flashing item with some random pattern. Then we just focus on looking at those code-based flashing item. Then there is also some special pattern in EEG. Then we can extract that to control other third-party devices. More about P300. P300, we usually do it using a grid with a flashing item. It can flash like individually with one single icon, or we can set it to flash in a row column manner. Yeah, it flashes like this. You can feel from this video, it is more of a vintage video already. And then you can see our boss, Christoph Huber, has started BCI research really many years ago. And using this P300 principle, we usually start from a calibration. You see here, we kind of selecting, you see, selecting SHOE, shoe, right? We want to calibrate how the brain response, how the P300 peak appears when we focus on counting the flashing item. So we do the calibration first, let the BCI system, let the software knows how your EEG pattern look like when you count the flashing item. After the calibration, then you can really use your mind to spell the word on the screen. That is the idea of this video. Yeah, so let's say the S, whenever you see the S light up, you count 1. Light up second time, you count 2, etc. Focus on the flashing target word, count it, how many times it has flashed up, and do not move your head. Yeah, then we can also speed up the flashing period to make the spelling even more efficient. Very nice background music. as well. We can also change the alphabet, the character on the P300 speller board, get into Japanese, Korean, Chinese. We can also do that. And in our internal research, we find out that actually humans really recognize and respond to human faces. Have a more obvious response to human faces. That's why in our unicorn P300 speller grid, it flashes with celebrity faces. Right, more about the steady state visually evoked potential. As I said just now, that we would flash a flickering item to the subject. And then when we analyze the EEG and mostly around the visual cortex, let's say when we flash a 7 Hertz item to the subject, we see around the visual cortex some spectral peak at the multiples of 7. So you see here, 7 Hertz have the peak, 14 have the peak. And what is it? 21 also have the peak. Then the researcher can extract these feature signal from this SSVEP principle to control the third-party devices. Let's say here you can have 2 flashing items flashing at different frequency. Let's say you have one flashing in lower frequency, let's say 14 Hertz, the other one, let's say 17 Hertz. The design of the frequency choices, the 2 frequency shouldn't be a multiple of either one frequency. So let's say if you choose 7 as the lower frequency, you shouldn't use a multiple of 7 as the higher frequency flashing item. You shouldn't use 14 or 21 or 28. So that's why there's a little trick in choosing the frequency number for the 2 flashing items. Then you can design your system. For example, when the subject focus on the lower frequency, then in your real-time EEG analysis and real-time feature extraction, you can extract the peak at 14 Hertz. Then when the system detects all those 14 Hertz, then you can control, let's say, the wheelchair to turn right, this kind of design. When the subject focus on the 17 Hertz flashing item, then you can also design it in your feature extraction algorithm. Any peak in the 70 Hertz is detected, you can control the wheelchair to turn left, something like that. g.tec systems, we have developed it in a way to make multiple binaural recording very easy for researcher. We, of course, is the strongest in the EEG-related brain-computer interface system, but we also have all different others body sensor that can integrate with our BCI system for your online processing, feature extraction, neurofeedback, biofeedback design. So much more easier. We even can integrate with eye tracker, Avnius, motion tracker, VR, all that. different fields of application in clinical or in research. And this paper published in Frontiers in Physiology, they have compared different brains here and different electrodes in the signal to noise ratio comparison. And they find that G ladybird, that is our gel electrode, has the fewest disturbance, and our G Sahara, our dry electrode system, has the best performance. This is a video showing you our unicorn 8-channel wearable EEG system that even it is lightweight 8-channel. This backflip, the artifact is there, but very quickly return to normal EEG. And even better, the good news is that we have already upgraded our unicorn system with our Oscar function. I will show you more very soon. There was Martin, our unicorn software engineer. You see, we need to be multi-talented to work in g.tec. Then this is Bernard. Bernard is also originally from Hong Kong, a specialist in unicorn, in VR gaming, and then kind of combine everything. In this video, he is demonstrating using unicorn and the code base visually evoked potential for calibration. Yeah, this is the calibration. Then it flash, then just focus on the flashing item. And then in this kind of attention task, then would need to focus on the flashing item that is indicated by the red bubble. And it's an experiment, but based on that, we have developed some BCI games as well. This is still unicorn. We have many applications in there. This is g.tec, our neural marketing application that the market researcher or the researcher can load in their own picture set and then flash to the subject, and then the visually evoked potential result per picture would be generated for the researcher, and also how important the picture is to the subject would be also shown in a picture ranking. So these are the flashing picture loaded into g.tec, and then after some just very short, maybe 2-minute measurement, then the ranking, the visually evoked potential result can be shown. The Oscar, it is really a good news for all EEG BCI researcher. This Oscar, online signal conditioning and artifact removal. We have this function in our unicorn system. Yes, and what does it do? We see Johannes, our researcher in Austria, put on the unicorn and then bite the teeth, rotate the head, more head rotation. The artifact from all these movement, real time removed by the Oscar function. Yeah. In my unicorn talk, I would do a demonstration as well. Then this one, it is using our g.tec software with the Oscar module in the spring school of last year already. Spring school of, doesn't matter. In the spring school, there is always a live event, live talk. Martin again showing his gymnastics talents. I'll put on the unicorn headset. Then you see all this jumping, walking artifact is real-time removed using the Oscar module. Then yeah, the squat. Push up. This is challenging. This is everyone shall see it. Handstand. Yeah, so with this Oscar function, that I think researchers in sports, researchers in children, researchers that need the subject to move around for more real life cognitive behavior study, yeah, would be useful. Even even hyperscanning with children, like 4 children moving around to investigate their interaction, their emotion. I'm just being creative. So just with this Oscar, many applications. Yeah, automotive research using g.tec. Polytechnic University Civil Engineering is using g.tec in the driving simulation research. For sports, like let's say the National Sports Institute can also use our g.tec with Oscar module in our software to investigate the athlete's emotion, how quickly they adapt to that very stressful competition environment. All that can be very creative. Then let me show you more about our wireless business system. This g.tec research is our CE approved research version of g.tec. The highest number of channel is 64 channel, 24-bit accuracy, 250 or 500 hertz. With 2 to 3 hours of charging can measure 8 hours continuously. Yeah, and I have tested it in person with a Japanese professor. He is curious to know about how far away your g.tec can can be separated from the recording computer. And it is true that is about 10 meters indoor. I did it in Tokyo in May this year. And yeah, fully integrated into our g.tec software environment. We also have g.tec multi-purpose. Multi-purpose means it can do multiple biosignal recording wirelessly in the same headset. And all the multiple biosignal can be transmitted to the computer through Wi-Fi. And you have all the raw multiple bowel signal data in your computer. And we also produce and supply different kind of body sensor. Let's say the more popular galvanic skin response, ECG, EMG, of course, and respiration effect. Yeah, this one is also popular. The g.Nautilus multi-purpose configuration is that the EEG electrode strands are customized with some safety connectors that you can detach, let's say, two from EEG electrode, then you can attach to EMG cable, let's say, then you can have EEG, EMG wireless acquisition and transmission. Yeah, it's also describe it very clearly with this diagram. Then g.Nautilus Pro. Let me quickly let you guys focus on looking at me. Now you can see me clearly, right? g.Nautilus Pro. This is our latest model. g.Nautilus Pro Flexible. What is flexible? Flexible means you can detach the electrode strand from the amplifier, and then you can attach to the other cap. Attach to the other cap more efficiently. So let's say you have one cap that is like this, with this G-Sahara hybrid electrode, and you have these electrodes customized with safety connector. So this is a hybrid electrode safety connector electrode strand. You have one cap configured like this. And then maybe for another research use, it would be better to use gel electrode, let's say. Then you have another cap that is gel electrode. This is our Scarabeo electrode, smallest profile, can do high density EEG measurement. And yeah, you see this one, the electrodes are assembled on the motor cortex. Then you can easily detach and attach to different caps for different experiment use. Let's say you have another cap that is EEG electrode with apnea sensor. You can also easily detach and attach. Also, the cap, we have different sizes as well. Small, medium, large for adult. Small, medium, large for kids. Then different cap sizes, easier changing of cap. Efficiency is important for Hong Kong, especially. So this g.Nautilus Pro series, the highest number of channel for g.Nautilus Pro is 32 channel. Yeah, it is a FDA cleared device. So you can use this to do research in hospital setting, do measurement on patients also. I want to quickly do a demonstration for you. Oh, 15 minutes left. Let me try. Just start video here. Then you also see me over there. Then we change to here. Then you see my another computer. Right. Then okay, my head. It is a dry hybrid electrode. So I want to show you how good the signal quality is. I twist a little bit the hybrid electrode to make sure the pin is touching my scalp and make sure it is perpendicularly penetrate through my hair and then touching the scalp. Then let me turn on the device by pressing it 3 seconds. Then I can see the base station already slowly blinking. That means connected to my amplifier. Then you see it is a g.tec Suite 2024. It compiles everything g.tec software. g.tec Suite alone have all the API, Python, C, .NET, Simlink API. So if your research team have a good pool of talent in all this API, then you can just buy g.tec Suite. Yeah, the APIs are here. Then I want to show you my EEG first. G recorder is the easiest to use research grade software. Where's my the reference electrode? If you look at me again, I now take away the liner, then I stick the reference electrode on my mastoid. Mastoid, the bone behind your ear. Yeah. Then I open the G recorder. Already running? No. Yes. Then we go to setting. Then yeah, because I tested it early in this morning, so it sees my G Nautilus already. When I click on this G Nautilus, you see this configuration window for the amplifier. You see there's a lot of setting. All the researcher would like it more control. So accelerometer is also built in the G Nautilus, and you can of course also see the battery level. Sampling rate, you can choose from 250 to 500. This network channel by default is the best channel for Wi-Fi transmission in case you are in a environment with a lot of Wi-Fi device, then you might need to change a little bit of this setting. Then you can highlight individual channel for further setting, and you can like choose which channel to measure the EEG from. And then of course you can highlight all of them and then change the input range, the bandpass and notch filter, all that. The input range for G Sahara hybrid electrode that is used in dry condition, no gel, then just choose 562. If you have added gel in this hybrid electrode, that is an option, then you can choose lower input range, 300 or 100. For now, 500. Then the bandpass for EEG, 0.5 to 30, or some researchers would like 0.5 to 60. I can change that for you to see. And the notch filter, the power line interference, we would filter it out. The notch for Hong Kong is 50, so I filter out 50. All good for now. Then let me do a little bit of setting of how it visualizes on the scope. I can apply all that is EEG value and the unit, and I have checked the acceleration data, right? Then I just choose from the drop-down menu acceleration. Then the battery, just choose other. Okay, then I can show like now my headset has 16 channels. So I just show 16 channel here. Let's see. I'm happy already. Are you guys happy with this dry electro measurement? Let me try to perform my alpha wave. I'll close my eyes now. Yeah, I hope that looks good to you, the alpha wave. And the acceleration and battery is there. Acceleration if I move around. Mm. Yeah, so this is the G recorder. Then we also have a very powerful online processing software that is called g.tec. This runs in the MATLAB environment, and our g.tec software engineer already have put a lot of used case Simulink template into this g.tec software. Let's say hyperscanning, integration with eye tracker, integration with Tobii Pro, integration with iLink, integration with FNews, all that we have it in the jihadis. So you see all these ready to use template created for you. Visual observer is a combination of EEG, ECG, galvanic skin response, mainly biosignal combined together in this template. Yeah, FNews. And yeah, more FNews template, EEG FNews template. Neuromodulation template, and we also have LSL streaming. This here, Oscar template. We can save data, the raw data or the Oscar processed data. Yeah, separately. Yes, the Tobii Pro, let's say Glasses 3 is the latest version. We have a template as well. TMS EEG measurement. Yeah, Mr. Hammond will talk about it later. We also have UDP interface. Right. And we also have offline biosignal analysis software that also runs in the MATLAB, but you don't need the coding. This is too shy to pop up for now, but doesn't matter for now. Yeah, so this is our software environment that you can choose the license. If you are very proficient in Python, C, .NET, Simulink API, just buy the g.tec suite. You will be happy already. Then if you need the Simulink, all the toolboxes and Simulink template for online processing, feature extraction, real-time neurofeedback, experiment design, g.tec Professional would be your good choice. G Recorder Professional, it can integrate with video recording and real-time evoked potential and heart rate variance visualization as well. BS Analyze our offline biosignal analysis. Yes, if I go back to here, I have 5 minutes left, and always this topic can overrun easily. Right, so this is G Nautilus Pro. I demonstrated the hybrid electrode. Hope you like it. Next page. And our researcher also have integrated our G Nautilus research with ChatGPT for this interesting project. We use Simulink to do some feature extraction, use a C# JavaScript, and then stream the data to ChatGPT. This is Sebastian put on the G Nautilus. You see some eye blink here. This is the Simulink template used for this experiment. And after measuring like 20 seconds of EEG, then we start to stream the data to ChatGPT. Yeah. Then after streaming the data to ChatGPT, we asked ChatGPT some question to see if ChatGPT can analyze the EEG band power frequency data as good as our researcher can do. So yeah, after streaming the data to ChatGPT, we have asked ChatGPT this question. I've acquired some EEG data for 30 seconds, calculated some biomarker from this data. Some delta theta ratio is 2.2, theta alpha ratio is 2.3, beta alpha ratio 2.1. Can you interpret this data? Then this is the ChatGPT answer, and then our researcher find that it is a good reference to ask ChatGPT to help analyze the data to reference that answer. We have an integrated system between G Nautilus EEG and FNIS. We have integrated with Artemis and also integrated with NIRIX. If you want the easiest beginner system, that you can choose our G Nautilus Artemis 8-channel FNIS sensor. If you want higher number of channel, or you want little kit solution, then we can offer you G Nautilus 9X system. We have a specialized Simulink template for you to easily measure EEG and FNIS in a synchronized manner. If you use G Nautilus research, of course, you can configure the EEG as high as 64 channel. If you use G Nautilus Pro Flexible, the EEG part would be as most 32 channel and gel electrode only. Yeah. We have this special design, this octo-thread, to avoid ambience lighting interference and to hold the EEG electrode between the opto sensor as well. And we keep the fixed distance between the opto transmitter and receiver. We can do MI experiment and Stroop experiment using the template that we provided in GHASIS. Okay, time is up for me. And I have 2 more topics to share. I will try to cover more in the upcoming session. Any questions for my talk just now? Let's see the Q&A. Right. Okay, then let me try to find Elena. Hello, Milena. Hello, Milena. You can unmute and turn on your video, and everyone can see you.
I'm trying, but it says you cannot start your video because the host has stopped it.
Let me change a bit of the setting. And you try to share again?
Yes.
Good to see you again. How are you?
Very good, very good. Thank you for this insightful presentation. I think everyone can find something at g.tec if the person works either in clinic or with research or doing both. I think g.tec is a place to go and get what we need. So thank you very much for explaining all of that and giving this amazing overview. And thank you for inviting me also to give presentation.
Thank you for joining us. Thank you for contributing your good experience in your scientific research, in functional mapping and neuromodulation. All that you are the best one to invite for this upcoming topic. Happy to have you. So Milena is a neurotech and brain computer interface scientist, also a podcast manager for neural careers, have a lot of experience in epilepsy, neuromodulation, functional mapping research. She is based in the US, Florida. Really happy to have you. So this is your time slot, and everyone is looking forward. The stage is yours.
Yes, thank you so much, Christy, again for invitation. Great to be with all of you here today. It's a great honor to speak to you, and I will be speaking about functional mapping and neuromodulation for neurosurgery, and specifically for epilepsy and brain tumor surgery. So epilepsy surgery was for a long time my main focus. I worked at the epilepsy center, specifically with the epilepsy surgery program. And now I established my own institute, the Institute of Neuro Approaches, and I am a neurotechnology and brain-computer interface scientist, and we also do consulting and coaching for people who want to establish successful careers in brain-computer interfaces and neurotechnology. So we always open, we welcome you to come and talk to us about your needs and about what you want to learn, and share with you the wealth of experience. And today I will share the wealth of experience in specifically functional mapping and neuromodulation. So where did I get all of that experience? It was first of all, in the United States, I started at Cincinnati Children's Hospital, a specific pediatric epilepsy program where I worked for 4 years. And then I moved to Orlando, Florida, where I am now. It's a warm place, nice place, and I worked at Advent Health, former Florida Hospital, also first of all with pediatric epilepsy program, and then already both pediatric and adult epilepsy surgery programs. So that's my experience from which I will be drawing upon talking to you today. So let's talk, what is epilepsy? Yes, because there might be many people from various backgrounds today joining us, and let's have certain definitions that we want to have when speaking about it. So epilepsy, it's in simple terms a neurological disorder, and it is caused by malfunctioning nerve cells activity in the brain. So something wrong is happening with the neurons, and they start malfunctioning. And those malfunctions cause episodes that we often term as seizures. Yes, so that is just a very general definition of epilepsy. And it's fairly common. So over a lifetime, 1 in 26 people will be diagnosed with epilepsy. And you can see it very well visually presented here. And about 150,000 people here in the States are diagnosed with epilepsy each year. The good thing about it is that it's curable. So most of the time, when we provide people with the anti-epileptic drugs, we can reduce the symptoms, yes, and sometimes people even can become seizure-free, and actually fairly often that happens. However, the problem is that about 20-30% of all people with epilepsy do not get better with this anti-epileptic drug treatment. That is a big problem. And when this happens, when people are resistant to anti-epileptic drugs, we call this pharmacoresistant epilepsy. Yes, so resistant to anti-epileptic treatment. So what to do in these cases? And there are not so many options, although with the developments of adaptive neurotechnologies, brain-computer-based technologies, we can hope that there will be treatments, but at the moment, the most valuable treatment that provides so far the best results, especially in certain types of epilepsy, because there are various types of epilepsy, is epilepsy surgery. And this is actually the picture. This is the neurosurgeon that I worked with at Florida Hospital, Dr. Baumgartner, performing epilepsy surgery. So what is this epilepsy surgery? There is a certain protocol that is established in level 4 epilepsy centers, and level 4 means that there is everything from the imaging to all the evaluations necessary at the center so that we can prepare a patient for surgery. And you can see here the protocol that we used, and very similar protocols are used in other clinical institutions, and we published it, so you can look at that. But what it shows that there are 2 phases of this evaluation for epilepsy surgery and epilepsy surgery protocol, which starts with the non-invasive evaluations, and then the invasiveness is increasing. Yes, so that if we're starting with EEG, then we are going to EEG, magnetoencephalography, doing WADA test, PET, and so on and so forth. And then we're getting to phase 2, where there are invasive technologies that we are using. So if you look, the degree of invasiveness is increasing as we're nearing that point of epilepsy surgery. And what is happening at that invasive stage, that we are using already methods that are not non-invasive, yes, like EEG, but already invasive methods. And this is one of the examples of those invasive methods, which is called electrocardiography, or ECOG. So we are opening the skull here. You can see the brain, and we are adding electrodes that will be able to record the signal from the brain and inform us about the seizure localization zone that we would need to remove. And there are a variety of those intracranial electrodes that we can use. So you can see here the ones that you saw over here, which are electrode strips, and strips over here, grids. Also, this can be depth electrodes that would penetrate, like over here, would penetrate the brain. And this is especially good if we are talking about the structures that are deep, situated deep in the brain. For example, if it's a hippocampus, yes, or anterior cingulate. And we would use other electrodes like grids and strips. We would use them if there is a seizure localization zone that is more on the surface. Or we can use the combination. And you can see here actually both, and intracranial electrode, depth electrodes. We also refer to it as SEG, stereo EEG. So we can insert many of them and create an image, stereo image of the brain of that seizure localization zone where it's located, and those more surface electrodes. Yes, so there are various ways to do that. And where this electrocorticography is, if we look at all the imaging methods, starting with MEG and EEG, and then going into local field potential, single unit recording, yes. So local field potentials, probably it would be something that Elon Musk is currently using. Yes, he is recording on that level. And here, non-invasive MEG and EEG. So we're somewhere in between when we are recording ECOG, just to give you a perspective where we are when we're recording this signal. So if we compare what is the time resolution and spatial resolution, yes, how well we can sample the signal in time and space, space meaning where exactly is that seizure localization zone, ECOG is giving us the best results because it gives the same perfect time resolution as MEG and EEG, which other methods do not provide, like fMRI, for example, yes, but it also provides spatial resolution, which fMRI is so good at. Yeah, so it basically takes the best of all imaging and provides us with this information. And that is why we are doing all of that. We're starting with non-invasive, but we need to go to that invasive phase. We need to sample the data with ECOG because it's the only ECOG that can provide us with the proof of what we really need to know. That's why we call it a gold standard. Yes, gold standard of our recordings. And if you look here, just to compare the signal of non-invasive and invasive, here we have EEG with the same with the patient. Yes, so you can see that there is this epileptiform activity that is going through all electrodes, so it's fairly generalized. Yes. We cannot really pinpoint where is it coming from. We cannot do a surgery based on this data. But if we are using ECOG in the same patient, you can see how the picture changes. Yes, so those black areas, those are the ones that were already localizing that seizure onset zone that we're interested in. So it's a huge difference between this and this. And that's why ECOG is so important to us, ECOG or SEG, dependent on what we're trying to sample, what we're trying to find. And sometimes even that is not enough for us to localize that seizure onset zone. And there's some of the papers that we published. In that cases, for example, we can perform bilateral implantation of electrodes, so implant the electrodes on both sides and perform a corpus callosotomy. So we can sever this junction, corpus callosum, and we can prevent the seizure spreading from one hemisphere to another in order to localize. And for example, here, if you see on the left, it's before we did this procedure, and on the right, after we did this procedure, we can clearly see that the seizures are coming from right hemisphere. Whereas on the left, before we did that, we were not able to see that. And the same here. On the right, it's another patient, and patient's activity here on the left, we cannot localize, even with echoc, yes, but after we do corpus callosotomy and sample signal from both hemispheres, we can localize that seizure activity in the right hemisphere, and then it gives us a clue, okay, now we know where we can perform the surgery. So, and everything would be really easy. So we found where the seizures are coming from, we can label that place, and then we can perform a surgery. So here on the left is still the brain that is intact. We're labeling it, and then we are performing a surgery here on the right. We're removing those parts that are causing seizures. Yes, everything is simple, and I think we would have a lot of success if that would be the only thing we need to do. So what can go wrong here? Yes, let's just think for those who are not maybe in the field. Let's think together what can happen. Yes, and we know that our brain is responsible for a multitude of various functions. Yes, and for example, here, yes, if it's motor, sensory motor cortex, if we remove that part of the cortex, the person might have problems with their hand movement, correct? And the same, for example, with visual cortex. The same can happen with the so-called Wernicke's area. It's how we perceive speech, our understanding of speech. So if we remove that place, so it's Wernicke first described that that area. So if we remove it, we will have problems with the speech, understanding, recognition of speech, receptive language function. And if we remove a neural substrate that is located in Broca's area, so it's Paul Broca who described it for the first time, then we will have problems with the producing speech. Yes, so anything really that we remove from the brain, we might miss the function which can lead to post-operative deficits. Yeah, so that's the biggest problem that we are facing. And for example, here, first we need to think, okay, so we have this possible problem. How can we avoid it? How can we avoid those post-surgical deficits that people would lose their motor function, wouldn't be able to walk well, wouldn't be able to hold something with their hands, wouldn't be able to talk, and so on and so forth. Yes, so for example, like here, you see the seizure-related activity is on the left in red, and the function, the motor function, is here in green on the right. And we see that there is an overlap over here between them. Yes. So, for example, if we remove this substrate, then the person might develop a surgical deficit. So what to do? And some people might say, no, we all have the same anatomy. Why to worry? Yes, we know exactly where the motor cortex is located, or where is Wernicke's area is located, or Broca's, and you know, we just can make sure we avoid those zones. But it's not as simple, because especially in people with epilepsy or brain tumors, there is transfer of function because of that area of the brain that is susceptible to seizures. It cannot perform the function anymore to the extent that we need. And sometimes the language, for example, might move to the area in the same hemisphere. So shifting language from one place to another, or even into a different hemisphere. For example, from the left, like here, it moves to the right. So here we should expect in healthy people to have activation in Broca's area on the left, but you see that actually in this person it's on the right. So that's why it's so important to have individualized approach to finding those areas and finding those eloquent cortex areas is called functional mapping. And for functional mapping, because it's so important to perform it, together with finding where the seizures are localizing, because it's so important, people developed so many various techniques. Yes, it starts with WADA test, electrical cortical stimulation mapping, electrocorticography, like we were talking, navigated transcranial magnetic stimulation, functional magnetic resonance imaging, probably you all know about, because also we have a variety of different functions. Yes, we want to localize. But what is interesting about all of that, there are so many techniques, but we still are struggling often to find the right area and to really map the brain so that we would avoid deficits, especially with language. And not only that, there are other, each of the methods has their own drawbacks and some positive sides. So there is no ideal method. Yes, we're still in search. We want to find that ideal method that would help us to perform this functional mapping. And this is just an example of how sometimes the results of all those techniques look like. Yes, because we have all of them in level 4 centers. We would have magnetoencephalography, functional magnetic resonance imaging, transcranial magnetic stimulation. Yes, all of that. And we put all of those results together to try to decipher, okay, what can be in trouble, what area. We also do tractography here. So you can see here is Broca's area, Wernicke's area. There is arcuate fasciculus identified with tractography that combines both of it. And we also have seizure MEG spikes. And so those are seizure-related spikes in black. And that was actually a very difficult case because the results of functional mapping were not concordant. Yes, so you can see here, for example, in red fMRI, then we are doing MEG and it's localized in a different area. So here in yellow, so really there is no concordance between that. So whom to trust? What type of method to choose? Yes, I just want to show the complexity of what sometimes we see and the problem that we want to solve. We want to have a method that would tell us exactly, yes, this is the area that we need to avoid when we are removing this substrate, whereas those black dots, okay? And at this moment, there is one technique that we consider a gold standard, and we call it electrical cortical stimulation. And already by the name, you can imply that it's a stimulation of the brain with electricity. Okay, so imagine the brain that is already with that abnormal electrical activity, and we want to stimulate it even more with electricity. Yes. So we're stimulating pairs of electrodes. And when we're stimulating those pairs, we should anticipate a change in behavior in the person. For example, we are asking a person to start reciting alphabet, A, B, C, D, E, F, G, and so on and so forth. And if we are stimulating the place that is not responsible for expressive language function, we should not expect any changes in person's behavior. So he or she, they will still be reciting this alphabet. But if we're at the right place and the person is reciting an alphabet, during stimulation, the person should stop to recite. He will not be able to perform this function. So the behavioral response will be different. And this would be a sign for us. Yes, we found an area that is responsible for expressive language function. However, if you think about it, yes, we're looking at behavior. What can it mean that the person stopped? Does it only mean that that we're stimulating the right area, or maybe the person just switched his attention or switched their attention to something else? Yes, maybe there was a sound somewhere, or maybe he just got lost in thoughts. And keep in mind that many of the people that have epilepsy, especially children, they have developmental delays. Yes, so what happened? They have comorbid conditions, ADHD, one of them, a lot. Yes, so they might easily switch attention to something different. So really counting on that behavioral change is a big stretch. And that's why we need to repeat this procedure over and over again, at least 2-3 times, if not more, to make sure that that was really not a behavioral response, but a response of the brain related to that change in brain function. So that's one big drawback. The second is that now we are using many lectures that we want to explore. Yes, we can have 64, we can have 128 lectures. So we need to test all of them, or sometimes a lot of them. Yes, maybe not all, but a lot of them, and several times each pair, which means that we need to work with the patient for hours, which is very tiring for patient, for clinician, yes, neuropsychologist or neurologist, whoever is doing that. That is another big drawback. And also, when we stimulate the brain, especially that is near that seizure localization zone, what happens that patients can develop seizures. And in this case, we are not able to map the brain. We cannot do that functional mapping. And we need to wait until the person feels better and then come in again and try doing this again. And I know quite a few cases that we had when we were not able to do the mapping at all, because exactly of this issue. Each time we would try to stimulate, the person would start seizing. Yes, which is also dangerous. So there is a need for something else. What can we come up with? Yes, especially if we're talking about neurotechnologies, about new approaches that allow us to sample signal and analyze in real time. Yes, let's think about this. And one of the, I think, really biggest innovations in the recent years was a development of high gamma mapping. Yes, so what is that high gamma mapping? This is a technique that was proposed by the team of Gerwin Schock, Dr. Schock. He was the first who analyzed that activity in real time, did gamma mapping in real time, although that gamma frequency augmentation was described before that. Nathan Crone from Johns Hopkins, he was that original idea behind it. Yes, but implementation in real-time conditions was done by Dr. Gerwin Schock. And so what is it? What are we doing here? And what we're doing, we are sampling signal from the same electrodes that we implanted. Yeah, so we're not changing anything. The same electrode that we used to identify that seizure onset zone that we were using for doing that stimulation mapping, yes? But what we can do, we can split that signal. One still will be going into the clinical system. This is the one that we used before. And another would go to a research arm or our, functional mapping arm, let's call it that way. And this is the initial device that we had. It was g.tec, but it was the first iterations. And what we can do, we can start analyzing the signal that we are receiving in real time. And we will be focusing on certain things and what during presentation of certain paradigms. So what we want to see what happens in brain activity when we are presenting a person with a certain stimuli that are provoking certain function. So it's very similar to what we do in fMRI. Yes, in fMRI, you remember we usually have a control task, which is labeled here with C, and active task, which is labeled here with A. Similar also we would do in cortical stimulation, in fact. But here we don't need to do any stimulation. We're just looking at how the signal changes when we are presenting people with this interesting stimuli. For example, here we're presenting a person with pictures and we're asking a person to tell us, okay, what's happening? What do you see? It's an apple, it's a bee, yes, it's a banana, this is scissors, these are grapes, and so on and so forth. So here we're trying to identify the function within the Broca's area, so expressive language function. And we are doing this in blocks in the same way as fMRI. Yes, so everything that we know from fMRI is applicable here with the big difference that fMRI we cannot bring into the intraoperative room. It's difficult to bring a person with all the electrodes into fMRI machine, yes, with implanted electrodes, so impossible. We cannot do those things, but this application is very flexible. We can do it at any time. The patient doesn't feel anything because there is no stimulation. There is nothing that is really affecting the person. Simply give him pictures and ask to name, and we will be looking at the activation in Broca's area. And the same, for example, here we can evaluate Wernicke's area. And Christy, please let me know if you will hear a sound. I will try to play a sound here a little bit. Okay.
Sure, please try.
Can you hear anything?
Nope.
No. Okay. So that's what about now?
What sound is supposed to be?
Yes. So this is the control condition for the listening task. Yes, so that is a listening task. And I can stop it. So this is a control, and we also have stories. So it's story listening task.
Flower in the garden. One day, a little boy picked the pretty flower. So the frog who missed his flower went inside. Where could my flower be? Thought the puzzled.
Yes, so this is an example that we would give to patients. Yes, similar to fMRI, this was recorded by neuropsychologist Anna Byers, specifically for children, so it's for pediatric epilepsy surgery. And this is how we would do. So there is really nothing that we are doing to patients. There is no risk of developing seizures. We can sample the signal from all the electrodes, yes, that we are placing at one time. So that's why it's happening very quickly. And what we are looking at, we're looking at the change in gamma frequency band. That's why it's called gamma mapping or high gamma mapping. We're looking at the frequencies between 70 and 90 hertz, and then 130 to 170 hertz. This is in the States. So it will vary based on where you are. We just don't want to have that 60 hertz power frequency noise. That's what we're trying to avoid it. But the most important is what happens. Yes. So we have that resting period, that control task where you just heard that white noise, for example, or motor task. Yes, when the person is not doing anything. So it's resting. The hand is resting. And this, you can see this green color activity. So it's a power of the signal. And then when we ask a person to perform a task, let's say listen already to the stories, yes, or open and close the hand, what we will see is the red colored signal. And what we can see in these areas of gamma activity, we can see an increase in that gamma power, which tells us that something is happening in the brain. Yes, at that specific location where there is an electrode, and we can see it at each specific electrodes, and without even any behavioral response, we can know by the signal that we are observing in real time that there is a change, that brain is reacting to that control stimulus, which is very important because it brings us from a simple behavioral observation to a brain-based observation, which is objective measure of what is happening in the brain. And still we can look at the behavioral, see how fast they're responding. So there are many things that we can do, but the most important that we're switching from not a subjective observation to really objective brain-based observation. And we can visualize those results on the surface of the cortex, on the cortical surface, by each location. So we can represent the correlation between the task and the signal that we're recording by the diameter of the red dot. So all those red dots here are those locations where we had that significant change in gamma power. Yes, so that change that we observed here. And the more is the change, the bigger will be this red bubble. Yes, that is how we can visualize and see it. So let's have a look. This was one of the first implementations of this approach by Dr. Kai Miller, neurosurgeon, who is now working at the Mayo Clinic. And you will see the patient is here on the left. Dr. Kai Miller is on the right. And Kai Miller will be shaking the hand of the patient. And please look at this screen. You will see the change in gamma power and also changes here on the surface. Yes, so let's have a look again. They're shaking the hand, and immediately in seconds, you can observe already an activity. So instead of hours spending on stimulating the brain and sometimes causing seizures, yes, and having only behavioral responses, we're getting to a place where in seconds we can get the response and we can see the change in the activity. You see, from nothing to very clear activation. We now know where the motor cortex is. Isn't it amazing? I'm amazed every time I looked at this video, and thank you so much for Dr. Kai Miller for everything he does. He does absolutely amazing, amazing work. So as you remember, I was showing you that old system that we used over here. Yes, so it was g.tec amplifiers, but we still were using the software that was different. It was BCI 2000. It's good software. I'm not saying anything, but it wasn't a one coherent system. So what g.tec did, and I'm very grateful to them for that, g.tec created a whole system, a standardized system that has implementation of everything that is needed. So we have an amplifier that can be connected to the clinical system. We still can do this output, yes, that I was talking about from a clinical system that is used for seizure localization into, and then splitting it into the clinical system. But look, this clinical system looks much better, yes, than it was before. And plus, it has a sophisticated software that can help us to perform everything in really very short period of time. So that's a great tool that we can use. Now let's see how it works. And what is also very important, that the system, it can record, but also we can have a stimulator that works with the system as well. So we can record from the same electrodes and we can stimulate them. Yes, why would we want to have a stimulation if we already have a nice mapping? Actually, we want to have it, and I will tell you later why. Yes, and how we can use this gamma mapping to actually make the stimulation a better tool than it was before. Let's learn about this. So, as I said, first of all, here in this already ready to use excellent software and hardware package, we can use existing tasks that we already implemented, that g.tec implemented. And also we can apply any fMRI task that were used in our hospital. Let's say you are at the hospital or you're doing your research and you have your fMRI task, various tasks. So you can bring them all here, all in the system and implement it. And like I said, it is better than fMRI because it's quicker, more flexible, and provides high signal to noise ratio. And of course, you know, it's a mobile system. So what is available? What can we do? What are those paradigms that are already implemented in the system and plus validated? Yes, so it is a task that evaluates somatosensory area, motor area. So we start with somatosensory and motor, and then we're going into language. So we can look at expressive language function, we can map auditory cortex, and we can map receptive language function plus vision. So that is all done. Many centers are now focusing on memory research. That's a new area, and the paradigms also are being developed for that, which I think will be a next breakthrough. So potential uses, where we can use it. First of all, extraoperatively at the patient's bedside. Because what happens, do you remember I showed you those implanted electrodes? After they're implanted, the patient stays in the hospital, if it's usually epilepsy program, stays for about 7 days, sometimes longer, sometimes a little shorter. So from 5 to 10 days, let's say that. And during all the time, the patient sleeps and stays in the room, and we can perform all this mapping, all this functional mapping, gamma-based mapping with him. But it doesn't end here, because we can also perform it intraoperatively. And this is something that centers that are working with brain tumor surgery. They are interested in that part of the procedure because tumor patients don't usually stay with implanted electrodes. Yes, so that's why we need a procedure that will be able to map everything very quickly, efficiently, without causing seizures during that intraoperative procedure when there is already the removal of the tumor. So all this is possible. And let's look at that. So how can we do this? So high gamma mapping with ECOG, because do you remember we also talked about SEG electrode, stereo EG, or we will talk about that later. First, let's go with ECOG. And there was a validation that was performed with the CARTIQ system, with this g.tec CARTIQ system, and it was done in my laboratory, where at Florida Hospital. And here we looked at the motor cortex functional mapping for sensory, and also for auditory perception. And we compared it to both fMRI and electrocortical stimulation. And when you have a look at the sensitivity and specificity compared to both, we see that the sensitivity is really, really high. And specificity is fairly high, mostly with the fMRI is a little bit better than electrical cortical stimulation. Yes, so we see that there is a concordance, not 100% concordance, but definitely a good concordance if we look at that. How does it look like? So this, you can see now software, yes, how it works with the patient. And you can see the incoming signal here on the left, the maps of that function of interest here in the middle, for tongue here on the left, for listening here on the right top, and then lower Rubik's Cube. It's for motor function. The person is just assembling Rubik's Cube. And here is the patient on the on the right, and you will see how this person is performing tasks that are being queued over here, and what are the results. And pay attention how quickly can we observe those results here. So let's go. So first we start with which part then? I think it should be tongue first. So let's see. Right now, the person is giving a command to relax. You see here on the right top corner. So because we need to get that baseline activity data so that we can compare then with task activity. And then you see here a tongue, which is a cue for person to start sticking out the tongue so that we can map the brain area responsible with the tongue motor function. Yes, and here immediately we have those red bubbles. Remember the red bubbles? Yes, the bigger the diameter, the better is correlation between the signal and the task, and the most it is more reliable. Mm-hmm. Mm-hmm. And now you heard the story listening task. Yes. We again, we immediately saw where is the listening. Yes, here auditory function localized, and now the person is working with a Rubik's Cube, but it's really not for frontal lobe. Yes, it's for motor function, moving. Yes, so this is what we do, and in basically a few minutes, we already have all the localization we need. Of course, we want to repeat it 3 times to make sure that the signal that we record is consistent. Yeah, so if it's consistent, then we don't worry. We know that, yes, this is exactly the function of interest. So this is how it works. And again, this is with the patient who is being evaluated extraoperatively. They're still in their room and we're still doing that functional mapping with them. And now, again, extraoperatively still at the bedside, but now we're using those depth electrodes, yes, those stereo EEG electrodes, SEG. And we will start with the motor function, yes, so you will see a person who is performing this motor task, opening, closing the hand. And here we see the shafts with the electrode. So each of those black dots is a little electrode that is sampling the signal in the brain. And we're trying here to look at the deep structures where the seizures are originating. So you will see the signal change in those electrodes that are responsible for motor function. And here on the left side, you will see all of them together and you will see exactly where is that signal of interest. So let's have a look. The person is opening, closing the hand. And we can see that there is immediately activity here. Yes, on these depth electrodes. Electrode over here. Yes, we can see it very, very clearly. And here it is over here. Yes, in the motor cortex. Beautifully here, red dots. Yeah. So you see, seconds. In seconds, we already can see the activation in the brain. And we can even track the dynamics. Yeah, so now this extension in GBS Analyze program, when we are looking at that, we can even see how the signal is distributing in time, how it evolves, which is another amazing possibility. So here is Dr. Takioka from Boston Children's. He worked with 34 patients with perineurologic regions. And over here, the stereo EEG electrode that he implanted. And here you can see examples of patient 14 hand motor function over here, very well evaluated and determined. And here for face sensory, so face sensory area, which was also obviously important for a person because there was possibly an overlap between the seizure onset zone and this function. And Dr. Takioka was able to identify this area and avoid post-surgical deficits. So, and there was no seizure during the mapping, which is not the case when we are doing a cortical stimulation. So we know that we can use it now and extraoperatively with ECOG, intraoperatively with the motor mapping, and now also with language mapping, which many centers are struggling with when they are using SEG. Why SEG is important? Because there is a switch, a global switch from ECOG recording to SEG. Yes, and there is the switch, but people are struggling with functional mapping. So with CARTIQ, you can do it. And this is again Dr. Takioka from Boston Children's, who examined 6 children with CARTIQ. And he evaluated, here you can see left hemisphere, a very nicely identified activity in Broca's area. Yes, and he was able to separate it from auditory responses. So here is Broca, expressive language function, and here auditory responses. So different locations. Now, what also we can do, we can combine SEG and ECOG, because there are some cases when we can have a surface area localization, and also we can have depths. Yes, possible problems in the depth areas, and here we would use both. Have a look at that very quickly developing, but here is the depth electrode, and here are our ECOG electrodes. And again, very quickly it goes and comes and goes, and you saw end activation here and activation at the depth electrode. Yes, so that was, I think, an anterior cingulate case. So We know we can do it extraoperatively with any type of mapping virtually. And now we have intraoperative mapping, which is very important for tumor patients. And again, this was validated both with fMRI and also with the cortical stimulation. So you can see here cortical stimulation results. These are fMRI results, yes, and these are CAR-TQ results for grasping and for picture naming. And we had a very nice correspondence between two. Look at this CAR-TQ and fMRI. Yes, and again, for example, speech arrest when we're stimulating the brain and when the person is naming the pictures. So we can do it. We can do it like here and with the language function, expressive language function, with motor, hand motor function. And this is a very interesting study in and use that was done in Israel. And here they did a mapping of tongue, foot, and hand movement sequence. And here they were also mapping foot, yes, function intraoperatively. So this happens actually during the surgery, which is absolutely amazing because we can do awake surgeries. These are mostly for adults. We usually don't do this with children because they get scared of that. So, and this is how it looks like. Again, on the left, ongoing activity, and this is the surgical area, and we can visualize what happens in seconds. Yes, what happens here in the brain. Again, red bubbles are those places where is our activity of interest. So you can see here a person is moving the hand. Here is the hand. And we can see immediately where is that hand motor activity. We did functional mapping in seconds. So there are examples of language as well, and you can look at that. There is a huge study with 75 patients done in Oregon by Dr. Ahmed Raslan and his team, who demonstrated the importance and the value of having that gamma mapping for surgery of brain neoplasms. And what is important, what I also mentioned, that we can combine of high gamma mapping with electrical stimulation, because CAR-TQ system allow us to do that. And what it gives us, it gives that absolutely amazing precision of cortical stimulation with a reduced risk for seizures, because we can use our gamma mapping to navigate cortical stimulation. So we can tell our neurologist exactly to which place they need to go to map the function, which reduces the time, which reduces the number of electrodes, and reduces the occurrence of possible seizures. We can merge those modalities, and we did an amazing study on that by using deep learning, by using other machine learning approaches, and we can also determine optimal stimulation timing. If we do that stimulation, when exactly we need to do it to obtain those best behavioral responses that I was talking about during my talk today. So you can look more into the articles. You can always contact me and I will provide this information for you. This is just an example how we can record that gamma activity and we can try to target our stimulation right before that. Because if we do this, before that blast of activity, we can get the best behavioral response. So that's another new opportunity for us to use it. And this is the study when we are using machine learning, deep learning. Other new developments in the future would involve changing the experimental setup. from this fMRI-based structure into the one that is natural. And it was done in Dr. Gage's lab by using computer vision. So during the person's stay over those 5-7 days, we can map function by simply detecting where the hand is moving and correlating it with the brain activity. So we would select the region, extract features, track the positions, and here is the nice result, identification of hand motor. That is a new development. Also expanding the band, not maybe necessarily only gamma band, but looking at the wide range of frequencies. And to conclude, why to use CAR-TQ for neurosurgery? It's enhanced patient comfort. So patient experience, no pain or discomfort during the mapping. Reduced medical risks, yes. Next one is efficient procedure. Mapping process is quick and streamlined, minimizing the time burden on patient. And increase accessibility. The ease and safety of the procedure allow for more patients to undergo essential pre-surgical mapping, which before was not possible, especially when people would develop seizures. And improve surgical outcomes. High gamma mapping leads to more precise surgical interventions, resulting in better post-surgical recovery and reduced functional impairment. And also, it's very important simply to use system that integrates everything we need for functional mapping. And we can use it for research. I will not go into the details, but there are neuroprosthesis, speech neuroprosthesis, like a group of Dr. Chang is doing. It's also based on high gamma mapping. So if you want to do research, you can implement it in your research with both ECOG electrodes. And here is the group from Netherlands that did it with the intracranial electrodes with stereo EEG. So there are so many opportunities. From research perspective, also, we can use CAR-TQ easily. This is just the team that I want to acknowledge, with which I worked during 11 years on these amazing studies and integration into clinical practice. And I want to thank you and all of you to invite to listen to the podcast that we're producing, Neurocareers Doing the Impossible. It's about the careers in neuroscience, neurotechnologies, and you can learn about these amazing studies and clinical advances that are happening in the field, including speech neuroprosthesis with invasive technology like CARE, for example. And if you're interested in participating and being on our podcast, please, I would love to have a whole session on careers in neurosurgery. So you're cordially invited. But in general, if you're interested, please contact me on any topic, and I would be happy to talk to you and have you as a guest on the podcast. So thank you very much, and I'm open to your questions.
Thank you very much, Milena, for your great presentation. Epilepsy surgery, auric craniotomy with this stereo EEG and ECOG functional mapping, and combine it with active ECS mapping would further enhance the surgery precision planning. And I know that there are some neurologists and neurosurgeons attending this masterclass. Hope you have some insight from Milena's talk. Feel free to ask some question. Please type your question in the Q&A chat. Then Milena would answer it. Milena, maybe you stay with us for like 5 more minutes and have a look at the Q&A chat.
Yes, I see there is one, I think, something Q&A. Why is your voice echoing? Oh, my God.
No, it's actually mine. During my session, I have handled that already. So
Yes, but if you still have questions, so please, of course, I will stay for at least 10 more minutes, but you can always email me at neuroapproaches@gmail.com. I'm happy to answer your questions or connect you with people that will help you, like Christy, and we have Christoph Kappeler that I collaborated for many years. So any questions, please just contact me, and I'm so happy to help and to make this technology available to as many people as possible to help them avoid post-surgical deficit and to make all the neurosurgical experience better for patients. So thank you so much.
Thank you, Milena, for being available for a lot of us for the furthermore questions. So yeah, please jot down Milena's email, then feel free to contact her. So thank you, Milena. I will jump to the next session. Happy to have you. Thank you for your knowledge. Thank you.
Thank you.
Keep in touch. Thank you. Right. So then next session would be me again. Right, let me click some buttons. Right, the next talk I will share is about brain-computer interface for neurorehabilitation that is efficacious for multiple neurological condition. That is a recovery system, is a combination of BCI, VR, and FES. It is already indicated for stroke and also multiple sclerosis of any stages. And we also have some successful cases in Parkinson's disease, long COVID, incomplete spinal cord injury, and some more. I will share more with you in this coming hour. So yeah, it is a system developed by g.tec and is widely used in more than 20 countries already. To recap a little bit, what is BCI? BCI, we have in this neuro rehab setting, we use neuro rehab wordings. So we have patient, then we non-invasively use the EEG cap put on the patient. Then with our brain-computer interface system, we ask the patient to focus on some task. For example, motor imagination task. Then we can extract some feature signal, use it as a control signal to control some third-party devices. Then these third-party devices will have real-time feedback back to the patient. Then the patient would know he has done something correctly. He has done the motor imagination correctly. In Recoverix case, in Recoverix design, we make use of motor imagination as the brain-computer interface principle. And this Professor Gerd in the 1970s, he had discovered that when we imagine a movement, let's say imagine rise up your left hand, then there is some event-related desynchronization EEG pattern change in your right hemisphere. Then, with this discovery, we make use of this motor imagination event-related desynchronization. Use this, in short, ERD as a control signal to control some third-party devices. In recovery setting, we use the ERD control signal to control two devices. One is the virtual reality VR avatar as a visual feedback. Second is the functional electrical stimulator, the muscle stimulation feedback back to the subject. So the patient during the recovery treatment would focus on imagining a movement, let's say imagining this wrist dorsiflexion, just imagine, no need to move. Then the EEG would be picked up by the EEG cap, then sent to recovery system for real-time analysis, extract the ERD feature signal, then to give a real-time feedback visually through the VR avatar. This avatar hand moving is a visual feedback. It is the patient's mind to think about the motor, to think about the movement, then to control the avatar hand moving. The avatar hand moving is a visual feedback. Then the second feedback is the ERD feature signal, trigger the functional electrical stimulator, the two boxes, one for the left side, one for the right side, to stimulate the muscle to actualize the imagined wrist dorsiflexion physically. So it's a double feedback, visual feedback and muscle stimulation feedback driven by real-time EEG after calibration. Then what is the task for the patient? Each paradigm, it looks like this, each paradigm lasts for 8 seconds. At the 0 second, there would be a attention sound. Then at the second, there would be a audio cue, left or right as the audio cue. And also a green arrow will pop out, that is the visual cue. The green arrow pop out is a visual cue and together with the audio cue, but the avatar hand moving is the visual feedback. Together with the functional electrical stimulator as the somatosensory feedback. So here, simulate again how the paradigm looks like. It lasts for 8 seconds for each task. Right after the audio cue and the green arrow pop out, at the second, it is the patient task period. The patient would have to sustain the motor imagination as long as possible right after the cue is given to the patient. Sustain the motor imagination until he or she here relax this cue. Each recovery training session lasts about 1 hour because like 5 to 10 minutes to put on the cap and stick the FES electrode on the arm or leg, depends on the upper or lower limb treatment. Then would be the training session. Each training session comprises 3 runs. The first run is calibration. Second run depends on whether the calibration is successful or not. It can be calibration or training. The third run, we always finish the training session with a training run. Each run would have 80 trials, 80 paradigm of 8 seconds. So 80 times 8 seconds is the time needed for each run. That is about 10 to 12 minutes. And each complete session would have 3 runs. That's why in total 240 trials. And a complete standard protocol recovery therapy, a complete therapy for stroke patient is 25 training session. So in total, the patient would practice the motor imagination task with the closed loop neurofeedback stimulation in total 6,000 times. That is about the number of trials that a little baby learn how to walk. In the system, we can set up the physician profile and the patient profile, and we have more than 20 languages to adapt to the patient's mother language. If locally, let's say in Asia, like let's say in Brunei, you need Malay or Hokkien, or in Japan, Japanese also have some like more local accent or dialect. If you need more local language, we can work it out together. record the audio and then load it into the system. We can further adapt it for worldwide market. And during the configuration setting, when we click this VR tab, the VR avatar will pop out. Then we can drag it to the extended patient monitor. The patient would just look at the patient monitor, look at the VR avatar to receive the visual feedback as a mirror therapy. When we click on this functional electrical stimulator FES tab, we will go to this page. We can set up the current value for both left and right side. When we start stimulation, there will be a bubble popping out, and then we slowly drag the current value, and at the same time observe whether the patient's dorsiflexion is at the maximum dorsiflexion. Then we stop there. Then one side is set up, the other side same. Then we click this tick button, and then we click start. The session is not started yet. When you click start, it allows the recovery operator or the recovery therapist to check a little bit the EEG signal. There are 16 electrodes assembled on the EEG cap for the recovery system. The 16 electrodes are assembled at the motor cortex because we find out that during the motor imagination task, mostly the ERD would come from the motor cortex. So we just optimize our EEG cap 16 electrode on the EEG cap. Then we check the EEG signal, all good. And the therapist would know what is the next target and what is the next 5 target. Then go back. Then when we click the play button, the training session will really start. The patient will start to focus on the motor imagination task and sustain the motor imagination, and at the same time receive the real-time visual and FES feedback. And in the middle of each run, like you see, there is about 40 trials already done. Then there would be some event-related disorganization pattern and also ERS pattern plotted in this ERD plot. If we look closely to this ERD plot, these 2 boxes here corresponding to right side imagination. Imagine dorsiflexing your right hand. These 2 boxes is corresponding to imagining dorsiflexing your left hand. Right, so C3 is for the signal from the left hemisphere. C4 is for the right hemisphere. We analyzed the mu and beta band power frequency for the ERD, and we focus on seeing the second all the way to the 8th second for each paradigm, and then plot the mu and beta band power frequency in this ERD plot. The red and orange color means the EEG value become more negative. ERD is the EEG value become more negative. Yeah. So in general, when we see more red and orange color, there is more brain activity from that patient doing that motor imagination task. Some results. So this is a young stroke case. She was a hairdresser before the stroke 14 months ago. Affected side, right side. She completed the standard protocol and some more sessions on top of the standard protocol because she wants to improve even further, better. In the beginning, the motor imagination classification accuracy is just 60 something, but we always see that the patient can improve in motor imagination. So as long as the patient is focusing on the motor imagination task and with practice, it would just improve, would do much more better. And the ERD plot, you see it after the session 1, There is not much brain activity, but after the recovery therapy, there are so much more ERD, so much more brain activity observed. We would not compare the ERD pattern of a stroke patient with a normal person. We would just compare the ERD result before and after the therapy. We also evaluate always with a clinical assessment tool, and Fugl-Meyer assessment is the primary endpoint of our clinical study for upper and lower limb treatment study. You see this young stroke patient improved 5 points in total for the upper limb Fugl-Meyer assessment. And more interesting is here, 9-hole peg test. It is a very challenging test for the stroke patient using the parotid hand to put some small sticks, small metal sticks from one plate to another plate and put it into the holes 9 times, and then remove the sticks from the holes, like very fine movement. Even though our standard protocol is just sticking the FES pad on the upper arm to stimulate the wrist dorsiflexion for upper limb treatment, we also see good improvement in fine movement as well. We evaluated with 9-hole peg test and also box and block test. So let's see the 9-hole peg test of this lady. After the first section, 7 minutes 26 seconds, complete the 9-hole peg test, and after 5 sections, already improved it to 1 minute 34 seconds. And along the treatment, all the way, the post assessment improved it to 1 minute 14 seconds. That improved it a lot. And we also observed the spasticity relief in the patient's wrist and hand. Some video report. This lady can hold the comb using the parotid right hand and do haircut for our recovery physiotherapist in Austria. And this is how she can do the 9-hole peg test. Okay, and then this gentleman, 53 years old, stroke 11 months ago, chronic case already, affected side, right side. For the motor imagination classification accuracy also started from somewhere 60-something%, but then improved it along the therapy and achieved it many times, 100% during the therapy. So as I said, motor imagination can be practiced. The ERD plot here, you see less blue and green is also a good improvement, and there is more orange and red here for the right imagination here. The ERD pattern is anyhow very much different from normal person, so we just compare the ERD result before and after the recovery therapy. Clinical assessment report, again, the Fugl-Meyer assessment, we see a positive correlation between high motor imagination classification accuracy with clinical outcome. You see this gentleman, as I said, achieved it many times, 100% here, and we see good positive correlation with the FMA total score increment, improved it like 20 points. He is a chronic stroke patient already. It works for chronic stroke patient that well. For acute, subacute, of course, the sooner you receive the recovery treatment, the better you would have the clinical outcome. But third index of this gentleman improved at 5 points, and the spasticity and the tremor also lowered. This gentleman understand the importance of 25 training sessions as the standard protocol and insisted and comply with this standard protocol 2 to 3 times a week, and in total 25 sessions. You see here when we evaluate the fine movement using 9-hole peg test in the first 17 sessions, that is still not possible to do a neuro-PET test with the affected side. But then he insisted, he understands the importance of the time needed to improve the neuroplasticity is different among different patients, insisted, complied it, and like suddenly after session 18 improved it to 10 minutes 22 seconds, and then all the way to the standard protocol completing 25 training session improved it to 2 minutes and 53 seconds. So from not possible to 2 minutes 53 seconds, that is a lot of improvement. Yeah. And more video result. On the left side, it is before recoverax, wrist rotation about 90 degrees. After recoverax, the wrist rotation, it is about 120. Voluntary, no help from the healthy hand or the therapist. And the video here, we ask him to put down the arm and then point to the nose. You see in the beginning, there is some compromise in coordination and a bit difficult to point to the nose. But after 25 sessions, so much more smoother, put down the arm and point to the nose more smoothly. Then this lady, I have goosebumps every time when I talk about her story. Had a stroke 30 years. 30 years of stroke, left side paralyzed for 30 years, left side couldn't move 30 years, and has been doing motor level, muscle level rehab for all those years, not much help at all. Her boyfriend got to know about recoverax, and then she commit 25 sessions standard protocol right away. The classification accuracy is so 70 something on average, and the ERD plot more deep orange like after 25 sections, so brain activity much more. Neuro-PET test for this lady Gabby. Before recoverax, the left side is the affected side. Left side is not possible to do neuro-PET test, but then along the recoverax therapy, like takes about 1 hour to finish neuro-PET test, but then right after 25 training session, improved it to 17 minutes 16 seconds. 2 more months later, come back to do assessment again, still improving. 3 more months later, still improving, even improved it to 5 minutes 36 seconds. And during these 5 months post recoverax, no recoverax already, just go home to do daily activities. So we see long-lasting improvement in neuroplasticity after the recovery treatment. But third index improved the 5 points. And let me highlight this one here. Modified average scale for WISP, the spasticity in WISP. In the beginning, it's very high, very high spasticity, like 3 to 4. Then it's lowered to 1 for the WISP spasticity. Some drawing by this Lady Gabby. In the beginning, you see the left hand like start from a straight line and then try to go through into the circle, a bit more touching to the edge. You see, after recoverax, it started more smoothly, more circular shape, and then trying better to draw along the spiral, the maze. Focal maze assessment, of course, we would evaluate using this. Before recoverax, the total score just 17 marks. And after 25 training sessions, standard protocol improved it to 28. 2 more months and 3 more months later, still improving. And of course, we have other happy recoverax patients who can play guitar using paralytic hand after the recoverax treatment. The quality of life, we just want to improve in the patients. This is one plot of the upper limb recovery study that we have published in Frontiers in Neuroscience. In that study, we have 51 stroke patients, no matter their stroke stage, no matter their stroke location or stroke type, they have stroke, they just come in to this study. 51 subject here. And the bar here is the accumulative score of different clinical assessment tool here. So the longer the bar here on the positive side, the better the patient outcome. So you see 99.9% positive patient outcome. Then these two guys here, what happened? This guy here unfortunately got the second stroke, and this guy here, very funny, couldn't concentrate on the motor imagination task because he likes to talk a lot, very talkative, always talk with our therapist. So as long as the patient really put effort in focusing on motor imagination, I have done recovery demonstration on many patients in Melbourne, in Nanjing, in Anyang, in Hong Kong, that the patient actually do so much more better than normal person in motor imagination. They have been imagining, like use their mind, want to control their hand or leg to move. Since they got the stroke, they are desperate to like control their limb to move again. They have been imagining it for a long time. They usually can do quite well, like better than normal person. Just that the requirement for the patient to do the recovery treatment, that would be no electronics implanted in their body. Let's say pacemaker, no electronics can be implanted. And also, what else? Can understand basic instruction and can differentiate left or right side. Fulfilling this, they can receive the recovery treatment. We also look into this kind of subgroup analysis. We are interested in looking into how good, how is the correlation between the classification accuracy with the clinical assessment patient outcome. And we use this 80% as the criteria in this subgroup analysis. And we see that the group that have achieved higher than 80% classification accuracy, the Fugl-Meyer assessment, the upper limb Fugl-Meyer assessment would improve so much more better than the group that have achieved lower than 80%. So that's why in our flagship clinic and in our recovery partner clinic, franchise clinic worldwide, we would encourage that in their clinic would have a competition among the patient. We would encourage the patient to achieve higher than 85% the classification accuracy. Then we want the patient to have better patient outcome. That's why with this encouragement, they are more motivated to focus better in the motor imagination task. And we also see in the severe and mild impairment group, the group that have achieved it above 80% classification accuracy would achieve better in the patient outcome. For moderate, it is as good for the patient outcome. What if the chronic or subacute stroke patient that have done 2 times the standard protocol recovery therapy? That means 25 times 2, 50 recovery training sessions in total. So you see here, this chronic stroke patient, before recoveries, it takes about 150 seconds. After 25 sessions, already improved it like a lot. 快咗1倍啊，中文就係yeah. And then after 50 sections, like the task proficiency and accuracy is much better. This is an example of subacute stroke patient, also finished 50 sections in total. So you see before recoveries, 144 seconds. After 25 seconds, 75 seconds. Do 25 sessions more, 40 seconds. Yeah, so standard protocol 25 training session would be a good start as a standard protocol minimum to give the patient a good boost, no matter how chronic he or she already is. Give him a good push in effectively improving the neuroplasticity. Then the therapist can always discuss the goal setting, what the other movement that the patient want to improve. Then they can have a choice to do one more round, the standard protocol, to improve even better, to like regain the motor function to a higher level. In our clinical study, we always use modified Everest scale for wrists and fingers and Fugl-Meyer assessment in upper extremity to evaluate the patient outcome. And these three parameters, we achieved it highly significant result. P-value lower than 0.05, already significant. And you see, we are lower than 0.001. So it is highly significant. This is our paper published in Frontiers in Neuroscience, the BCI treatment for motor rehab for upper limb for stroke patient. And the plot that I have shown you with 51 bars, it is extracted from this paper. We have updated our upper limb treatment with this slap a rat game. Since the beginning of, since I joined g.tec, I have asked a question. The VR avatar is a bit boring. Do we have a more funny game? So here it is. It is the more funny game. When you try to imagine it, the mouse is actually replacing the green arrow. It is a visual cue, the mouse coming out, and then wow, you are so motivated to like raise your arm higher to threaten the rat, to slap it away. You have to sustain and imagine to lift up your hand higher, sustain and imagine it higher to threaten the rat, or else your cheese would be stolen. It is more exciting. Yeah, then upper limb, I have finished talking about it. And of course, we have the lower limb treatment. The setting is a little bit different. Just turn the patient to sit by the recovery table, and the eye is still looking at the patient monitor. Our design of the avatar is always a mirror image of the patient's sitting position, because this VR avatar visual feedback also acts as a mirror therapy as well. Yes, and rest the paratic foot on the footstool. In our lower limb treatment, we would pair healthy arm with paratic leg, because the left and right leg control in our brain is too close to the CZ position, too close to the middle of our brain. So we need to pair the healthy arm, which is further away from the leg control, to better differentiate left and right side motor imagination. Yeah, we stick the FES pad on the TA to stimulate the ankle dorsiflexion driven by real-time EEG. This is how it looks. So recovery is very good that it doesn't occupy too much space. This is just a 4-feet table, and the therapist would control the control computer here. Patient look at the patient monitor with the VR avatar. The parietal leg rests on the footstool, and it's very safe for the patient. 0 falling risk, and it's not physically exhausting for the physiotherapist or neurophysiotherapist or the occupational therapist, anyone, not physically exhausting. And yeah, after listening to the cue, imagine the dorsiflexion, then the EEG would be analyzed, and then the ERD feature signal extracted would be used to trigger the VR avatar to actualize the movement visually. And then the ERD feature signal also trigger the FES to stimulate the muscle, actualize the imagined movement physically. Yeah, and the therapist would see how good the patient is doing by looking at the control computer ERD plot here. The therapists do not need to all the way 1 hour sit by the patient. In our flagship and also worldwide clinic partner clinic, one dedicated trained recovery operator can take care of 3 recovery systems in the same hour. So that is more efficient, effective management of human resources. Some video result for lower limb treatment. This gentleman would do the 10-meter walk test, followed by time up and go test. We have a lot of these pre and post video on our YouTube channel. You can feel free to subscribe to our g.tec Medical Engineering YouTube channel. And there's one playlist that have collected all our pre and post recovery video, all these walking tests, 9-hole peg tests, box and block tests, before and after. You can enjoy all the videos. So of course the videos I show here is showing you the speed, the time needed to complete this walking test is become faster, shorter time needed, and the balance is better after the recovery slow limb treatment. This lady here, before recovery, she needed a walking stick to do to do this time up and go test. After the also standard protocol, 25 training sessions, no need to walking stick already, and walking faster in this time up and go test. We always give the patient a therapy report with the name and then how many sessions in total he or she has done, the classification accuracy that he has achieved it, and cognitive assessment, modified level scale, Fugome assessment, time up and go, 10-meter walking test, Bethel index. We do all these so that the patient would kind of have a report card, how good I have done. And like this is the result of 25 sessions. Then with this report card, then can think about what is the next improvement that he or she want to do, then can discuss with the therapist also. In our lower limb clinical study, we always use this clinical assessment tool to evaluate the patient outcome. Modified level scale to look into the specificity in knee and ankle. Video recording, video is important. We do not base on the feeling of the patient. We record it as a evidence, of course, with the patient's consensus, as a video record that to show the improvement evidence to the patient. Time up and go test, 10-meter walk test, stroke color, work test, range of motion, ankle and knee, Bethel index. We do all this. And we have achieved a significant outcome in all these clinical assessment tools. And out of all these significant improvement assessments, time up and go test and 10-meter walking test is highly, highly, highly significant. We have also evaluated recovery with those electromechanical training, e-gates, that is including exoskeleton, in 10-meter walking tests, recovery patients achieve so much more better than those patients. Recovery is very much effective because we designed this system based on really evidence-based neuro rehab recommendations. We have motor imagination task specific training. We have mental practice. We have bilateral training. We train both sides because stroke patient, like their lesion is there in the brain, and their rewiring is already different from normal person. So bilateral training is always better to help improve the neuroplasticity, to rewire the brain so much more better by training both sides instead of just focusing on the paralytic side. Virtual reality and FES is also included in our recoveries. Canadian stroke recommendation, also the recommended a therapy that is included with mirror therapy to trigger the mirror neuron as well. Our VR avatar visual feedback act as the mirror therapy. Mental practice, the motor imagination task, bilateral arm training. You saw here. Virtual reality training, FES, is also here. This is our paper published in 2023, last year, October. Oh, 1 year anniversary already, for recoveries for gait rehab, published in Frontiers in Neuroscience. We also have a case report from Finland that is using recoveries 25 standard protocol training session 3 times a week for 12 weeks that on this failed back surgery syndrome. The pain level lowered from 8 to 1. It is from this case report, that's why we are now also doing study in Austria using recoveries on chronic pain patients. Recoveries is the only system in the world that is indicated for multiple sclerosis as well. In Asia Pacific, in Australia and New Zealand, there are more multiple sclerosis cases. Well, while I was in Auckland 2 weeks ago, I met some multiple sclerosis patients. They are desperate to have effective tool in the country so that they can receive treatment to improve their quality of life. Multiple sclerosis is the demyelination of the nerve cell. The myelin is the fat component of the nerve cell. Is out of, don't know why, the fat component of the nerve just degenerate. We don't know why. Then it kind of affected how efficient the EEG, the electricity transmit through the nerve cell. The patient usually would feel fatigue very easily, just 2-3 steps of walking, already very tired. The control of motor function, motor movement is also compromised. In the multiple sclerosis therapy protocol, is a bit different from stroke. Multiple sclerosis protocol in total would do 30 training session, and it would be alternate. It would be alternate, like let's say the session 1 would be right hand paired with left foot, and the session 2 would be left hand paired with right foot, alternate. So it is alternating left and right lower limb therapy 15 times. So in total, 30 sessions, 2 to 3 sessions per week is the standard protocol. In our multiple sclerosis study, we use, again, modified Everest scale to evaluate spasticity, video recording, time up and go test, timed 25 foot walk, 6-minute walk test, multiple sclerosis impact scale, and modified fatigue impact scale. We use all these assessment tool to evaluate the patient outcome of this multiple sclerosis therapy. This multiple sclerosis patient here, age 63, male, affected side both sides. Multiple sclerosis patient just got affected both sides. So that's why we need to train both sides, bilateral training. MS time, 26 years already, very long time. And this is the ERD plot of this patient. After finishing 30 sessions, there are more ERD, more orange, more deep orange means more brain activity. And the 6-minute walking test of this gentleman here can walk so much more longer distance in 6 minutes. And the time up and go, like shorter time to finish the time up and go test, and also shorter time to finish the 25 feet walking. We also see improvement in gait endurance, balance, coordination, and walking speed. We evaluate also with multiple sclerosis impact scale. The lower the value, the better. So you see the improvement in the MS impact scale as well. This is him. This is his walking test. This video here is 10-meter walking test. Yeah, reached the target already. This gentleman also stated that in the beginning, his wife need to drive him to the recovery clinic for the therapy, but then after the third therapy, he can already drive to the clinic on his own. Yeah, and he's also able to lift his foot again. We have another case, a 47-year-old female affected side both sides, MS time 24 years, also long time of MS already. This is the ERD plot. And a 6-minute walking test, again, walking so much more longer distance in 6 minutes after the recovery therapy. Shorter time in time up and go and timed 25-foot walk. The multiple sclerosis impact scale also lowered in value here. And the fatigue scale also lowered. This is the video of her 10-meter walking test. You can see here in the beginning, it's very difficult for each step to be done. Here looks easier, so the fatigue level is really lowered. Her statement, after the therapy, he can go outside more often, more joyful, less sleepy, can lift off her foot better. While standing, the foot sticks a lot better to the ground. The coordination also improved it. Concentration and memory always were good according to this patient. 6-minute walking test performance doubled. Yeah. Multiple sclerosis clinical study, we have all these parameters, as I said earlier. And we see significant improvement in all these time up and go 6-minute walk test, multiple sclerosis impact scale, fatigue impact scale, 25-foot walk, modified effort scale, all significantly improved it. And out of reach, time up and go test is highly, highly, highly significant. We have a recovery gym worldwide. In Europe, of course, we have a lot. In Asia Pacific, now we have a recovery clinic, of course, in Hong Kong, in Bangkok, in Seoul, in Sydney, Philippines and Sri Lanka already signed a contract opening very soon. And I know Brunei is also very soon. Right. And we can always discuss this franchise partnership program. We work together as a business partner. You can take up one territory, and then we come up with a business plan to grow into the network of recovery clinic in 3 to 5 years. It's like opening up Starbucks in your territory. We can discuss. Just send me an email. I can show you the return on investment. Then the last few minutes is the highlight of this talk. It already have some good case result in Parkinson disease. This is the result of one of our cases. The time up and go test, you see here, it is improved like double, like the time needed improved it from 26 to 13. 10-meter walking test also improved it from 13 to 7 seconds. Box and block test for the hand fine movement, 9-hole pack test. Again, the fine movement. Also, the time needed is lowered. Spasticity is in the width is a bit lowered. This is the time up and go video for this Parkinson patient. You see here, right? Jump up and then walk to the cone and then return already. And on the left, median not found. But you see the result here, improved it from 13 to 7 seconds. Long COVID, it also works. Why do we work on long COVID? Because in the multiple sclerosis study, we see the improvement in the fatigue impact scale. Then because we also like hear the long COVID patient feedback that they kind of suffer in the fatigue issue after they recovered from COVID. They get tired more easily. So that's why we are inspired to also look into the effect of recovery in long COVID patient. This is the 6-minute walking test, a result for long COVID patient. They also walk further distance. And the fatigue severity scale also lowered like quite a lot, like improved, doubled, like shortened the time from 53 to 27. And we also evaluated with modified cognitive assessment, because some long COVID patients also reported they kind of get brain fog, memory kind of a bit compromised. And we see improvement in the MOCA, modified cognitive assessment as well. Box and block test and 9-hole pack test, also the time needed is lowered. Focal assessment, lower limb, they walk better. Oh, what is it? Oh, it is incomplete spinal cord injury. Yeah, for the lower limb, you see the point is improved at 2 points for incomplete spinal cord injury. Time up and go, you see like from 42 seconds to 26 seconds, like working so much more faster. Let's see a video. This is incomplete spinal cord injury patient. Yeah. So you see it's faster reaching the target. And also see from the back side. We also have an application launched in Android and Apple App Store. This Recoverix at Home, it is the VR avatar of our Recoverix system. It is just the VR avatar component. It is for the patient to practice the motor imagination at home. Look at the green arrow and then practice the motor imagination and imagine lifting the ankle or wrist as high as possible. Just practice the motor imagination part. is nothing replacing the complete recovery system, because the complete recovery system, it is EEG-driven, visual and FES neurofeedback closed-loop exercise. And this recovery at home, this app, it is just the motor imagination part for the patient to practice the MI at home. It can be practiced and improved. So in summary, Recoverix works for 10, 20, 30 years of stroke and onset of multiple sclerosis, and we have tested it for chronic pain, Parkinson's, long COVID, and complete spinal cord injury. And if you check our website, we also have Guillain-Barré syndrome, Condor-Coats syndrome, so many more neurological conditions to come. If you are interested to do research locally in your hospital, let's say you are interested in cerebral palsy, you are interested in ataxia, you can contact me, buy a system in your hospital, then do research in your area of interest. We have already done more than 50,000 recovery therapies and in more than 20 countries worldwide. And Recoveries, we also have a franchise business model to partner up with franchisee to grow Recoveries clinic network to benefit the patient more conveniently, get access to Recoveries clinic to receive the treatment. What a time management. I have finished my talk on this BCI neuro rehab for multiple neurological conditions. Any questions? Any questions for me? Let me check the Q&A. Someone asked, how can Indian students do partnership with Recoverix? Is there any way? Yes, it is. Yes, it is. We have a distribution partner in India, and you are always welcome to buy in a recovery system. Let's say, do research in cerebral palsy, because I know Sri Lanka also have the need for cerebral palsy data also. So maybe Indian students with their research institute can buy in a recovery system to draft a clinical study proposal to do study on that. Great talk, Christy. Thank you. Greetings from Tokyo. Happy to have Japanese participants. You mentioned long COVID rehab with MOCA improvement trials. How about Alzheimer's? Is there research on recovery style interventions for memory rehab in Alzheimer's disease, similar to motor rehabilitation in post-stroke? Good question. You know, every time I talk about recoveries and people in the first 3 questions Q&A session, they will ask, does it work in dementia? Does it work in Alzheimer's? So if directly doing research on Alzheimer's disease, we don't have it yet. But in our study in stroke and multiple sclerosis and other cases, we observe memory improvement and cognitive function improvement. We can just say that we observe that, but we cannot claim it as an indication for Alzheimer's. So you are very much welcomed, Tokyo friend, that you can buy in the recovery system, then just use your assessment tool to assess the memory function, cognitive function in the Alzheimer's disease patient. Then we can have more data to support that. Yeah, you can. Yeah, in my video here, bottom left corner have my email. We can email and talk. Hi, Kathy. Kathy asked, do you connect the Recoverix franchise around the world? Oh, yes. Recoverix franchisee, we have a worldwide WhatsApp group. So franchisee has all the best resources to support and to ask questions. Like let's say in the WhatsApp group, from time to time, there are some partner ask about, does it work in ALS? Does it work in ataxia? So, and does it work in cerebral palsy? So that's why in the talk I mentioned these diseases. It would be good to have a researcher to buy in the recovery system, then just change to another disease group, then to evaluate the outcome using your assessment tool tailor-made for this disease group. Then can have more data to support. It's a worldwide contribution to this recovery indication. So why not? Thank you, madam. Thank you for joining. Tom Max, great answer. Thank you. Oh, thank you, Tom Max. So thank you for all your nice questions. It makes a presentation and give and take interaction more complete with question, and it's more fun. So thank you for joining and listening. And now let me welcome our next speaker. Our next speaker is Dr. Winston Chen from Hong Kong, Haven of Hope Hospital. Dr. Chang, please accept my invitation to become a panelist, and so that you come to our panel. Hello, Winston. Good to have you, and I make you co-host so you can also share your video. Hello,
Okay.
Winston. Good to
Hello.
see you. Hello.
Good afternoon, everybody. Good afternoon, Christy. And so, may I introduce myself first?
Oh,
Actually,
yeah,
I
sure.
am the physiotherapy in charge of Haven of Hospital in Hong Kong. My hospital is a rehab hospital serving having about 6,000 rehab bed. So our hospital mainly is an extended rehab hospital, serving the stroke rehab, post-fracture injury rehab, and also some amputee rehab as well. So today, I have 1 hour of presentation, Chrissy?
Yes, you have.
How long
you
I
have.
have?
You have.
Okay,
You
okay.
have 1 hour.
And that's good
Enjoy
enough.
your talk. Yeah,
So
yes.
this is a good chance to share my hospital usage, the clinical evaluation on the usage of recovery X on the upper limb rehab in stroke patients. So may I share my screen first?
Yeah, you can share.
So let's check. Okay. No, so may I need to open it? Just wait for a while. Yeah. Okay. So. Okay. So can you see? Can you all
Yes,
see?
I can see your screen. Yeah, you can
Good.
put it in the full screen mode.
Oh,
Then,
full
yep.
screen mode. Okay. Yeah. So today my
Great.
topic is I have a clinical evaluation on the usage of recovery in stroke patients in our hospital, mainly emphasis on the upper limb rehabilitations. So this is a clinical evaluation report rather than a very scientific randomized control trial. Therefore, the data present it has some limitation on behind. Okay, so but at least we apply the recovery in the clinical setting in the stroke rehab, and we want to have some clinical evaluation result to see the effectiveness of this recovery, whether it is helpful or it is not helpful. Okay, so the mechanism of recovery is quite relative complex, but I just want to present it in one page diagram. So actually, the recovery, the ECG, the CAP can detect the brain activity or the EEG of the cortex area, particular motor cortex area, and this EEG signal has been detected and collected, sent to the stimulator of our wrist extensor. When the EEG activity reach certain threshold, that we can preset it. Okay, for the weak patient, we can set it in a lower threshold. But for a stronger person, you can set it in a higher level so that it becomes more difficult to trigger the FESC. So when the first hole of EEG reached a level, it will switch on the functional electrical stimulations from the machine to the wrist extensor. And then the patient will follow the actions shown on the screen to do the active wrist extension exercise, so that this peripheral training can further reinforce the central activity of the motor cortex. And we hope through this recovery X and the both central stimulations, central training, and the peripheral training altogether can link up the bridging between CNS and the peripheral vascular system and enhance the recovery. So how we carry out our evaluations. So we have defined some inclusion criteria for our patients. They must primarily diagnosis of stroke. They must cognitive sound and able to follow command well, and also follow our treatment protocol of recovery. Because this is quite a cognitive demanding exercise. It needs the patients to have a very good concentration, follow the activity for nearly 1 hour, okay? So cognitive is the must for these patients. And the patient should be able to keep sitting upright for at least 1 hour, because this is the working position for the patients to start with. And they should have some level of control in the affected upper limb. We exclude the total facet case because we find if we apply it on the facet case, it will have a they will have great difficulty, and the result may not be, it will affect our result. So we start to choose some patient with some certain kind of control. That means at least grade 2 in power in any part of the upper limb. So they should have no contraindication to functional electrical stimulation, such as if they put on the cardiac pacemaker, they have some built-in deep brain stimulator, or some they have been undergone cranial surgery, then they will be excluded from this study. And the other exclusion criteria is they should be cognitive sound. If they are not, they are unsound, say they are dementia, or they can't cooperate and follow command, then they will be excluded. So what is our treatment protocols? First, basically, for all the stroke patients, we'll provide conventional physiotherapy treatment for 1 hour daily. And this conventional physiotherapy treatment include functional electrical stimulation to the upper limb and lower limb, upper limb ergometer, lower limb ergometer, ambulatory training, and some functional training. And for the patient who receive recovery aids, an additional 1 hour of session of recovery will be provided on top of the conventional physiotherapy program. So, the frequency of the recovery is around 2 to 3 sessions per week for 12 to 50 sessions. And for the recommendation of the company, recovery company, then the optimal session should be 25. But because in Hong Kong situations, the length of stay of stroke patient in our rehab hospital usually only stay for 3 to 4 weeks. So we may not able to complete 25 sessions before discharge. Once the patient is suitable to be discharged, they will stop there. Or in another choice is they will keep on the recovery exit on an outpatient setting basis when they come back to our hospital for further treatment. Okay, so what outcome we are going to collect? Include the upper limb active range of motion, the upper limb power in terms of Oxford scale, 0 to 5, okay? And the hand grip of the affected hand, the modified Ashworth scale, a scale measure for the spasticity of the elbow flexor and the elbow extensor, and also motricity index. This motricity index range from 0 to 100. And it is indirectly proportional to the upper limb power. The better the power, the higher the motricity index will be. If a subject score 100, it means it is a full power, and all the joint of the upper limb is have grade 5 in power and good control as well. So the study period, we start our recovery exit from on October 2022, and up to now, we keep collect the patient status to this November this year. And the total number recorded should be more than 22, but the completed is 22 now. So the average session per patient is 16 sessions for average per patients. They vary from 5 to 27. For the patient background, the mean age of our patient is 59. It ranges from 35 to 80. For the gender ratio, man to female ratio is 14 to 8. For the affected side, left and right hemi is 8 left hemi and 14 right hemi. For the stroke type, 30 of them is infarct case and 9 of them is ICH, the beating case. So now let's look at the outcome about the active range of motion. For the active range of motions, including shoulder flexion, shoulder adduction, elbow extension, wrist flexion, and wrist extension. We have pre and post comparison and the mean differences. And all the changes is statistical significance. That means all of them, after the recovery treatments, have significant improvement in all range of motion, including shoulder, elbow, and wrist. For the power, you see there's overall improvement in the muscle power in shoulder flexion, abduction, elbow flexion, extension, wrist flexion, wrist extension, and the power grip as well. So the power grip is has a significant improvement. For the recovery, the mean is 1.66 kg, okay? But after the treatment, the mean increased to 4.56 kg. So this is a very significant improvement in our experience of upper limb rehab in stroke patients. And all the power on over the shoulder, elbow, and wrist has been improved and reaches statistical significant level. So how about the spasticity? We found this not statistical significant changes in spasticity, even though patients receive the recovery X. But how about the motricity index? As what I say, it is related and closely proportional to the power change of the upper limb. So there's a significant change in the total score from 39.14 to 49.36, and it is statistical significant. So apart from the objective assessment of the clinical outcomes, we have sent out some Chinese questionnaire for the client to see their perceptions on the treatments of this recovery X. So say we have asked 3 subjects to complete the questionnaires. This questionnaire includes 7 questions. First is, do you think it helped you in your upper limb recovery? And the second question is, do you think it is helpful in your upper limb recovery? And the third question is, do you think it can help to improve your muscle tone? The fourth question is, do you think it helps to improve your active range of motions? The fifth question is, do you think the recovery can improve your mobility, ambulations? And the question 6 is, do you think the recovery can improve your quality of life? And the last question is, do you think the recovery X treatment is acceptable to you? So the mean is range from 2.33 to 2.77. So it is in between the client seems acceptable to the, have favorable feedback on this new treatment modality. And how about the therapies? Let's have a look. The therapy, the question is, do you think recovery X can be helpful in stroke rehab? Or do you think it can help improving the manpower? Do you think the recovery is a manpower demanding treatment modality? Do you think the recovery is user-friendly on usage? Do you think the recovery is safe treatment modality? Or would you recommend to others therapists when other therapists have tried to use it? So the range, the mean range varies from 1.12 to 2.75. And so we have some subjective feedback. Say, for the therapists, they think the advantage of recovery is it could be applied on facet affected lane, even though the limbs have no power at all. We can apply the recovery to enhance it. Okay, so this is the advantage of recovery, and it is easy in learning how to set up. So the setup is not difficult, and it provides visual cue for the patients to guide the patient's upper limb movement. But the disadvantage is the treatment time was a bit long. It need 1 hour for single treatments on top of the conventional. So it's a bit time consuming. And also the regime is a bit repetitive. And sometimes some patient will have feedback if that machine includes some interactive game with patients in order to make it more attracting and fascinating. Okay, so that it won't be that boring for the patient. So as a result, we have done a recruit and complete 22 subjects. The average number of sessions per subject is 16, and the improvement is very significant compared with the baseline. And they both the active range of motion in all shoulder, elbow, waist, the power of shoulder, wrist, elbow, and power grip has been improved. The motility index of upper limb has been significantly improved. However, there's no improvement in the upper limb spasticity after the treatment seen. So as a conclusion, we found that stroke patients undergone recovery X have positive improvement in promotion of upper limb functions in terms of active range of motion, muscle power, power grip after stroke. The recovery X, however, has no improvement in upper limb spasticity. But however, our clinical evaluation subject number in our study is small. So if we can carry out further RCT study with larger number of subjects, it's deserved to perform in future. So that is the end of my presentation. I think it's a bit fast, right? It takes 22 minutes already only. But
No worries.
I think we have time and plenty of time to have discussion. And in case in my presentation I go too fast, somebody want to have some more discussion or clarification, it is welcome to do so.
Thank you very much, Dr. Chen, for your nice study and presentation. And it's also fascinating to see a clinical evaluation in a hospital setting because it's kind of more real life. That yes, the patient usually would only stay in the hospital for like 1 month. How does it look if the standard protocol 25 sessions cannot be completed? Does it still beneficial to the patient? The inpatient is a good reference for hospital physiotherapist team to reference it also. Dr. Chen, I have a question. These 22 patients, they are subacute patient or chronic patient already? I might have missed
Actually,
that.
internationally, the definition of subacute is around the acute definition of acute stroke is within 3 months. Okay, and subacute is 3 months to half years. Okay, or 1 year, and chronic is more than 1 year. But in Hong Kong situation is a bit different. Although we are extended rehab hospital, suppose we are going to provide rehab to the subacute phase patients, okay? But in Hong Kong, actually, for those stroke suffer from stroke who are admitted to acute hospital, once they become stabilized, say within few days or 1 week, if they are medically stable, they will transfer to our hospital to start the rehab. So, if in that sense, most of our patients is in acute stage actually. Here with reference to the international definitions. Most of
Thank
them
you.
are acute phase patient. Yeah.
Great. Thank you for this supplementary information, because I know there is a friend hospital in Singapore also want to look into the acute stroke patient data. So Dr. Chen, you have done it as well. Then I can also promote your study to the region. Then they can use it as a good reference start as well. Thank you, Dr. Chen. Let's see the Q&A chat. Oh, there's a attendee asking about the cost of recovery. Let me answer you later. Any questions for Dr. Chen for his first clinical evaluation study using recoveries in acute patient, sample size 22. And because the system in Dr. Chen's department is upper limb treatment only, yeah, as I can recall. And back to your question, Dr. Chen, that patient feedback, does it have a more fascinating game, right? The upper limb treatment, we have added a slap a rat game that need to imagine rising up your hand to slap away the rat. The rat coming out is a visual cue to replace the green arrow. Then the rat coming out, sustain your imagination higher, your hand to slap away the rat to protect your cheese. If the patient didn't sustain the motor imagination, one piece of cheese would be stolen.
Okay, that's good.
Yeah.
That's good. I think it will be more attractive for the patients. But I'm not sure whether it's attractive or distractive. Sometimes we demand the patient to be quite concentrate on the thinking of the actions, and so that to promote their EEG, appropriate EEG productions.
And if I may also share a study when we kind of change this gamification, we have also launched a study to evaluate the change of gamification on the patient outcome of recovery. So let me quickly take this paper out. The game gamification effects of BCI gamification.
Chrissy, I see some questions on the poster.
Yeah, this paper showed that even though we changed the gamification, the efficacy is still as good. Let me check the Q&A chat again.
Okay, good.
Right. Tom X from Tokyo is asking, would it make sense to continue rehab or intervention at home with some home-based application of recoveries? Yes. What do you think, Dr. Chen?
I think it's a good idea to have some training, home-based training, because neuro rehab, the essence of neuro rehab is the intensity of treatment, and the repetition is very important. If home-based treatment can be introduced, it can increase the intensity of treatment and enhance the spasticity. That means the return of the upper limb recovery. But the problem is the recovery exit is a kind of sophisticated machine. It needs specialized training and professional input and supervisions. So it will be a bit difficult to be applied in the home setting, except we can find some way how to make use of the tele technology to transfer the visions and the visions, the video through the internet to the home. And also we have to face the difficulty to how to detect the home-based patient's EEG and transmit the signal back to the center. So it is technologically, I think, is feasible, but we have to deal with a lot of technical problems before we can apply the recovery X in home-based setting. Christy, do you think so?
Oh, and then I have further on this question to Dr. Chen, you as well. Because yes, some stroke patients, they are more stay at home. They are less easy to move around, to go to the clinic or even for outpatient service. Would Havenhoff Hospital or like you have more intelligence from the hospital authority, would any Hong Kong public hospital kind of would establish a outreach team to bring like neuro rehab service to the stroke patients home? Do we have any planning in that kind of?
Yes, sure. In Hong Kong, we develop a lot of tele means we now have tele rehab, so we can make use of tele medium to have some home-based treatment and home-based exercise training, and we have devised a lot of exercise video. The patient can get the video at home and follow the exercise video to do the exercise. And also our center, we are quite promoted and we are quite familiar with a kind of therapies. Means mirror therapy. Okay, mirror therapy. Mirror therapy is a kind of simple treatment approach that once you teach the patient to do it in your center, then you can tell them to go back to home to carry on the mirror therapy by themselves. So it's not as sophisticated as the recovery X. Patient just have a mirror at home, simple mirror at home, and they can perform it by themselves, follow our exercise training regime. So it is quite effective and quite powerful. But concerning patient of Thomas, he is asking whether we can apply the recovery in home-based setting. And up till now, I think it will be rather difficult. But I encourage a lot of home-based treatment instead of always come to our center, because the patients only say, even for the active one, we only can provide 2 to 3 sessions per week. That's the most of our treatment sessions. But the patient actually can do a lot of rehab training exercise in their home setting environment. It can definitely can be quite helpful and effective in promoting their recovery, return of the operating function.
Thank you for the information about how the public hospital is establishing the outreach service for the I would say less techy treatment to bring it to the home. Just say additional information, the recovery service in the private setting is already providing outreach service. So yeah, at 5 p.m. today, Hong Kong time, Will would also share with us more about how he has been providing the outreach services as well. So because recovery is actually quite portable, like me, very short, just 5 feet 5 feet tall, with a hand carry luggage and also a computer bag, can also bring it to the home for service. And of course, yes, different home, different setting, different furniture material, the recovery operator would need to get trained to get to know how to put the setup more properly to adapt to the home setting. But yeah, it can be done and it can be excelled with practice. So yeah, it could be a good additional service as well. Right, then let's see the other questions. Someone asked, could you give a summary on your pipeline of recoveries? I'm not sure what pipeline you are referring to. Is it development pipeline or the research pipeline? If for development pipeline, we have already integrated lower limb treatment into recoveries, and for the research, there are more neurological disease that we are doing research on. So I hope that partially have answered your question. Thomas from Tokyo is appreciating Dr. Chen your answer on his
Thank you.
question
Thank you.
for the home-based treatment. Any more questions for Dr. Chen? So, Dr. Chen, in your presentation, you mentioned about maybe the next stage of the study would make it into larger scale or make it into RCT. So what is your target sample size if it is a larger scale? And if it is RCT, what would you compare it to?
Well, I think we have to, for the subject size, we think with reference to our present study, I think 20 something is good enough because the effect size seems up to medium already. So if I try another RCT, I think 20 more control groups, totally 40 something should be good enough if we in order to get a significance level, okay? And for the RCT, because we in this clinical evaluation, we can only prove the change, compare the before our recovery X and post recovery X changes, right? The changes may not solely due to recovery X. Of course, I believe it has great contributions, but we can't delineate the lateral recovery the effect of lateral recovery first. And the second is we provide the conventional intervention as well on top of recovery X. And these two factors can contribute to the improvement and not solely by recovery X. So if we really want to delineate the contributions, whether the recovery of the upper limb function is due to recovery X or due to natural recovery or due to unconventional therapies, we need to do another randomized control trial. At least one group only received the conventional therapies. The other group received the conventional plus recovery X. So when you compare the group between group, if there's significant differences, then we can have confidence that the additional gain in improvement is due to the recovery X itself rather than other factor. So this is why we need to do our CT in order to have a clear picture about the sole contribution of recovery X.
Right. Thank you for sharing your RCT idea. Many therapists are asking for it, the RCT data. But yeah, in my presentation, the clinical study that our Austria team has done, it is the patient, they are in chronic stage. Most any stage they would come, but most of them are chronic patients. Then in the chronic stage, they didn't do any treatment already, then they just come to a recovery clinic to receive the recovery treatment. But in your hospital setting, they are actively doing different exercise. So yeah, it's all natural and makes sense to compare. Like if just a conventional physical exercise, on top of it, add recoveries, as it gives significant patient outcome, further improvement. It's also interesting data to see.
But I have to
as well.
say that in our clinical observations, the patient receiving recovery X on top of conventional, and we see those group will have a greater improvement when compared with our conventional. But this is, I mean, a very subjective observation. But we need to have an RCT and more objective data to support for our observation. Yeah.
Great. Thank you for this additional information on your observation. Happy to know that science is always give us better insight on what to do next, what to investigate further. Happy to have local Hong Kong data. You know, all these 3 years I've been talking about Austrian Europe data. Finally, we can share something from Hong Kong. And
I think there is
yeah.
another center who will, I mean, in Hong Kong, there is another center who will, I mean, purchase your recovery and have a clinical study. They will start an RCT in future. So in coming future, we can have a more objective data that can be presented in out of Hong Kong setting. Yeah.
Great, great. So it is going to be a multi-centered RCT with
It should be a
another
single center first.
single center first.
Yeah,
but
the
RCT in Hong
center
Kong.
is, I mean, collaborate with an university. So they will join effort to perform the study on the recovery, yeah, effective depth of recovery six, also on the upper limb rehab in stroke, yeah.
Great, exciting. More reference data from Hong Kong, more stories from Hong Kong to share with others. Yeah. So from the audience, any other questions that you are interested? I know some participants interested in installing recoveries in your hospital or bringing recoveries in your country. Feel free to ask Dr. Chen his experience and his observation. Yeah, don't be shy. Just ask.
Maybe
Right.
I have some highlights if there's no questions.
Yeah. Presented.
You can see one of the drawback is actually the period of our study is lasting long. And from
1 year.
October 22 up till now, we only got 22 patients completed. The number of subject recruited is a bit small. And actually, when you find the subject for this recovery treatment, you have difficulty, great difficulty, because the prerequisite of this application of the recovery is a sign cognitive function. So in our acute phase, patient with acute phase, and the elderly age group, most of them has a problem in cognitive functions. So not many of them can have a sound cognitive function to follow and participate in the recovery treatment. So in my experience, before I did neurotherapy therapies in stroke rehab as well. So we screen 500 patients. Only 10% of them could have sound cognitive function to participate in the neurotherapy program. So this is the same proportion, similar in our clinical evaluation in the recovery. So that's the limitations. When you want, even recovery is a good treatment modality and approach to enhance the recovery of upper limb in stroke patients. But the prerequisite is patient must have cognitive, sound cognitive function and follow all those instructions, and must be very concentrative and motivated as well. So cognitive function is a great limitations in the application of recovery, I think. And it leads to difficulty in subject recruitment as well.
I see. Thank you for this additional information. Yeah, the brain-computer interface principle of these recovery is really the motor imagination task that yeah, we extract the event-related synchronization to trigger the VR and FES. So being able to do the motor imagination, understand basic instruction is a requirement. Yeah, it is also a frequently asked question about the motor imagination. I remember the doctor asked me, what about the patient cannot do motor imagination well in the beginning? But because I have also done demonstration in patients for quite many times, and even within the recovery session, that 1 hour, we have 3 parts of run, right? In the calibration, maybe the patient would achieve like 61. Then we have generated a report. We see that the calibration is not done very well. It's her first time in his life to do recovery. So we repeat the calibration. And even though in the second calibration, we also see improvement in the motor imagination classification accuracies. It's like going up to 64, usually around that. Then we always end each session with training session. Then we also see a improvement in the motor imagination accuracy at the training session as well. So MI can be practiced, but the prerequisite ability to understand simple instruction, differentiate left or right side, it is something to take note. And also the understanding of the importance of the standard protocol, 25 sections. They understand the time needed to improve neuroplasticity is different among patients, and they are willing to comply 2 to 3 times a week. And Dr. Chen, is really very good that you have designed your study that the number of sessions that the patients on average completed is about 16 session.
15,
yes.
16.
16. Yeah, I think hospital therapists, yeah, really in the inpatient setting, they are interested to see like just 1 month, how much improvement can recovery additionally bring to the patient. So yeah, although the sample size, the recruitment of patient take a bit of time, yeah, need to understand basic instruction. But helping one patient is one patient. If without this tool, even though those 10% might not enjoy, may not benefit with this significant. I remember your result table, even the power grip, highly significant. That's fascinating to know. Yeah, in 1 month, in those 10% patients, they have the evidence-based hope that in your Haven of Hope Hospital physiotherapy department can have this good result if we add 1 more recovery hour on top of the traditional training. There's also something that your department can especially offer to the inpatient. Yeah, so yeah, technology keep advancing, right? Like let's say 10 years ago, nothing like this existed in the market. Yeah. So it's already a good start.
So
Thank you, Dr. Chen.
I want to add another remark.
Sure. Yeah.
The advantages of this recovery because in the traditional rehab approach, for patients suffer from very flaccid or very poor upper limb functions, we have not much treatment modality or choices to deal with them. The conventional thing is we do passive movement. We only can do functional electrical stimulations. That's all. Okay. But now Recoverix is another a novel treatment modality that can enhance the patient's self-initiation of the motor cortex activity, because they can make you train up their EEG to exercise the motor cortex area to activate the cortex of the damaged lesions area. So traditionally, we make use of peripheral training to put a sensory input to enhance the neuroplasticity of the central nervous system. But now we can make use both central stimulation. That means we train up the motor cortex activity, and also make use of peripheral training, the FES of the peripheral limbs. And also we make use of visual feedback that is have similar effect with mirror therapy or imaginary therapy. So we add three things together with the use of Recoverix. Hopefully we can enhance, in that sense, we make use of central stimulation, peripheral stimulations, and imaginations or visual stimulations to enhance the whole process of recovery. So that is the beauty of that. I'm not promoting that the particular brand of machine. I am trying to tell the concept of we have nowadays is not just as our old days make use of peripheral training. We can think of a lot of choice and stimulation. We can make use all those of perspective to enhance the recovery. Yeah. So nowadays, even for the very flaccid upper limb or the very low upper limb functions patients, we can have a lot of choice. Even we can't do a lot of peripheral training, we can make use of a lot of central stimulations like the EEG training or the mirror therapy or RTMS. All those things is a kind of central stimulation that can enhance the return and the reactivity of the CNS system, particularly over the cortical area. Yes. That is
thank
what I
you for
want to share.
Thank you for this additional highlight. Yeah, is a fair and objective evaluation on this novel therapy. Yeah, it is worldwide available for like 10 years already, and Hong Kong, as always, can adopt new technology very fast, and good that Haven Hope have it and get it evaluated. And maybe like let's say the Hong Kong public hospital setting. I'm not sure that establishing neuro rehabilitation department in a public hospital, would that be also a direction that you know better than me? I'm just guessing that, because I see in other countries they already have a neuro rehab department in the hospital. So I'm wondering if Hong Kong we have this direction as well.
Yeah, sure. Hong Kong public hospital setting has been trying to build several very specialized neuro rehab center. And at which they have concentrated all those high technology, high specialized training personnel in that center, so that it can hopefully promote the efficiency and become more cost effective, particularly for those enhance the recovery of, particularly for the young stroke, because the young stroke, the need is much more than the elderly. They
they're
face a
much
lot
more
of problems.
desperate.
Yeah. They need to to handle with their social problem, their vocational problem, their entertainment problem. So for the young stroke, their need is and their expectations much higher than the geriatric group. But I mean the need is, I mean the service we provide is far left behind from their need. Yeah. So we hope we can have a better improvement in that sense, with the
Yeah.
establishment of specialized rehab center for the younger stroke. Yeah.
Yeah. Oh, Dr. Chen, let's work on it together, like promoting what is the available technology in shaping the neural rehabilitation service that is can be provided in Hong Kong for Hong Kong patients. It will be good, like just educate the professional and like let people know that there's already technology available and more education program integrated together can shape this neuro rehab service in a more systematic, in a more structured way. One more curiosity. I have a question. Because I also observed like in physiotherapy, that like let's say in Australia, I see they have distinguished two streams, neurophysiotherapy and
Musculoskeletal.
yes. So in Hong Kong, in our like qualification training, do we already shaping into like differentiating different streams already?
It is a worldwide development, I think, not only in Hong Kong, Australia. Physiotherapy usually after the graduation, they will specialize in several fields. The first is musculoskeletal or sport rehab. Okay. The second is neurological rehab. The third is cardiopulmonary rehab, and the fourth is pediatric rehab. So they may develop themselves after graduate. It depends on their work nature and their interest to which field they like to develop. So in Australia, they are very strong in musculoskeletal and sport rehab. Yeah. But you know, every place, every country have neurological patients that need to be rehabbed. So definitely they should have physiotherapy who are interested in neurological rehab and they become specialized in that field. But I have to say that although they specialize in that field, but they are qualified physiotherapists, so they can entitle and to do whatever they as a physio can do. Yeah. But of course, if they are specialized in musculoskeletal or neurological, they will be more familiar with that field and have expertise and very good experience about it. Yeah.
Great. And may I know what is the percentage of the physiotherapists that would choose the
I have
neurological?
no idea. I have no idea. And in Hong Kong, we have up to now in Hong Kong we have 3,000 physiotherapy already. At the time of my graduate, at the time very long time ago, of course, there in Hong Kong only 600 physiotherapy there, but now it's 300,
Expanded a
3,000,
lot.
3,000 already. But it's still left behind. I mean, compared with the worldwide ratios, the patients to therapist ratio is still very high. And I think in the future, long way, both locally and worldwide, the demand for physiotherapy is still quite huge because our group, our group is become aging in all society. And the demand for the elderly service is quite huge, yeah, in future.
I see. Yeah. Thank you for your insight, like in the physiotherapy field. Yeah, maybe I can consider to learn. physiotherapy as well to help the market, the aging ratio. Right. And oh yeah, because a relative of mine also take a physiotherapy course, and he just want to take care of his mom better.
Yeah.
So yeah, it's out of love, out of more personal caring caregiver. So it's very heartwarming to see someone taking physiotherapy course to take care of family. All right. Thank you, Dr. Chen. And thank you for this very nice first in Hong Kong clinical evaluation on this BCI VR FES novel therapy in a hospital setting in the acute stroke patient to see a good result, and looking forward to a larger sample size in RCT, and very happy to collaborate together to enhance the neurological physiotherapy education so that more healthcare professional and neurological like proficient caregiver can be available in the market to serve the patient. Thank you very much, Dr. Chen.
Okay.
Hope to see you
Nice to
in
meet you
person
all.
soon. Yeah, thank you. Thank you. Thank you. We keep in
Bye.
touch. Thank you, Dr. Chen, for your contribution. Thank you. Thank you.
Okay.
Thank you. Bye-bye. Thank you. All right. I hope everyone have enjoyed the talk of this morning so far. Don't get me wrong, we don't have a lunch break. I'm just doing a few clicks to prepare the next topic. This is ultra high density. Let me share screen. This is the next topic is ultra high density EEG, G-pangolin research applications and unicorn hybrid black research and education. And it is me again on this topic. So for BCI research, there are different needs from different researchers. Sometimes budget concern that they are more bound to 8 channel or even less. Some already used it a lot of time, the 32 channel EEG, and then want to have higher number of channel to have better spatial resolution to look into more specific region of interest in the brain. And there are currently like from 4 channel to like 128 channel. There are quite a lot of choices in the market. But then the g.tec, of course, we see the need of higher spatial resolution and need an optimized system that not too difficult to assemble, but can break through the spatial resolution limitation. And like 2 years ago, we have developed a very ultra high density EEG. It's not high density. High density means like 128, 258, that is high density, but we are ultra high density. So let me share with you all about from 8 channel unicorn to 1024 channel ultra high density EEG in their research application. Let me start from lower number of channel, 8 channel. See my screen. Okay, so this unicorn, you see, it looks very cool, right? Black fabric and nice LED, and the EEG electrodes are here. Standard 1020 system montage, 8 channel on the EEG cap. This is a easy to use beginner grade unicorn EEG headset that pick up the EEG signal through this G-Sahara hybrid electrode. that can be used with or without gel to pick up the EEG signal and transmit to the computer through Bluetooth. We have a basic package of the unicorn that with the medium size cap, with 8 hybrid electrode, with the housing, with the Bluetooth dongle, and the C and .NET API included in that basic package. And we also have a naked version of Unicorn for brain computer researcher, brain computer fanatics that you really want to develop your own housing, your own very high tech look helmet, whatever design you want. You can also use this naked BCI to develop your own housing as well. This is the specification of this unicorn system. It can acquire high quality EEG data and process it in our application. The signal to noise ratio is high, 24-bit resolution and 250 hertz as the sampling rate. We do 4,000 times oversampling to increase the signal to noise ratio. The G-Sahara hybrid electrode, we can use it dry or use it with gel inserted in the hole of the hybrid electrode. The position of the EEG electrode is fixed in this unicorn headset. Fixed as the standard 1020 8-channel montage. And we have different API available for you as an add-on as well. It is trusted by many research institutes. And every year, because we also organize brain-computer interface hackathon in our spring school and also in IEE SMC, that there are different institutes in the world that is interested to become our hosting institute locally for the hackathon event to take place and give local support to the participants. So yeah, like University of Melbourne and of course our Austria office is always the hosting institute. And hopefully in Asia Pacific, like maybe next year, Japan, we would have a hosting institute. I hope so. Let's see. Will this happen? Yeah, Unicorn, we have patented design, especially for our hybrid electrode. We have done several projects with this unicorn device in our hackathon event. This is one of my favorite. This BCI g.tec machine. Like you see, 2 weeks ago in Europe, we also have a hackathon group that have done a BCI bartender service. Like use the BCI, use the unicorn, and then with a robotic arm, then based on the band power frequency to see how alert or how tired, how calm the subject is, and then use the band power frequency like feature control signal to control the bartender robot to add in different ratio of gin and tonic water, and then the robotic arm would shake it, then serve you the gin tonic. Yeah, so this one make use of the band power frequency analysis. And this is a BCI art project. The researcher group has done an algorithm to convert the EEG signal frequency into like different color or different line pattern, and then draw it on a LED wall. This is also a brain painting project that use BCI device to control robotic arm to draw painting on a wall. This unicorn, like let's say you make use of the naked BCI unicorn, then you can develop your own housing, then make it more interesting for the, let's say for the children, for the kids to wear it on. Then you can maybe develop some BCI game to train up their concentration or their attention span. We organize hackathons every year in spring and in fall. In spring, that would be in our spring school. Brain.io, this one is our website. You can always check our latest dates for the upcoming hackathon and join it online. In the basic package of the Unicorn Hybrid Black, the basic application and API are included. The Unicorn Recorder, that is to visualize and record the EEG data in .csv format. All the raw data you got access to. You don't need additional subscription, whatever, to our software. No expensive subscription plan for you to get access to raw EEG data. You have all the raw EEG data for your processing. And standard package, we also have .NET and C API included. Unicorn band power, it is the real-time band power analysis application that I will also show you in the demonstration later. We also have UDP and elder cell streaming and transferring of data. These are the add-on. Unicorn Speller, remember in my first talk that we make use of the P300 BCI principle to focus on some splashing word. And then after calibration, you can focus on the flashing word and then spell it on the screen. Unicorn Speller will flash the characters, the icon with celebrity faces to make the accuracy even higher. Unicorn Blondie Check, it is a neuromarketing application that the researcher can load in their own picture set and then evaluate each picture that the triggered response in the subject in evaluation with the visually evoked potential, and then the picture set would be ranked picture by picture, which one is the most trigger the most obvious response from the subject. Simulink interface, Python API is also a popular add-on by the researcher. Unity interface is something new, like since last year, we have this Unity interface that the researcher can easily overlay some code-based flashing item in 2D or 3D games to develop some BCI games. Yeah, it has many many applications for digital arts. Let's say some tech art museum, like let's say M+ from Hong Kong or Team Lab from Tokyo, Ars Electronica in Europe, and also make use of this unicorn, and then maybe every visitor can contribute like 30 seconds of the EEG, then collectively make a brain art and then print out a postcard to bring home. Just my random idea. If you are interested in making the tech art museum more interesting, we can discuss. And of course, I can line you up with our unicorn researcher to make it happen. Yeah, for students, Unicorn is also a good tool to have a first taste of how BCI works. I have done a demonstration of Unicorn in a special need in a, why is it jumping? In a special need school in Hong Kong. Those special needs children, yeah, they have difficulty in formulating words. Then the teacher want to use the unicorn speller to help the students to say what is in their mind. Yeah, so the application can be creative and useful to benefit different user groups. Unicorn Education Kit, we have 2 kits. Big kit is 8 standard set of hybrid bag. Small kit is 4 standard sets of the Unicorn hybrid black. Yeah, and depends on your class size, dear lecturer and professor of university. Depends on your class group size that you can consider having the education kit to facilitate your teaching on BCI concept and encourage and guide the students to use, let's say, Python API to develop some interesting project. And those drones are getting popular nowadays. You know, we have a lot of drone show at the Victoria Harbour and also in Singapore, in Shenzhen also. What about BCI drone control? It would be very interesting for even junior high school, junior secondary school, or even primary 5, primary 6 student. They can also have a taste of BCI already. Yeah, so this is how it looks. One box for one set, and 4 box is a mini education kit already. Yeah. And it's also gaining popularity is BCI gaming. That use your mind to play game. That's also fascinating to try it out. This is my boss again. Upside down. Oh no. Let's turn our head a little bit. So you see, boss is wearing the unicorn hybrid black. And in the game design, we use the keyboard, the arrows to control the avatar to move. And then the subject focus on the flashing code-based visually evoked potential flashing item to break down the obstacle and then collect the coins. Using the Unity interface that we have developed for game developer, you can easily overlay this 3D code-based flashing item into your game design. There. These are some examples of the games that we have developed by our Austria researcher team. This is a Brain Buddy. This is the space avatar coin collection game that my boss demonstrated. This is the 食鬼啊，英文係咩？食鬼嘅 game 啦。Also focus on some flash-based flashing item to control the ghost to move around. This is how it looks for the Unity interface. It is quite easy to overlay it on the game. Just drag and drop the flashing item into your gaming. This is how it looks for the 3D Unity interface. We have a 15-minute tutorial video to guide game developer to use our Unity interface to add this flashing item into your Unity game. So just subscribe our YouTube channel and get this tutorial video. This is a comparison of the median selection time and information transfer rate of ERP or CVEP principle while using dry or wet hybrid electrode on the unicorn. So yes, we see wet with wet electrode like added gel would be a bit better, but depends on the subject group, depends on the willingness of getting gel. You can also reference the time and the transfer rate here using dry. Some gaming integration ideas. You can use short-term focus. This principle, like just focus on the flashing item short-term, to use it as a button press. Let's say you can use short-term focus on the flashing item, use it as a single selection to trigger something. Let's say to some gunshot, explosion, open doors, all this. You can use long-term focus, like hold the button, that kind of concept. Use long-term focus, focus on the flashing item to do multiple selection. Let's say like focusing longer on that flashing item, then maybe the higher the elevator will go up. All that just being creative. Yeah. This is spring buddy. How about let me do the demonstration for you. That would be more fun to see me doing demonstration. Stop sharing and let me choose. Then you see, okay, you look at me first. So this is the unicorn hyper black, very handy, lightweight, 8-channel assembled here, standard 8-channel montage, hybrid electrode made of conductive polymer, and there's a hole here, optional for you to add gel or not. Then of course there is a power button. Remember to turn it on whenever we do a demonstration. Right, so I put it on. We have also different cap sizes, small, medium, large. The standard one included in the package is medium. So you have your own choice to add a small or large size. This is the chin strap, so put it on your chin. Then there were 2 hanging electrodes here. What are they? So there are 2 reference electrode here. So I peel off the liner and stick it on my nostril, the bone behind my ear. There you twist the hybrid electrode a little bit to make sure it is touching the scalp properly and standing perpendicularly like this on the scalp. Right, let's look at the software. Okay, here, Unicorn Hybrid Black, double click it. It is our latest version. Skip it during live event. My unicorn, go to connect with my headset. The serial number of my headset now is 50. So 50 with a tick connected. Then go back to the apps. Let me show you the basic application that by default included in the standard package, Unicorn Recorder. It is our recording, visualization, and recording software for Unicorn. And in this application, the latest version, we have added the most sought after function, Oscar. You see here, Oscar. Check it, then you have it. Remove all the artifacts in real time online. Right, then just a little bit of fine tuning. This 100 microvolt is better visualizing the EEG here. 0.5 to 30 or 50. Let me show you 0.5 to 30 for EEG, not for Hong Kong, filtering out 50 hertz. Let me show you how it looks now. Yeah, it looks good now. And now let me move. And you see all these artifacts, right? And it's annoying for the researchers. Annoying, especially during the data analysis. Then let me keep moving. And then I check the Oscar. Even though I keep moving, it's all good EEG. Even the eye blinks are removed. Let me uncheck it and let me show you my eye blinker. You see here? I blink my eyes. You see? Blink, blink, blink. On channel 1 and 2. See again, I blink now. You see this one? Blink again, blink again, blink again. Then when I add the Oscar function, I blink now, blink again, blink. Now I blink I also removed. So it make life easier for researcher. When we click record, it's recording. Then let me quickly show you the recorder file. It is saved locally in the computer, and let me think. Yeah, switch. I would like. Yes, it is today, and you see here your CSV file. Wow, scared me. The 0 in the beginning. So you see all these raw EEG data here. Just a few seconds I have measured. And we have all this data already. Yeah. Then let me also show you the brain power application. Is measuring the EEG already. Let me close my eyes and you guys can see this bottom right corner. Try to see the change. real-time change. And close my eyes, so I'm expecting it will change to alpha. Let me try. There's alpha. Okay, I saw it also. So it's working. So this band power real-time analysis of the different band power frequency, then of course using UDP, then you can stream it and then control other third-party devices as well. This Brain Buddy is the game that we have developed 2 years ago for impaired children that they can play in one pair. One is to control the keyboard, the arrow. The other one just focus on focusing the flashing item to break down the obstacles. This is my device, as you would have known. Instruction. Play with the body. research need that we really can optimize different system for your research. Let's say for BCI gaming, of course it makes sense that for easy assembling of the cap, easily put up the cap on the subject. It makes sense to use just unicorn. But let's say tech art project in team lab, tech art museum, they will not expect themselves to be shaved or gelled. So hybrid electrode tech art BCI gaming project that will be most popular. And what's the use of this ultra high density? Why do we develop it? We want to tackle spatial resolution limitation because there are also some literature already show that the higher resolution, the better to do the source reconstruction and source localization for so we want to one step further, where is the limit? Try to break through that spatial resolution. It is called the pangolin because we have developed this specialized ultra high density evaluation system. This pangolin grid. It is a gold-plated flexible PCB. It's very small size. This small size already have 1/16 channel. Can go up to 256 channels. So we can connect 4 pieces of GIM with 4 pieces of parallel lines. Then what we do is it better? Sorry, I didn't hear anything. Is it better now? Okay, all better. Thank you. Sorry for that. So, back to this pangolin. Yeah, this is Josephine. This head is Josephine. Okay, let me then repeat again. This pangolin grid, very small, already have 16 channel. It is gold-plated PCB. Then when we prepare this grid, you see this liner sticker here. This is labeled grid size. This is no labeling, so that means we peel off this liner and then can stick to the grid like this. We do it on this grid preparation kit. Yeah, like this. Then we would add the alephis paste onto the grid to make it into a sticky electro grid. We put the alephis paste into the syringe. Then we would squeeze the alephis paste into each 16 holes of the grid. Then after that, everyone shall have my name card. Then we can evenly distribute the alephis paste on the grid. Then we can remove the liner on this side. We can remove the liner of this side. Then we can stick it on Josephine's scalp. After sticking the grid on Josephine, we would add this preamplifier, plug on it, and then connect to HDMI cable. Then all the way, you see here, go to this pangolin connector box here. One slot for 16 channel. So one connector box, 16 slot, can go up to 256 channel. We also have a ground electrode here, of course. EEG electrode always need a ground. Stick it on Josephine like this. And here, the connector box all the way go to this g.tec. It is the backbone amplifier of pangolin system. We can also connect 4 pieces of g.tec together. Then all the way can connect to 4 pieces of pangolin connector box to make it into 1024 channel system. Any questions so far for this pangolin? Right. Okay. Then let me continue with the slide here. I have explained how the system is connected. Yeah, so we can skip this slide already. The preparation explained that already. And we have a pangolin montage creator that can add the grid on free surfer MRI scan or on some standard brain diagram to create the montage file, and then to facilitate your EEG measurement, then for your analysis would be easier. This is how high our resolution is. This dark circle here are the 1020 system. The standard clinical EEG, some are 1010 system, the pale gray circle here. Our pangolin, it is this smallest hollow circle. Smallest hollow circle. So the spatial resolution is really another level. This is how we stick the grid on a shaved head, well lined up, and make a montage in our montage creator. Connect with the preamplifier. Then first design your paradigm of your BCI experiment. Then checking the impedance, then do the measurement. We have done several research projects already. This one published it in Frontiers in Neuroscience, that we have decoded individual finger movement using this pangolin system. Like we have the avatar as the video cue, that when the cue is like sticking out the index finger, then the subject would do it accordingly. Then we analyze which particular point region in the motor cortex is activated. Then in the result, we see that we even evaluated different finger movement. It is correspond to which particular point of the brain is activated. You see this thumb, when we move the thumb, it's actually these three dots. The ring finger, it is these dots here. So with pangolin system, we are bringing the EEG research into really another level. We are not talking about which region is activated when we move the finger. We even can see which point in the brain is activated when we move each finger. And the accuracy is high, 70%. In those 32 channel system, that usually the accuracy could be like buffering around 50 something or 60, but with this pangolin, we can achieve 70 accuracy. This project is also interesting because the mapping of the central sulcus, this kind of experiment usually could be done with ECOG, that is invasive ECOG measurement, to find out where is the central sulcus. But we try to use this pangolin ultra high density EEG system to map where is the central sulcus. We do it by median nerve stimulation here. This is the FES stimulator, stimulate the median nerve. Then because we know in theory that there is a phase inversion from the sensory to the motor cortex, and the thin lining that has the phase inversion take place, that is the central sulcus thin lining. So we use this median nerve stimulation, then evaluate the peak and valley at the EEG data. Then we see the phase inversion take place, then corresponding to which line of the pangolin electrode lining. And after we have mapped it, of course, from the EEG analysis, we see the thin lining, and we have co-registered it with fMRI to confirm that it is correctly mapped, mapped the central sulcus. And this project is published in Nature. We also have this tech art project. The tech artist is Anouk from Europe. We have made use of fully utilized the 1024 channel G-pangolin system. You see connecting 4 pieces of GHM with one single computer, 4 pieces of connector box of pangolin, and all the way connect to this model here. Then we real-time analyze the end power frequency to see how alert the model is. If the model is more relaxed, the 3D printed scale dress, the LED would light up in blue, and the scale would move in more rhythmic way. If she is in meditation, it would light up in magenta and the scale movement is lower. If she feels more alert, it would light up in white and hectic movement of the scale. So it's sort of creativity that you can have for BCI Tech Art project. If more clinical use, I would say this pangolin, we are moving more towards using this pangolin system to do source reconstruction, neural source reconstruction, and source localization. For example, like epilepsy. Let's say we are moving more towards neural source reconstruction and source localization for epilepsy seizure onset soon. Because in the current clinical EEG system that the hospital use it for EEG monitoring, the resolution is about 1010 system, and the sampling rate only can go up to like 2000 hertz. Our pangolin system, the resolution, you have the idea already, and the sampling rate of each channel can go up to 38.4 kilohertz per channel. So we just simply get more useful data for the post-analysis of the seizure onset soon. And we are having more interest in Asia Pacific region to use pangolin in the epilepsy seizure onset soon localization. We are doing some pilot study to compare it with standard clinical EEG. In the ICCN conference in September, we have already presented a poster that neural source reconstruction for fibrotactile stimulation on the finger, we can really source locate which point in the brain is activated. So with all these done project, we are getting more and more confident in using pangolin to do source localization for seizure onset soon. I think in 3-4 months, there will be some Asia Pacific data in the epilepsy seizure onset soon localization. Right, so that's what I want to share for this session. Sorry for the brain body music interference just now. And I hope my repeat in the first part of pangolin introduction would help. If you have any questions about this 8-channel beginner grade but good signal quality unicorn EEG, all the way to this 1024 channel pangolin system, yeah, we have all these systems to cater your research need. Any questions, you're welcome to ask. Alexander raised your hand. Yeah. Was it because of the brain body lucid? Anyone have any question? If not, I can also try to show you the paper published with pangolin. So yeah, here. Hand gesture decoding, decoding paper, scissors, stone. Hand gesture. Use pangolin to decode it. And you see here, analysis here, the region, the point that is activated. Individual finger movement paper on frontiers in neuroscience. Baseline 30 seconds, rest 3-4 seconds, and then extend the indicator finger for 5 seconds, and then rest, and then extension. 25 trials, 10 runs in total. Feature instruction for the band power for male and beta. We have 5 subjects in different finger movement. We have actually different activated point or small region, focal region among the subject. And we find out the ring finger paradigm gives the most distinctive activation of the EEG pattern. Then this one, the mapping of the central sulcus. Yeah, stimulate the median nerve using pangolin. Feel free to email me, then I can send you the paper for your good reference. People has been using like 16 or 32 channel EEG system for their research for quite a while already. Why not some breakthrough to see more clearly? It's like a camera. Old camera, like 10 megapixel, everything blur. So this pangolin is like 100 megapixel camera. You just see more things more clearly. Yeah. And recently from Japan, Tokyo, Alaya Corporation, they have pangolin system as well. Yeah, they use our pangolin system to measure 175 hours of EEG data to try to decode speech. Yeah, because the higher the resolution, it really tackles the spatial resolution limitation. You just can source locate different function more precisely, which particular point. Like let's say language related is particularly need more precise mapping, non-invasively or invasively, because language is just relate to like different parts of the cortex. So yeah, your good choice to think about your next research idea. Right. Okay. Feel free to ask me question in the Q&A box. I'll be here to answer them. And next, we can move to the next section that is by Mr. Hanmen Wu from the Chinese University of Hong Kong, Department of Linguistics and Modern Languages. Let me try to find you in the guest list. Yes, I promote you to the panelist. Please accept it. Hello, Henry.
Hi.
Thank you for your time.
Start video.
Yes, you can.
Start my video. Yes.
Great. Good to see you.
Good to see
Thank you for your time, despite your busy schedule. I'm sorry for clashing. Like, thank you for arranging your time to join us live, share with us live.
Thank you for your invitation.
Yeah.
How can I share my screen?
Yes, sure. So Henry is a PhD candidate in the Department of Linguistics and Modern Languages. We have known each other, is it 1 year, 1.5 years? You know,
Yeah,
your teammate Sui Feng has approached me for TMS EEG integrated system. So it's a TMS compatible EEG system for your linguistic research. Very interesting project, and I'm happy that finally I can come up with this Masterclass Hong Kong, and you are like top in my mind that I have to invite you in this masterclass. So yeah, looking forward to your presentation. Everyone, please enjoy Henry's
thank you.
presentation. Thank you.
But I think the combination of TMS and EEG study will come like later, maybe your next masterclass. And for this year's masterclass, we have some studies using only EEG.
Yeah, no problem. Step by step. Yeah, and it is like one step at a time. Yeah.
Mm-hmm. So you can see my full screen, right? So now it's a full screen. Okay, so good afternoon, everyone. I'd like to thank Christy for inviting me here today at the first BCI Neurotech Masterclass Hong Kong. And my name is Hanlin Wu. I'm with the Department of Linguistics and Modern Languages at the Chinese University of Hong Kong. And as a linguistic researcher, I focus on how the human brain processes language. And in most of my research, I use EEG. So in this talk, I'll be giving a friendly introduction to how EEG is used for language research, and I'll be focusing on 3 studies that we conducted recently in our lab for speech comprehension in particular. And as I know, my audience today is mainly from medical engineering, neurology, and neuroscience. So this is an audience. I don't need to convince the neuroscience is interesting. So instead, I'm trying to tell you why I think language research is interesting, and I hope in the end you will have some more idea about language research, and hopefully you will consider doing language-related studies in the future. So let's start with two fundamental linguistic concepts, semantics and syntax. So semantics refers to the meaning of words, phrases, and sentences. So it's related to the content and the interpretation of language. And syntax refers to how words are combined to form larger units, like phrases and sentences. So it's related to grammatical rules. And in psycholinguistic research, we often study how the brain processes semantic and syntactic information by presenting participants with linguistic stimuli that often contain anomalies. For example, a semantic anomalous sentence might be, the trains are sour and very crowded. And you can compare this with the correct version, the trains are yellow and very crowded. So because sour is a flavor, so you can't use this word to describe a train. So it's anomalous in terms of meaning. And for syntactic anomalous sentence, it might be, the child throw the toys on the floor. And you can compare this with the correct version, the child throws the toys on the floor. And so we can see this is related to grammatical errors. So with these linguistic concepts, we can design experiments to see how humans process language, especially semantics and syntax. And here is how a typical psycholinguistic experiment is designed. So in trial-based experiment, each subject is usually presented with linguistic stimuli under two or more conditions. And each condition has a set of trials, and each trial has a specific linguistic item, for example, a sentence or a word. And you might have, let's say, 30 participants in an experiment. And traditionally, if we do by subject analysis, we average their responses across all trials within each condition. So basically, you will have an average response in each condition for each subject. And we can collect many types of responses. For example, we can collect reaction times, verbal responses, or ratings and things like that. And if the responses we collect are EEG signals, then you will get an average brain potential in each condition for each subject. So this is the basic logic of an event-related potential, or ERP. And this method has been around since, I think, around 1960s. So it's a very, very classic method. So basically, you record the participants' continuous EEG signals, and you mark the onset of each stimulus. And in linguistic research, that's, for example, that's each word in a paragraph. And so when you average the waveform, you are actually time locking these stimuli. And I think the beauty of the ERPs is that when you get a smooth average brain potential, those peaks and deflections become components. And these components, based on their polarities and latencies, are given different names. For example, like P1, P2, and P3. And these components are always there in the EEG signals, but because the signal to noise ratio is so low that you can't really see them with your own eyes. And I think the most compelling thing about this is that we have a wealth of existing research on the cognitive functions related to each component. And so when you get this average brain potential and you identify a component, and you have a lot of literature that you can resort to when interpreting the results. And I guess this is why this traditional method is still very useful today, even when we have so many new methods. Right. Now, let's look at some classic experiments. So this is the seminal study published in 1980, and they presented participants with sentences that either ended with a semantically coherent word or a semantically anomalous word. And for the coherent version, it would be he spread the warm bread with butter. And the anomalous version would be he spread the warm bread with socks. And they presented the sentence word by word in text. And they time-locked their analysis to the onset of the sentence final word, which differentiate the semantically coherent and the semantically anomalous versions. And for the first time, they found that the anomalous version triggered this large negative deflection that peaked around 400 milliseconds after the word onset. And they termed this component the N400 component. And since then, thousands of papers have used the N400 as a reliable index of semantic processing in the brain. And another useful index is the P600 component. And this component was first reported in a landmark study in 1992. And in this experiment, participants read sentences that either followed the correct syntactic structure or contained a syntactic error. For example, they had syntactically correct sentences like, the broker hoped to sell the stock. And you can compare this with the incorrect version, the broker persuaded to sell the stock. So you should actually say the broker persuaded someone to sell the stock, right? So at this critical word two, when they contrasted the two versions, they found a large positive deflections. They found a large positive deflection that peaked around 600 milliseconds after the onset of this word. And similar to N400, this component was labeled P600. And this is the first evidence that the P600 is sensitive to syntactic processing. And since then, a lot of studies using the P600 as an index for how humans process syntax in language. Right. So even today, we are still using these indices to understand interesting questions. For example, this is a recent study we did to understand how humans comprehend language when it's from an AI instead of another person. So if you think about it, I've never had a real conversation with an agent that is not an actual person until ChatGPT. I used to speak to AI HomePod and Siri, but you can tell it's not real conversation. It's more like you send a command and they respond, but you couldn't actually take turn to speak like with real people. But today, AI chatbots are everywhere. So I use GPT every day, and people interact with AI every day. So AI agents have become an important type of social actors, actually. So in the past, all language you heard or read are from a real person, but now it's also highly likely from an AI agent. So that got us really curious about whether people's brain responses to AI language is different from their responses to human language. So we did this experiment. We used a paradigm that is very similar to the two classic studies that I have just shown. We presented them sentences that contained semantic and syntactic anomalies, word by word, and we time-locked our analysis to the onset of the critical word in the sentence. So for the semantic anomalous sentences, it would be, after setting the moon, Xiaoling immediately went to sleep. So you can compare this with a well-formed version. After setting the clock, Xiaoling immediately went to sleep. So the correct word, the critical word here is the contrast between the moon and the clock. So for syntactic anomalous sentences, it would be tomorrow, Xiaoming ate a hamburger and he will not eat one today. And you can compare this with the correct version. Yesterday, Xiaoming ate a hamburger and he will not eat one today. So the critical word is actually here ate, and the anomaly is determined by the temporal adverbial yesterday or tomorrow here. And the critical manipulation in this study is that we divided all participants into two groups. So for one group, we told them that we are doing language research. So we have a corpus containing people's real conversation, and we want them to read those pieces of language produced by people. And for the other group, we told them that we collected language generated by an AI chatbot, and we have this interface very similar to Poe. We let them talk to the AI chatbot for a while, and then we said we want them to read pieces of language generated by the AI chatbot. But actually, all the language materials that they read were identical. Right. So we divided the whole study into two separate experiments corresponding to the comprehension of semantic anomalies and syntactic anomalies. And for each experiment, we had 64 healthy young adults. So in total, we had 108 participants. And we collected their EEG while they read the text. And this is the first author, Xiaohui Rao. This is my colleague. And for this experiment, we sampled the EEG using this G.tec amplifier. And I guess this is the reason why I'm here today. Right, so for semantic anomalies, it showed a clear N400 effect for semantically anomalous sentences when we compare this to the well-formed sentences. And this replicated the classic finding. And the key finding here was that the N400 effect was actually smaller for sentences that participants believed were generated by AI, and compared to those believed to be produced by a human. And this indicates that people are more tolerant of semantic anomalies when they thought the text came from an AI. And for syntactic anomalies, we found a clear P600 effect for syntactically anomalous sentences. And very interesting, this effect was larger for sentences said to be produced by an AI. And this is compared to those sentences that said to be produced by a human. And this indicates that people has less tolerance for syntactic anomalies when they thought the text came from an AI. And for syntactic anomalies, it's quite straightforward. We can think that's basically the reflection of the linguistic ability, especially your grammatical knowledge. But for the semantic anomalies, we want to see what it actually represents. So after the EEG test, we let them do a survey. Basically, there are a bunch of questions related to their usage of AI chatbots in their daily lives. And one question is related to their perceived knowledge resemblance between humans and AI chatbots. And that is, how similar do you think are AI chatbots and humans in terms of their knowledge? And they rated on a 7-point Likert scale. And we use these ratings to predict their semantic N400 magnitudes with a linear model. And the magnitude is calculated by the amplitude of a anomalous condition minus the well-formed condition. And we found that the perceived knowledge resemblance between AI and humans did not predict their brain responses to human language, as you can see here, but it negatively predicted their brain responses to AI language. And because the N400 is negative, so the negative prediction means that when people do not think AI and humans are similar in terms of their knowledge, they have a small brain response to the semantic anomaly that the AI chatbot produces. But when people think AI and humans are very similar in terms of their knowledge, they have a big brain response to the semantic anomaly that the AI produces. And so with these results, we can actually see people's expectations about AI's capabilities from their brain responses. And this is probably one of the first studies showing people's neural responses to large language models. So why are people tolerant of their semantic anomalies, but show less tolerance for their syntactic errors? So we guess this reflects the fact that people know that AI will sometimes say nonsense, just like what Yan Legan said. Large language models are subject to hallucinations. We all have this kind of experience where you ask GPT about something and it gives you an answer. But when you go to Google to verify that answer, you find it is wrong. And then you go back to the GPT and say, you are wrong. And it will immediately apologize and give you a new answer. So people know sometimes it just produces nonsense. That's why people are more tolerant about their semantic anomalies. But on the other hand, we trust them for their linguistic ability. We use them to write up reports every day. We use them to polish the language when we write papers. So we don't expect them to have grammatical mistakes. So when they produce grammatical errors, we have a bigger brain response. So that's the first study. And actually, our finding on the AI language comprehension is part of a broader principle in language comprehension. And that is, people's neural responses to language are heavily influenced by their expectations about the speaker. And the same linguistic input can be processed differently depending on who we believe is producing it. In other words, we don't just process language in a vacuum. Instead, we interpret what we hear in a wider context. And this context is the speaker's identity. And this is what we termed speaker contextualized language processing. And now let's consider this example. I'm going to have a manicure this weekend. When the sentence is spoken by a female speaker, it's very easy to understand. This woman is going to get her nails done this weekend. But if you can consider if this is spoken by a male speaker, then it might give us a little bit of a pause, because stereotypically speaking, men are less likely to do nails than women. But anyway, we can still understand the statement. And now let's consider this example. The first time I got pregnant, I had a hard time. And this is quite straightforward when it's spoken by a female speaker, but you can imagine if this is spoken by a male speaker, that becomes a bit confusing because to some extent, it violates our biological knowledge. And previous research explored this kind of speaker contextualized language comprehension by exposing listeners to what we called implausible self-referent utterances. And these are utterances where the speech content mismatched people's traditional expectations toward the speaker in terms of their identity, like their biological gender, age, or social status. And for example, in this paradigm, they had participants listen to sentences like, every evening I drink some wine before I go to sleep, spoken by a young child. So we can see at the critical word wine, it actually violates people's stereotypical view toward children because children are not likely to have a habit of drinking alcohol. So when they contrast this condition with another condition where an adult speaker says the same sentence, every evening I drink some wine before I go to sleep, they got an N400 effect. And this is very similar to the N400 we just introduced for semantic anomalies. But later in subsequent studies, other groups of researchers, they used the same paradigm, but you can see they ended up with a P600 effect, which is similar to what I had just introduced for the syntactic anomalies. So over the years, there are quite a lot of studies using similar paradigms to explore the neural correlates of speaker contextualized language comprehension. But as you can see, they either ended up with an N400 or a P600, and the debate is going on for quite a while. And actually, in language research, despite the N400 and P600 are originally associated with semantic and syntactic anomalies like we just introduced, they also represent two distinct cognitive stages. And for those who found an N400, they argue for an early stage integration where the speaker context are processed simultaneously with the sentence semantics. And for those who found a P600, they argue for a later stage integration where the speaker contexts are actually processed after the construction of speaker-independent sentence semantics. But in our study, we propose that the N400 and P600 effects, they may not reflect the distinction between earlier and later integration, but instead, it reflects a mechanism of rational inference during language comprehension. So what is rational inference? So basically, it is the process where you consider the probabilities for something to happen. For example, in one of the rational language processing theories, listeners would model the perceived input as not perfect, but often contaminated with noise such as environmental distraction or production or perception errors. And on the side of the speaker, you have this intended meaning. And from this intended meaning, you will have this intended sentence. But on the side of the listener, you have this perceived sentence. And from this perceived sentence, you have this perceived meaning. And in between, you have this what we call a noisy channel, where the sentence can be corrupted by noise. So when you hear an implausible sentence, you would consider the likelihood that the sentence has been noise corrupted from an otherwise plausible one. And when the sentence is extremely implausible, you would tend to detect an error and try to correct it by revising the perceived input. So now let me give you some examples. When you hear the sentence, the mother gave the candle the daughter, you would likely to unconsciously insert a to make it the mother gave the candle to the daughter. And similarly, if you hear the sentence, the mother gave the daughter to the candle, you would likely to unconsciously delete this to and to make it the mother gave the daughter the candle, just to make the meaning more plausible. And in EEG research, they found that the relative magnitudes of N400 and P600 are modulated by the plausibility of the perceived language input. And when the input is relatively plausible and no explicit error is detected, the N400s will be more salient. And that indexes the difficulty of lexicosemantic processing. But when the input has extremely low plausibility and is detected as an error, for example, a speech error or a mishearing, then the P600 becomes more salient, and that reflects an error correction process. So we go back to those literature and we examine the experimental material that they used. We identified two types of implausibilities that we think might elicit distinct types of brain responses, and they might elicit either a N400 or a P600. So the first type is what we termed socially implausible utterance, where it violates people's social stereotypes toward the speaker's population, like in the sentence, I'm going to have a manicure this weekend, spoken by a male speaker. And at this critical word, manicure, it actually violates people's social stereotypes toward the male population. And second type is what we termed the biologically implausible utterance, where it violates people's biological knowledge, like in the sentence, the first time I got pregnant, I had a hard time, spoken by a male speaker. At this critical word, pregnant, it actually violates people's biological knowledge. So we propose that the violation of the biological knowledge may lead to a P600 effect, and this reflects the detection and the correction of an error. For example, in this study where they found a P600 effect in the end, they included a lot of stimuli where the linguistic content actually violates the biological feature of the speaker. For example, they used a stimulus like, I talk a lot to my son at the weekend. And this is spoken by a child. Or the stimulus like, today I'm feeling sick, I will need to visit my pediatrician again. And this is spoken by an adult. So you can see at the critical word son and the pediatrician, it actually violates our biological knowledge about the speaker. And in the original study in 2008, they found an overall N400 effect for the speaker message mismatch. But they also mentioned that only the male-female speaker implausibilities elicited an additional late posterior positivity, aka P600. And only male-female speaker implausibilities were sometimes biologically implausible. And as I don't like having my period when I'm on vacation in the male voice. So to test our hypothesis, we designed 40 social sentences and 40 biological sentences. For each sentence, we had two versions. So for the plausible version, the speaker's identity mismatched. The speaker's identity actually matched the speech content. And that is a female speaker saying, the first time I got pregnant, I had a hard time. And for the implausible version, it mismatched the speech content. And that is the male speaker saying, the male speaker saying, the first time I got pregnant, I had a hard time. And now it's very convenient to get spoken materials. We don't need to invite speakers to record materials. We just use Microsoft Azure to generate realistic speech. And this also allowed us to control for confounders like the audio volume, speaker habits, and stuff. And we matched for the critical word frequency and length, and these linguistic variables significantly influence the EEG amplitude, as previous research has shown. And we also match the critical word duration between the plausible and the implausible version, just to control everything that we can think of. And here are some examples. Because the biological gender and age are the two most salient features of a person. So we included a gender contrast and an age contrast, and we only contrast between a plausible version and an implausible version, so the sentence content is always the same, but the speaker is different. So before the EEG testing, we want to make sure our manipulation is valid. So we recruit participants for material rating, and we want to balance participant gender. We recruited 30 participants, half male, half female, and we invite them to the lab and present all the audio utterances to them. And they listen to the audio one by one, and they rate on a 7-point Likert scale how plausible they think it is for this speaker to say this sentence. And as we expected, the socially implausible utterances are rated significantly less plausible than the biologically implausible sentences. The socially implausible sentences are rated significantly more plausible than the biological ones. And these two types of utterance show different patterns actually. And for social utterances, you can see something like a spectrum. So for some items, there is only a very small difference between the plausible and implausible versions. But for some items, there is a very big difference between the two versions. But for the biological utterances, you don't see this spectrum. For most of the items, it's a clear-cut difference between the two versions. So it's either plausible or implausible. Right. So for the EEG testing again, we want to balance the participant gender. So we included 64 participants, half male, half female. And after the data exclusion, we still make sure that there were 60 participants for the statistical analysis, just to get enough statistical power. And because we are using audio stimuli and we are doing time log analysis, so we really want to use precise time markers to mark the onset of the critical word in each sentence. So we included a device. Here we are using a E' Chronos to play the audio and send a time marker to the amplifier at the same time. So this makes sure that we get a relatively precise time marker for each sentence. And this is the result. So the results showed that social and biological implausibilities elicited distinct brain responses, as you can see from the topographic maps. So the social implausibility triggered a negative potential, and the biological implausibility triggered a positive potential. And from the waveform, we can see that social implausibility elicited a long-lasting EEG N400 effects, and the biological implausibility elicited a standard P600 effect. And more interestingly, before the EEG testing, we actually measured our participants' personality traits using the Big Five personality survey, and we found that the social N400 effect magnitude is positively predicted by their openness score. So openness basically refers to how open-minded a person is. So how open this person is to new experience or new ideas. So that means if a person is very open-minded, their brain responses to the violation of social stereotypes are quite small. But if a person is not that open-minded, their brain responses to the violation of social stereotypes are very big. And for their brain responses to the biological implausibilities, we didn't see this kind of prediction. And this pattern is also true for the plausibility ratings we did before the experiment. A more open-minded person tend to rate the plausible version of a social utterance similarly with the implausible version, but a less open-minded person has a bigger difference between the plausible and the implausible versions. And again, this rating pattern only applies to the social utterances, but not the biological utterances. And we explain the results using a Bayesian framework. So this part is a little bit theoretical, but bear with me. In our life, we interpret everything based on probabilities. So first we have our prior knowledge, and that is our estimated probability for an event to happen. And in this case, in our experiment, it's our knowledge about different populations. And this knowledge includes our social stereotypes toward those populations and our knowledge, our biological knowledge for those populations. And second, we get evidence. So in this case, it's our perception of the speaker and their speech content. So is it a male speaker or a female speaker? And what do they say? Things like that. And finally, we calculate the posterior probability based on our prior knowledge and the evidence. And this is our interpretation about the fact that this speaker is producing this sentence. So when we hear a socially implausible utterance, it actually activates social stereotypes that we have. And because social stereotypes are distributed flat and wide, because everyone has different social stereotypes, and the social stereotypes can be flexible. So it's flat and wide. So our perception is likely to lie within the range. So we can probably calculate an interpretation integrating our social stereotypes and the utterance we hear. And the N400 effect here reflects an effortful semantic integration. But in contrast, when we hear a biologically implausible utterance, it activates the biological knowledge that we have. And because the biological knowledge tend to distribute concentrated and narrow, that means so we either know this knowledge or not. It is not very flexible, and different people has something like the same biological knowledge. So it's likely that our perception lies outside the biological knowledge distribution, and there is no way to calculate the reasonable interpretation. So instead, we revise our perception to align with our biological knowledge, and the P600 effect here might reflect an error revision process. As for what errors are being revised, we think it's more likely that we revise our perception of the speaker's identity according to what they say. For example, when I hear a low-pitched voice, so initially, I assumed that this speaker is a male speaker. But when I hear that they got pregnant, then I revise my assumption and think the speaker might be a female speaker just with a lower voice. Right, and a person's openness trait actually modulates their social stereotypes, but it doesn't modulate their biological knowledge. So if we summarize, our brain responses to speech is actually modulated by the probability that we estimate for a certain population to say certain thing. So in the next study, we want to know whether this probability we estimate for a certain population is fixed or it can dynamically change through the course of communication. So you can imagine a scenario. When you listen to an adult man saying things about himself, and you find that he frequently mentions things in his life that do not fit his age. For example, he likes to play water guns. He can't sleep without his teddy bear in his arms. He's often scolded by his mom for being too naughty. You might adapt your expectations for this person, and maybe he's very young, but has an adult-like voice, or maybe he has a unique lifestyle. So we want to see whether we can detect this kind of adaptation from people's brain responses to these implausible utterances. So we designed speech materials that stereotypically fit certain populations, but we don't include materials related to people's biological knowledge because our previous study shows that people's biological knowledge is not likely to be changed. And for the materials, for example, for a stereotypically male sentence, we had I've done gatekeeper work in a lot of places before. And for stereotypically female sentences, we have I always wear dresses when I go out shopping. For stereotypically adult sentence, we have last month I served as best man at our best friend's wedding. And for stereotypically child sentence, we have he took my toys away from me. And for this study, our critical manipulation here is the base rate of incongruency. And that is the probability for a speaker to say things that is incongruent with their identity. So for being incongruent, that means mismatch. So incongruent means mismatch. The speaker's identity mismatches the speech content. And we manipulate this by including different filler trials between the two conditions. So a filler trial means that the participants do this trial, but we don't analyze the data from this trial. And so in contrast, a target trial means that we do analyze the data from this trial. So you can see for both the low base rate condition and the high base rate condition, their target trials are identical, but their filler trials are different. So for the low base rate condition, half of the filler trials are speaker congruent, and the other half are neutral. And for the high base rate condition, half of the filler trials are incongruent, and the other half are neutral. So if people are sensitive to the unique trait that individual speaker shows during their self-description, their brain responses should differ between the high base rate and low base rate conditions. This is our hypothesis. Right, so these are the results. So for the brain potentials, we examined the N400 time window and the P600 time window, but unfortunately, we didn't see any difference between the low base rate and high base rate conditions. So one possibility is that our approach didn't detect the effects that actually exist. Because if we look at the brain potentials, it only reflects the phase-locked activity. So in other words, that is the activity consistently time-locked to the onset of a stimulus. But if we think about it, the cognitive process of considering the probability of someone speaking something can be a complex process. So it can involve the brain activity of a person to dynamically adapt to someone using the incoming information from their speech. And the activity might not be strictly phase-locked to the onset of the critical word in the sentence. So we decided to look at the induced neural activity, which involves the non-phase lock brain oscillations. Right, so we want to look at the time frequency representations. And this allows us to examine both the time and frequency of the neural oscillations. So basically, we decompose the EEG signal into its constituent frequencies over time. And this provides a more comprehensive picture of the brain's oscillatory patterns. So if the brain potentials are like an average snapshot of the brain's activity at some particular moments, the time frequency representations are like a video. So it shows how different frequency bands of activity actually evolve over time. And for some details, we used Morley wavelengths to decompose the EEG and extract the oscillatory power information around the critical word onset in the sentence. So you can imagine the first time I got pregnant, I had a hard time. So the critical word is the pregnant. And we decomposed it in a frequency range from 2 Hertz to 44 Hertz that normally covers the cognitive neural activities involved in language comprehension. And we calculated the change in oscillatory power after the critical word onset relative to the baseline before the critical word onset. And to reduce the risk of multiple comparisons in finding differences in time frequency representations between low base rate and high base rate conditions, we used a cluster-based permutation method, and we found 2 clusters that showed significant difference between low base rate and high base rate conditions. So the first cluster is an early stage high beta band activity from 220 milliseconds to 330 milliseconds after the critical word onset. And this cluster, as you can see from the map, is distributed around the central region of the scalp. And the second cluster is the later stage theta band activity from 320 milliseconds to 580 milliseconds after the critical word onset. And this cluster is detected at a small region in the left central parietal area. So based on the time frequency representations, we show that listeners are actually sensitive to the probability of speaker incongruent speech. So next, we want to know the cognitive mechanism behind this sensitivity. So there are 2 potential mechanisms that we think might be behind this. So the first one is a speaker general priming mechanism. So that is, listeners are sensitive to the probability of hearing speaker incongruent utterances in general. So they just got used to hearing such kind of utterance. And the other one is a speaker-specific modeling mechanism. And that is listeners construct a mental model for a specific listener and dynamically revise the mental model during language comprehension. So this speaker model contains all kinds of characteristics that the speaker has. So at first, when I hear an adult speaking, I may construct a mental model of a typical adult. But as the speaker keeps saying something incongruent with their age, I revise my mental model for that speaker. So to test that, we did another experiment. So this time we used 2 speakers and they show up interleaved. And for all target trials, we used the original speaker that we used in the first experiment. But for all the feeder trials this time, we used a different speaker. So if listeners are sensitive to the probability of hearing speaker incongruent utterances in general, then we should replicate the effect we detected in the first experiment. But if listeners construct a mental model for a specific speaker and revise the model during language comprehension, then we should not detect any difference between the low base rate and high base rate conditions, because as you can see, the base rate is only manipulated by another speaker, which we don't analyze. So as a result, we found a high beta band oscillatory power change between low base rate and high base rate conditions. And this is very similar to the high beta band effect detected in our first experiment. And also for this study, we also measured our participants' personality traits, and we extract the average power change, and we found that the high beta oscillations are not predicted by participants' personality, but the theta oscillations are predicted by the participants' openness score. So overall, we suggest that the high beta oscillations might be related to a speaker general priming mechanism, but the theta oscillations might be related to speaker-specific modeling. So across these studies, we saw evidence for individual differences in language processing. And in particular, we found that the personality trait of openness modulated the neural responses to violations of social stereotypes. And this finding suggests that the way we process language in social contexts is not just related to the linguistic input and the speaker's identity, but it's also related to our own personality and our social attitudes. So I think this might open up possibilities for future research and application. For example, could we use people's neural responses to social speech as a way to decode their personality traits and the social stereotypes, or even their risk for certain psychiatric disorders? So I guess this highlights the need for an interdisciplinary approach among medical engineering, computer science, psychology, and of course the linguistics communities. So with this, I'd like to thank my collaborators, the principal investigator, Professor Gary Tsai, and my colleague Xiaohui Rao. And these studies are funded by the University Grants Committee, Hong Kong. Thank you.
Thank you very much, Henry. Very nice presentation. Very nice lecture. It also like broaden my horizon in the neuro.
This can be quite different from other talks. I know.
Yes, linguistic knowledge. Voila, thanks for teaching us all these, and very informative, like from your first experiment to your second experiment. So you kind of find the can I describe it this way? Find the biomarker like beta oscillation and theta oscillation when the subject is interacting like in
Yes,
the
I think
interaction.
you can put it this way. Yes, the biomarker for a listener to think about the speaker's characteristics and dynamically adapt their cognitive processes to this specific speaker.
And okay, because this knowledge is very new to me, so excuse me if I ask some basic question. So maybe I have missed out something. So the beta oscillation, it is you observed it when the subject is like in what situation? Beta oscillation.
Yeah. So how do we found the beta oscillation? So that's when remember that we have like two factors. The first factor is the congruency. So you know the child's speaker say the teddy bear sentence versus the adult speaker says the teddy bear sentence. So this is the first factor. And the second factor is the base rate of incongruency. And when we like examine the interaction between the two factors, then we found this significant cluster in the high beta range. So this is how we got this neuromarker.
And the beta oscillation,
yes. So that's the same method. Yes, we actually found the two significant clusters.
Great, interesting, and wow, it makes me feel more interested in neurolinguistics also. And yeah, and
I'm glad.
your next stage of your experiment, you would like add on the TMS component to stimulate.
Yeah, that would be a total different topic. So we have different projects, and for the TMS project, we want to examine so we nowadays people often have this thing called character amnesia. So I want to write down some characters, some Chinese characters, but I totally forgot how to write it. So we want to see if our TMS stimulation can like change the rate of this character amnesia happening. So if we can use TMS to treat people, that would be a wonderful thing. Yeah.
Great. So we look forward to Masterclass Hong Kong 2.0 next year for that. Great. Thank you for this wonderful presentation. Any questions from the audience side? Please feel free to type it in the Q&A chat.
You can also drop me an email if you got something to talk about. Yes.
Yes, maybe you put your email in the chat room so everyone can also see it and contact you. Yeah. So the audience seems quite shy on asking questions. It's typical Asian. We share this trick together. Right. So feel free to email Henry for further question or maybe research collaboration. Thank you very much, Henry, for your nice work and nice lecture. Thank you for the presentation. It's very good to have you. Look forward to next year also. Thank you. Thank you.
Thank you.
So
thank you.
we keep in touch. Thank you.
Thank you.
Then let's move to the next talk. It will be presented by Dr. Jack Zhang from the Department of Rehabilitation Sciences, the Hong Kong Polytechnic University of Hong Kong. Dr. Zhang is at the moment in Germany. So it is good morning for Dr. Zhang. Dr. Zhang, I have promoted you as a panelist. Please accept my promotion for you. And then, yes, I can make you co-host and you can share screen anytime. Good morning. Maybe I need to check a bit your microphone and your video. Just try to unmute.
Hello.
Hello, Dr. Jack.
Hello,
Oh, I can
Christy.
hear you clearly now.
You can hear me.
Hello, hello.
Hello.
Yes.
Let me share my screen first.
Yes. Good to see you. How's Germany? Did you have enough beer?
Not yet. Just
Right.
arrived a few days ago. So you can see the screen now.
Yes, I can see. Thank you for all your preparation. You're going to share with us the use of EEG and closed loop BCI neurofeedback in post-stroke rehabilitation. Looking forward to your presentation,
Okay.
everyone.
Thank you very
Please
much,
enjoy.
Christy. Thank you very much. Thank you for inviting me again to give this talk in the BCI Neurotech Masterclass. Today I'm going to share our recent research regarding the use of EEG and the closed loop brain-computer interface BCI in stroke research. And first of all, I have no conflict of interest to declare for this talk. So today I will cover several neurotech technology which has been widely used in stroke rehab, including EEG, BCI neurofeedback, non-invasive brain stimulation, and in the last part, I will briefly talk our future research about the use of closed-loop neurotechnology and BCI in rehabilitation science research. So this year is exactly the 100 years since the first human EEG was recorded. EEG is very important for BCI research because it provides a non-invasive way to measure the brain activity, which allow for real-time monitoring of brain signals. So EEG enable the development of BCI, which can interpret the brain activity and translate the brain signal into command for controlling external device, which can be very helpful for people with physical disability. According to the study design, EEG can be very roughly categorized into resting or task state EEG. Resting state EEG, or we call it spontaneous EEG, has been used in stroke research because at resting state, researchers already observe some pathological neurophysiological pattern over the cortical oscillation and the network property in people suffering from a stroke. For example, according to some previous study, typically we can observe an increase in the low frequency oscillation and a reduction of high frequency oscillation in the people after acute stroke, especially over the lesional hemisphere. And these increase in low frequency and the reduce in high frequency leads to the interhemisphere asymmetry, and the power asymmetry is actually predictive of the functional recovery after stroke. And in addition to the power analysis, the resting EEG can be also analysis and can be analyzed by using the graphic theory-based network measurement. And these measurements are calculated based on the function connectivity matrix among the EEG sensor or the EEG source. And they can be used to evaluate the brain network properties such as functional segregation or integration. And in our lab, we perform an EEG study in stroke patient. And in this study, we have stroke patient, chronic stroke patient, and also healthy control, age-matched healthy control, and young healthy control. And consistent with previous observation in these group of chronic stroke patients, we observe the increase in low frequency activity, specifically in the delta and the theta band, and also a reduction in high frequency activity, specifically in the beta band in the epicelational hemisphere compared with the contralational hemisphere. And we also observe the between the low beta band power over bilateral hemisphere, especially the sensory motor area, and the functional motor function of the affected upper limb in this group of stroke patient. And we also perform network analysis based on these resting state EEG. We calculate several index, including node strength, which is a sum of the connectivity value, and the clustering coefficient, which is a measurement of functional segregation, and also the global efficiency, which is a measurement of functional integration. So by comparing these network measurement from stroke patient and the healthy control, we see the differences between the two hemispheres. Specifically, the epicelational hemisphere of the stroke patient showed reduced node strength and functional segregation in the alpha and the beta band. And we also observe the correlation between the node strength in the delta band in either epicelational hemisphere and the contralational hemisphere, and the functional motor function outcome in stroke patient. So it means the resting EEG already very helpful, very useful for clinical research to help us to understand the pathological change after a stroke onset. And the task-related EEG is also commonly used in stroke research. We use the ELD, event-related desynchronization. These ELD liberally means the suppression of oscillatory power, and which can be induced by movement or movement observation in mu and the beta band. So as you can see from this plot, at resting state, the oscillatory power of mu or beta band is high. And during the task state, such as action observation or movement execution, you can see the power amplitude is suppressed. And it can return back to baseline after the termination of the functional task. So the mechanism of ELD has been described. At resting state, the sensory motor neuron, they fire together, leading to a high power of synchrony. However, when the participants start to move or they observe others' movement, the sensory motor neuron fire out of this synchrony, leading to the reduction of the oscillatory power. So the ELD can be used as a neural correlate of the cortical activation. So according to previous research in stroke patients, these ELD induced by movement has been used, and the researcher observed a reduction of movement-related ELD over the sensory motor area, over the epicelational sensory motor area in stroke patient, which means a low excitability state after stroke, especially over the affected hemisphere. And this power suppression, this ELD value, was found to be correlated with the upper limb motor function in stroke patient. It means the low excitability state may be relevant to the functional deficit of the upper limb after stroke. And we also use ELD, but we use a different paradigm. We use mirror virtual feedback, which is a paradigm for inducing ELD by using the action observation. As I already mentioned, the ELD can be induced by movement or movement observation. So in the context of stroke rehab, for mirror therapy or mirror virtual feedback, we ask the participant to move the unaffected side, and simultaneously they can observe the virtual feedback from a mirror look as if the affected side is moving normally. And according to the study on the right-hand side, this is actually a MEG study. Movement execution with mirror virtual feedback can help revise the imbalance of interhemisphere activity measured by ELD in stroke patient, which somehow interpret the treatment mechanism underlying the mirror therapy. So this is our study. We use the mirror observation paradigm, and we have several experimental conditions. We have mirror virtual feedback condition. So the participant move the affected hand, but they look at the mirror virtual feedback. And the second condition is direct virtual feedback. The participant just direct look at the moving finger of the affected hand. And the third condition was a cover mirror condition. So participant move, but they cannot see any virtual feedback because the mirror was covered. So we perform comparison between healthy control and the stroke patient, and in general, the ELD power in the beta band and also mu band, alpha band was lower in people after stroke. However, we observe more ELD in the low beta band over bilateral hemisphere in both mirror virtual feedback condition and action observation condition, which means the virtual feedback can somehow activate the sensory motor area in stroke patient. However, because we found the ELD over bilateral hemisphere was lower in stroke patient compared with healthy control, this finding indicate a reduction of responsiveness to virtual feedback in stroke patient, means they have limited capacity to perform information processing of the virtual feedback regarding motor information. And we also perform correlation analysis, and we found the ELD value in the two virtual feedback conditions at the beta band was correlated with functional levels of affected upper limb in stroke patient. So in addition to resting state EEG and the task state EEG, we also utilize a technique called concurrent TMS EEG measurement. TMS means transcranial magnetic stimulation. The previous speaker already mentioned, and we use TMS EEG as a measurement. So the single pulse TMS can induce a invoke potential. We call it TMS invoke potential, TEP. And the TEP is a measurement of the cortical excitatory and inhibitory neuronal activation. So specifically, TMS can invoke several peaks, positive or negative peaks, and the early components such as P30 or P25 is correlated with cortical excitability, while the late components such as N100 is correlated with cortical inhibition mediated by the GABA B receptor. So this is a previous study using, I think it will be the very first study using TMS EEG, TEP, in stroke patient. So you can see by stimulating contralational side and the epicelational side, we see very different TEP response. So you can see over the contralational side, we see several positive or negative peak, which is similar to the typical TEP response. However, if we apply the TMS over the lesional hemisphere, you can see the TMS invoke potential is much simplified. So this is a simple, very simple simplified wave. So many peak are lost. We can observe the functional component in the TEP response if you apply TMS over the lesional hemisphere. So this is another study which replicate the TEP finding in stroke patient. So you can see from the health control, this study have a group of health control. The healthy control show typical TEP response. So you can see several positive or negative peaks in the TMS EEG response. However, over the epicyclical hemisphere, we see a simplified response. And the researcher quantified this response by using two indicators. One is the local mean field power, which means the amplitude of the signal, and the second is the number of deflection, which is a measurement of signal complexity. So compared with healthy control, the local mean field power in the early phase was larger in stroke patient because we can see these simplified but large response in the epicyclical hemisphere. And these mean field power was negatively correlated with the functional recovery because in these research, they have follow-up data. However, the signal complexity was positively correlated with the functional recovery. So it means if the response in a patient showed more deflection, the recovery outcome will be better than those without many deflection in the TEP. So in summary, the large and the simplified early TEP is predictive of the poor neurological recovery in stroke patient. And the same research group report the follow-up data. So they measure the TEP again at follow-up. And this is the paper they published this year. So you can see a reinstatement of TEP signal capacity. So here I'm showing two representative case. You can see in both case A and B, in the acute phase after stroke, we observe the simple and large response. However, in the chronic phase, we see there is an increase in the number of deflection in the TEP response. So the functional recovery is paralleled by the reinstatement of the TEP signal complexity, which means TEP is possible to be used as a predictive biomarker of the stroke recovery. So in our lab, we also utilize the concurrent TMS EEG measurement at PolyU. We had in our study, we enroll a group of chronic stroke patients, and all of them were well recovered in terms of upper limb. They already recovered from stroke, and most of them are subcortical stroke patients. And by using a TEP, we observe the TMS EEG response from both epicyclical and the contralateral hemisphere. And we also observe the large and the simplified response in two of the participants we enrolled. Although they already recovered from a stroke, the upper limb function of them is nearly normal, but we can still observe the simplified response in two of the participants we have. And we perform comprehensive neurophysiological measurement using TMS EEG and also MEP, motor involved potential. So in terms of motor involved potential, we observe a reduction of cortical excitability over the epicyclical motor area, which is consistent with previous finding. And we also use the cortical silent period, which is a measurement of the GABA-B receptor mediated intracortical inhibition using TMS EMG. And we found the cortical silent period over the epicyclical hemisphere was significantly prolonged compared with the contralateral CSP. And by using TMS EEG, we observe a reduction in the magnitude of intracortical inhibition because of the N100 component of the epicilitional hemisphere was significantly lower than the N100 from the contralateral hemisphere. So in summary, we utilize TMS EEG and MEG, TMS EEG and MEP, and we found the impairment of both cortical excitability and the cortical inhibition. So which means these comprehensive neural examination can provide a detailed measurement of the cortical functioning after stroke. So in summary, we perform resting state EEG, task state EEG using ELD and the concurrent TMS EEG, and we observe the lesional brain show the lateralized slowing in the cortical frequency, and also a reduction of cortical responsiveness to sensory modality, as well as a reduction of excitatory and the impairment of the intracortical inhibition. So EEG suggests a dysfunctional state of the affected hemisphere, which can be a target for our interventional study for stroke patient. So the second part of my talk is about BCI, brain-computer interface. In rehab, BCI can be used either as an assistive technology or rehabilitation intervention. So as an assistive technology, BCI allow the user to control the computer or machine through self-regulation of the brain activity without any actual body movement, because in people with a disability, they may have limited function to talk or to move, but they can use BCI to control an external device, such as wheelchair or communication device, to communicate with outside or move freely. And BCI is also an intervention method because participants can learn how to regulate the brain activity associated with movement execution by using, for example, motor imagination. And by using the BCI intervention, the adaptive neuroplasticity over the cortical and subcortical motor system can be facilitated, therefore leading to the functional recovery in people with neurological disability. So BCI is potentially effective in rehab because BCI provides a so-called BCI learning for neuroplasticity induction. So in the normal motor physiology of motor functioning, when person need to move, the motor command from the brain is sent out to the peripheral nervous system, muscle, or like your hand or muscle upper limb or lower limb, and the sensory feedback can be transferred back to the brain, forming a so-called sensory motor loop. However, in the people with disability, this sensory motor loop is disrupted, is damaged. So in this case, BCI can be a bypass for involve of the descending motor pathway, peripheral nervous system, and the muscle in the movement execution. So the motor command from brain can be read by BCI, and the BCI can transfer this input command into external device such as computer or external device, and the overall task performance can be relayed back to the individual. So the participant, the user of BCI, can receive feedback, so which allow them to regulate the brain state to achieve a better control of the external device or better performance in a functional task. So this bypass process is called BCI learning. So through this process, the user of BCI can consolidate the modified brain activity pattern. So layer form BCI can induce so-called neuroplasticity, and the layer form leading to functional recovery after stroke. So BCI, the process of BCI includes signal acquisition. We need to have the brain signal using EEG, MRI, FNES, or the biological signal acquisition device. And the brain signal need to be transferred into certain feature. We need to use a certain feature from EEG, fMRI, or FNES. And we need to translate this feature into computer command. So this computer command can be further read by external device. So for rehabilitation research, we have several research questions. So for example, for the signal acquisition, we would like to know what's the optimal image modality for capturing brain signal in BCI intervention. And we also want to know what's the key feature, what's the useful feature, which BCI can recognize in a reliable and effective way. And we also would like to have the most effective feedback in this closed-loop BCI intervention. So by review literature in terms of signal acquisition, there's no doubt EEG now is the most commonly used imaging modality because EEG is portable. Some of the devices are portable and low cost, very convenient in clinical application or technological development. However, recently we also see some other new imaging modalities such as FNES, functional near infrared, or fMRI or MEG. They are more expensive solution. And in terms of the feature, if we use EEG, we have talked before the ELD, which is the measurement of cortical activity, has been widely used in EEG-based BCI. This is a very reliable feature of the multiple reset system. And according to a previous literature review, ELD, the sensory motor rhythm ELD, has been the most commonly used feature in stroke BCI research. So in terms of the most effective feedback, we provide frequency meta-analysis. This is a meta-analysis from our group, and we review the clinical study, including single group study and the randomized control study, using BCI in post-stroke upper limb motor rehab. And we found BCI compared with sham BCI can significantly improve the upper limb motor function in stroke patient. And we're also looking into the task paradigm used in the BCI, and we found that the motor imagination is the most commonly used one. And we also see some people use observation or movement attempted, which are less commonly used. And in terms of the feedback modality, previous literature in stroke rehab use robotic device, functional electrical stimulation FES, or virtual feedback. And we found the FES and virtual feedback when combined with BCI was more effective than sham BCI. However, the robotic subgroup was not significant. So based on our meta-analysis, we performed a pilot randomized control trial using the Recovery X device. And the objective of this project was to investigate the efficacy of a BCI system with two feedback modality, functional electrical stimulation and virtual feedback in post-stroke upper limb rehab. And in this study, we would like to know what's the key therapeutic component in terms of feedback modality in this closed loop upper limb interventional program. So in our pilot study, we have 9 patients in each group. We have 3 groups, BCI, FES, virtual feedback, or BCI with FES, or BCI with virtual feedback. And we'll offer 10 sessions, so participants receive 3 sessions per week, and in total 10 sessions. And we perform outcome measurement using standardized rehabilitation upper limb assessment. And we also assess the capacity of motor imagination of the participant, because this Recovery X program is based on motor imagination. So as you can see, we have 3 groups. The first group is actually the original setting of the Recovery X. So participant perform motor imagination to control the external device, including FES and virtual feedback. While in the second group, we remove the virtual feedback, we remove the monitor. And in the third group, we remove the FES, so the participant just receive the virtual feedback. So here are the results. This pilot study will only have a small sample size, but we already observe significant time effect. Means these 3 type of intervention significantly improve the FMA score in chronic stroke patient. However, we did not see the significant interaction effect. Means there was no between group differences. And we also did not observe a significant interaction effect in the ALET assessment, which is a measurement of functional performance of upper limb. And for the motor imagination capacity, we did not see the session by session improvement in the maximum motor imagery accuracy. So we see participants maintain similar level of accuracy, around 70 to 80 across the session. And we use a questionnaire to assess the kinesthetic and virtual imagery capacity. And we also did not observe significant effect. However, the time effect was marginally significant. And the third outcome is mental imagination capacity to mentally manipulate the block. The block. This is a non-biological item. So still, we did not observe a significant change after intervention compared with baseline. So we perform exploratory correlation analysis. We observe significant correlation between the baseline motor imagery accuracy and the functional improvement in certain subscore of the clinical outcome. It means the accuracy, how the participant can accurately and effectively utilize MI in this BCI intervention may be associated with the functional recovery, which makes sense because this intervention, we ask the participant to learn to self-regulate brain activity by using motor imagination. So the accuracy is the key for the successful treatment. So in summary, we in this pilot study using Recovery X, we observe all type of BCI intervention, BCI with FES virtual feedback, BCI FES or BCI virtual feedback. All of them can improve the recovery of upper limb function in chronic stroke patient. However, the combined use of 2 feedback modality does not appear to be more effective than single-use FES or virtual feedback. And the key of the successful BCI intervention is the accuracy of motor imagination. So I have introduced some previous work using EEG and BCI, and the third part is regarding non-invasive brain stimulation, which has been commonly used in rehabilitation research, and which is also very possibly combined with EEG or BCI. So here's a very brief introduction regarding TMS because I guess some of you know TMS before. So TMS is a non-invasive brain stimulation modality. So it can be an outcome measurement or an intervention. So for outcome measurement, the MEP, single pulse TMS induced MEP is the most commonly used one. So if you apply a suprathreshold magnetic pulse over the primary motor cortex, so you can observe muscle response in the form of motor evoked potential from the contralateral upper limb. Usually we use a hand muscle. And this amplitude of MEP is actually a non-invasive measurement of cortical spinal excitability. And for intervention, usually we apply the several repetitive TMS pulse over multiple sessions. So typically the low frequency stimulation such as 1 Hz can decrease the neuronal activity by inducing the so-called long-term depression-like effect, while the high frequency such as 10 Hz, 5 Hz, 20 Hz can increase the cortical excitability by inducing a so-called long-term potentiation-like effect. So in our research, our lab, we actually use another form of RTMS protocol called theta burst stimulation. So theta burst stimulation is an artificial rhythmic stimulation which mimic the firing pattern of hippocampus neuron. And this protocol has been used for around 20 years ago. People found if you apply theta burst in an intermittent manner, so the brain excitability can be enhanced. While if you apply TBS in a continuous manner, the brain excitability can be reduced. So the key to determine the after effect is the delivery pattern, which is different from the conventional RTMS. And the very good advantage of TBS is the conditioning time is very short. For the ITBS, intermittent theta burst stimulation, a single session will only need 3 minutes, while for the CTBS, continuous theta burst stimulation will only need 40 seconds. So here is a review article from our group. We look into the neurophysiological effect and the clinical effect of TBS in stroke patients. So on the right-hand side, this is we perform meta-analysis of neurophysiological study, and we observe the ITBS over the epicilitional M1 can increase the MEP amplitude after stimulation. And consistently in the clinical study, we also perform the meta-analysis of a clinical study. We found the ITBS was found to be effective on improving the upper limb function in stroke patients compared with sham stimulation. However, for the CTBS, the inhibitory protocol, the result was not consistent. So based on literature review, we believe these excitatory ITBS applied to the epicilitional M1 seems to be effective for post-stroke motor recovery. So later we perform a randomized control trial using theta burst, using ITBS. But here we use a so-called priming protocol. So before the ITBS, we perform a CTBS first because neuron has a self-regulating mechanism. So if you suppress the activity before first, the neuron, the threshold for inducing further LOTP-like effect can be the threshold can be lower, it means easier for the second ITBS protocol to induce the exaggerate effect. So in our paper, we investigate the differences in terms of the clinical efficacy among the priming protocol, in which we apply inhibition first and then ITBS, and the non-priming protocol, which actually was the standard protocol, we just apply ITBS and also a placebo sham control. So we still offer 10 sessions, 3 times per week. We have a group of chronic stroke patients. And because TMS is a form of priming, so we provide motor training after stimulation to consolidate the neuroplasticity induction. And we also assess the upper limb motor outcome and also EEG outcome before and after intervention. So here we can see the result. We observe a significant time effect and also interaction effect. So we perform a post-hoc analysis, and we found the priming protocol, the CTBS followed by ITBS protocol, was more effective than sham stimulation. However, we did not observe the differences between non-priming and the priming protocol, means CTBS and ITBS was not statistically more effective than the non-priming ITBS protocol. And we also look into the ELD outcome. We use the MIRA-induced ELD we have introduced before. And in this study, we use the ELD as an outcome measurement. So we assess ELD before intervention and after 10 sessions of intervention. And we found only the priming protocol was able to increase the ELD because ELD is a suppression, it's a negative value. So we see more negative after intervention, more negative after intervention. And the priming protocol was the only one was able to increase the ELD induced by virtual feedback in stroke patient. So as you can see from this map, we can see more blue color over the epicyclial hemisphere after stimulation in the priming group. And we also found a very interesting correlation, and we performed partial correlation. So after controlling the baseline severity, baseline clinical score, we observe a correlation between the baseline ELD and the recovery amount after intervention. We see a negative correlation because ELD was a negative value, but it means a high level of ELD at baseline, which means a high acceptability baseline, was correlate with better clinical improvement after intervention. So it somehow showed ELD can be a prognostic biomarker in research. So I have introduced EEG, BCI, and neurostimulation. And here I would like to share some of our possible future study regarding the use of neurotechnology. And one recent study we are currently planning is the FNIS-based brain-computer interface as a neurofeedback, because stroke is a cerebrovascular accident. So FNIS can be used to measure the cerebral blood flow, cerebral hemodynamic response. And this information can be used to help us to provide a target intervention to revise the pathological state of the cerebrovascular perfusion in stroke patient, which can be potentially effective in terms of clinical outcome. So we collaborated with a team in German, RWTH, and we propose a combination between our expertise. So we will use a neurofeedback system based on FNIS. So in this neurofeedback system, participants perform can self-regulate brain activity based on by using the motor imagination. And the neurotherapy system is an interventional platform. Participant can observe the virtual feedback from the digital system, and also we can add some AR or VR component in this digital system. But we are planning to have a connection between neurofeedback and the neurotherapy, because conventionally the neurotherapy is open loop, so we don't know the brain state during the exercise, during the intervention. So here we will use FNES to read the signal during motor imagery of the participant. So we will only provide the feedback from the digital mirror therapy system when we detect the high excitability state by using the FNES. So it somehow can create a so-called closed loop intervention for our participant. So the first study is more relevant to the signal acquisition device, but the second direction is to modify the external device to find out the most effective feedback modality in BCI intervention. So robotic device has been widely used, and at our lab we are now running a pilot study. We replace the original recovery external device FES and the virtual feedback. We use a robotic device because in certain situation, electricity may not be suitable for certain case or certain situation. I hope I did not present the result today, but based on our pilot preliminary observation, this BCI robotic system is also effective in stroke rehab in terms of uplink function, uplink recovery. And also we may use brain stimulation as the external device, as a form of feedback modality, because BCI can be used to control the delivery of brain stimulation, because previous brain stimulation, TMS or TDCS, they are also open loop. And we apply stimulation to the brain, but we don't know the brain state. We don't know what's happening before, during, and after stimulation. But if we combine with BCI, EEG base or FNES base or even fMRI base, we can have a real-time monitoring of the brain state, which can be used to guide the delivery of our brain stimulation protocol. So the brain stimulation can become individualized by using the closed-loop neurotechnology. This is the end of my talk. I think we still have a few minutes for Q&A, and thank you very much. I can't hear you. Sorry, Christy.
Okay, how about now?
Yes,
How about
okay.
now? Is it better? Thank you,
Yeah,
Dr.
okay.
Zhang. Like from retrospective EEG research, what has been done and what is your ongoing research? That one of it, you used it, recoveries, that kind of separate the component to see how effective it is. Then like all the way to more neurotechnology in rehab use, TMS, and the upcoming FNES neurofeedback closed loop with your robotics. Very nice presentation. Thank you for all the knowledge and sharing of your research insight. In the Q&A chat, we have one question. Dr. Zhang, how we can verify that the subject is imagining the correct activity during the motor imagination experiment? Or like, how can we tackle this issue?
Yeah, I think it's a valid question. It's a main issue of BCI intervention. We ask participants to perform motor imagination, but we don't know whether they perform or not. I think in recovery X system, the system relies on calibration. So in the calibration session, we need to teach the participant how to perform motor imagery of left hand or right hand, And these data will be used in the training session. So in the training session, the system will perform matching between the ongoing brain state and the previous calibration result. But yes, if during calibration participant did not perform any multi-imagery, so the session will be not valid because the feedback will be totally wrong. Yes, well, there are several ways to monitor whether participant perform motor imagination, such as we can use EEG, because motor imagination can actually induce the ELD, sensory event-related desynchronization, or motor imagination can actually change the hemodynamic response by increasing the oxygen, the HBO level, for example. They can be a measurement to help us to understand, to know the participant is performing the right task we ask them to do. But it still can be challenging because some response may not be specific to motor imagination. For example, if we ask the participant to perform motor imagination, but subject have some spontaneous movement, we always observe during our session. We ask them to think, okay, please only think the movement, but some of them start to move. Okay, but movement can also induce the brain signal change in terms of ELD or HBO or activation in MI, for example. Yeah, I think we don't have a answer now specific outcome measurement of MI, motor imagination, but in our lab, we have neurophysiological measurement. We look into the brain signal, and we also have a questionnaire or clinical score to measure the MI vividness after the session. So we ask the subjective feeling of the participant. So by combining the objective brain signal monitoring and the subjective questionnaire, it can help us to know whether the participant perform or experimental session.
Great. Thank you, Dr. Chen, for answering that question.
In a way,
Right, and Rohit have a follow-up question. In a
yes.
way, we can ask the subject to perform same motor imagination activity multiple times in the calibration process and consider that as a reference.
Yeah, I think this is the built-in function of a recovery X. So you have 80 child,
Yes,
to the
correct.
left or right. I still remember.
Randomized.
Yeah, randomized left or right. Yeah. I think it's a built-in function. It somehow reinforce, consolidate the motor imagination in the participant. Yeah.
Thank
But
you.
based
Thank
on
you,
our
Rohit, for your
our observation, we still need to teach the participant before the session, because if they are not really they don't really understand the instruction, they do not perform any motor imagination, the calibration session will be misleading.
yeah, because like in the traditional physiotherapy, the kind of exercise, when they can move, they would try their best to move, right? But in the motor imagination task, that we just emphasize on asking them concentrate on just imagine, no need to move. So the healthy side is a little bit tricky, and is even more complicated if we do the demonstration on normal person. So on the patients is already more adept to the imagination, but yeah, can be practiced. Thank you, Dr. Chen, for answering that motor imagination question from Rohit. And we have Ayman from Pakistan. I guess he want to ask something. I guess he is typing.
Yes,
So,
please.
Dr. Zheng, what do you think that how about combining TMS with recoveries? What do you think about this kind of combined
I think
treatment?
you already have the solution for TMS EEG. I'm very interested in the concurrent TMS EEG solution from Recovery X because now we are using I have experience in using 2 different EEG amplifier. But Recovery X seems to be another option. We may discuss.
Oh, thank you for having us as one of your options.
Yeah,
And
because we have several studies using concurrent TMS EEG, but this is purely for measurement. So we stimulate the brain and we measure the EEG as a measurement. But in terms of intervention or experimental condition, we need to make it to be a BCI, to be a neurofeedback. I guess Recovery X can be an option because Recovery X is already, at least g.tec is already a BCI company.
BCI company, yes.
Yeah.
Right. So Recovery X, how to say that G-Nautilus EEG cap, the electrode, that is a little bit thicker than the one that we have especially developed for TMS compatible EEG measurement for BCI closed loop system. So to answer your question, like looking for a BCI closed loop system that is TMS compatible, yes, we have, but it might not be like exactly Recovery X. It is like the research device. That would be a wired system. The special electrode that we will develop would be much more thinner, so the TMS stimulation would not be interfered, would not be like blocked. And and the ring in that electrode is also specially designed so the electrode would not heat up. Yes, we have already sold a TMS EEG system to the linguistic department of Chinese University that had gave us a talk before your session. And and I have helped them to like make sure the Simulink template is all working. So depends on your research study. I can also have our software engineer to tailor-made, further customize the closed loop Simulink template for you for your easy management of the measurement with TMS stimulation. We can discuss.
Yeah, we will discuss for sure.
Yeah, when will you come back to Hong Kong?
It will be February.
Very long trip. Oh,
Yeah.
you are like taking a course there or teaching there, or what are you doing there?
I'm in a fellowship program.
I see. Oh, nice. Enjoy your
During the winter.
winter time. I'm saying, then you cannot visit the botanical garden. When I was in Austria, like last year, I went there in winter and I forgot it is winter. I went to the botanic garden and I want to see some rose. No rose there. All like wood branches. All right, we have some more questions here from Ayman from Pakistan. I want to ask that how do the patient in post-recovery stage, like almost 10 months post-stroke, can perform through this experience? I guess it's, yep.
I think for chronic stroke patient, because in our lab, we will also offer interventional program because in chronic stroke patient, although the functional level is stable compared with the early stage, but they suffer so-called learn not use because of the affected deficit of the affected side. They seldom use the affected hand. So the functional level is declared. So we still provide intervention and we still can observe some improvement in the affected hand, which can be helpful. Some of the improvement in clinical score can be further translated into daily activity. But yes, based on our recent finding, because some of our study are very experimental. For example, we provide only 1 hour intervention for 3 weeks. Perhaps the improvement score in these chronic stroke patient is smaller than the acute stroke patient, but it can be a very good example for future research if we would like to translate this interventional program into early stroke rehabilitation. Yeah, but I think for chronic stroke patient, there's still hope for recovery because neuroplasticity is always there.
Great. Thank you. And yeah, Armin is also referring to a massive stroke impact patient. Like how can they perform? How can they perform in the MI?
Yeah, I think
better?
this is relevant to severity of stroke, lesion size of the stroke, because we know every stroke patient is different. Some of them have mild stroke deficit, some of them have severe impairment. I think in most of our research, we include the subcortical stroke because we use FNIRS or EEG from they are cortical measurement. So we measure the cortical brain signal. Because if the patient have a severe damage over the cortical area, perhaps the signal can be much more complex than those without this cortical lesion. But I'm not saying they don't have hope. But because for those severe case, we may also offer some different type of interventions. For example, BCI, maybe they can use the convolutional side to control the system, for example, or we offer TMS over the unaffected side in order to reduce the abnormal brain activity. But this is a ongoing topic in stroke research, how the severity of stroke impact our interventional program. Yes, because we see many experimental study, we limit it to a specific group of stroke patients, such as subcortical, ischemia, or mild to moderate deficit. But how can we translate the finding from these specific group to other type of groups, such as a severe stroke patient? We are still not very sure.
Thank you, Dr. Zheng. Right, and Sean, I think, yeah, Sean is asking, we often focus on motor recovery in cases of hemiplegia, and there is some evidence suggesting that this can have an impact on neuroplasticity. However, have we considered the possibility that the neuroplastic effects may primarily target motor pathways rather than addressing the underlying mechanisms of neglect? What do you think, Dr. Zheng?
Let me try to answer this question. Can be
More
yes,
challenging question for you.
Yes. Some evidence suggest it can be having neuroplasticity. Yes, motor recovery is relevant to plasticity. Have we considered the possibility of primarily target motor pathway? Yes, because in my talk, for my research, I focus on motor rather than addressing underlying mechanism of neglect. Neglect means the behavior inattention in stroke patient, I guess. Yes, there are interventional program for attention problem, cognitive impairment in stroke patient, because we also have we also see some of the stroke patient, they have cognitive deficit after stroke. I think some of the intervention can be applied to cognitive deficits, such as unilateral neglect, such as TMS or BCI. I'm not very sure we have attention BCI using P30, which can be helpful for neglect, because this is not my expertise. I guess there is a attention training program using BCI to improve the attention in stroke patient. Some of the content can be used in cognitive recovery after stroke as well, because neuroplasticity of different brain region in charge of different function in the disease population. Yes. I hope I answered the question.
Thank you, Dr. Zheng, and thank you, Sean, for your question. So, great. We have one more question for Dr. Zheng. Ninu is asking, while selecting the participants for motor imagery for collecting dataset, how can we recognize whether they are cognitively active?
Yeah, we have inclusion criteria. We have cognitive screening to ensure they are cognitively intact. Yes, cognitively normal.
Great. Thank you, Ninu. Thank you, Dr. Zhang. Great. It's a great talk, great presentation, great interaction Q&A. Thank you, Dr. Zhang, for joining us all the way from Germany. Enjoy your stay there and stay warm, and enjoy all the beer, like all the nice food over there. And we keep in touch and see you in Hong Kong in February.
Thank you and see you. Bye.
Thank you. Keep in touch. Thank you. Right, then all the way, let's move to the next session. We have Mr. Ren Xu from g.tec Austria, our senior research scientist in g.tec Medical Engineering GmbH, our Austria headquarters. Wow, long time. You have been 9 years with g.tec already. Congratulations, Ren. And thank you for preparing this talk. Ren is going to share with us the MindBeagle, the brain-computer interface for brain assessment and communication. He has also prepared a demonstration with us. So everyone, please enjoy this talk by Ren. Ren, feel free
Hi,
to.
Christy.
Hello.
Yeah.
Hello.
Thank
Thank
you
you.
for your introduction and for your organization.
Thank you.
And also thank you all for joining this session. And I bring my greetings from Austria. So I'm going to share my screen. Okay. I just want to see if I can also share the sound, but I think it's not possible.
I think in the video setting, when you share it, yeah, you can also share the sound.
This option is not possible. I don't know why, but I think it doesn't matter anyway. We only have a short video, a short sound, but it doesn't matter. I will just share my slides in this case.
Okay. I see a slide clearly. All good.
Yeah.
So
Okay.
please go ahead. Thank you.
Great. Okay. So, yeah. So today I'm going to present MindBeagle, the brain-computer interface for brain assessment and communication. So, my presentation includes 3 parts. I will start with a brief introduction about brain-computer interface and specifically for DOC or disorder of consciousness, because that's a target for the MindBeagle system. And then after that, I will give introduction about the MindBeagle paradigms. And following that, at the end, I will show a live demo with my colleague. So first of all, what is disorder of consciousness or DOC? Since this is like the target group of the MindBeagle system, so you know, the DOC or disorder of consciousness is like defined according to the awareness and arousal level of the patients. They can be divided into 3, let's say, subgroup. One is the coma. That's the most severely impaired patient group. So you usually have eyes closed and they don't have like any response to the environment. The second group is UVS, unresponsive wake syndrome, or previously it is called vegetative status. So for these patients, they do present sometimes kind of waking, such as eyes opening, but they don't really have like motor or verbal response to their external stimulations. The last one is the minimal conscious state or MCS. So for this patient group, they do produce signs of responsiveness. This is somehow inconsistent depending on the motor and the cognitive functions. So for the assessment of these DOC patients, there are some new behavioral scales, such as the Coma Recovery Scale Revised, or CSR, which is one of the most popular scales. Here you can see there are like 5 subscales. which assess different functions such as the auditory function, the visual function, motor function, and or motor or wave function and communication functions. So in general, it works quite well, and it is quite common use in the clinical settings. However, it has also some limitations. For these DOC patients, they are seriously injured. So lots of them cannot really speak or cannot really move, or some of them cannot really respond correctly to the instructions. So which makes obstacles for this assessment. That's why previous study shows that up to 40% of the patient could be misdiagnosed. So because of this, they're blind or they're deafless or they cannot move, for example, then the scale is not accurate, and which of course could cause another problem, because misdiagnosis, and of course treatment could be different, and also the prognosis could also affect the whole decisions of the family, for example. So that's why we think it's necessary to have some more active or alternative ways to assess these DOC patients. That's why we think it's ideal to use BCI or brain computer interface for DOC patients. Probably you have already heard a lot about BCI today, so I'm not going to repeat more here. Just the basic idea, it's we have some maybe different brain signals. We record like EEG or FNIRS or other brain signals, and we use these signals to provide feedback to the patient. And when we are recording the brain signals, we could apply some stimuli, different types of stimuli like visual, auditory, or tactile stimuli to evoke some potentials in the EEG signal or the FNIR signal, so that we can use such kind of features to assess the brain functions of the patients, for example, the DOC patients. For the paradigms used for the BCI, one of the typical one is so-called audible paradigm. So for this audible paradigm, usually you have two different types of stimuli. One is called standard, the other one is called target. So these two stimuli appears in a sequence, where the target stimuli usually appears less frequent. And if the patient can focus on their target stimuli, and we could observe evoke potentials around 300 milliseconds after the onset of the stimuli. So this also called the P300 paradigm, because this kind of auto paradigm could evoke a P300 evoke potentials in the EEG signal. Of course, as I mentioned before, there are different types of stimuli we can use. But for the DOC patients, since not of them has were limited visual function, so we would prefer to avoid use any visual stimuli. Instead, we could use other stimuli that doesn't rely on their visual functions, such as auditory stimuli or the tactile stimuli. For example, the first paradigm, of course, I will also explain later in details in Malbigo, is the auditory paradigm. So for this auditory paradigm, usually we have two different tones. As here, I'm not sure if we can still hear the sound, because I was trying to, maybe you cannot hear it, but it doesn't matter. It's just basically the standard is kind of low tone. So let's say it's a da, and the target is kind of high tone is a D. So we have a sequence of da 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 D, da 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 D. And for the patient, the task is to count silently these high tones and ignore the standard low tones. And if they can do this, we could observe a evoke potentials, as you can see here, the green lines here. When they focus on this task, and we can observe these evoke potentials in the EEG signal. So we can use these P300 evoked potentials to assess brain functions of the patient and to see if, for example, if they can follow the instructions, and if they have such response in the brain, then it's a direct proof that they may be able to follow instructions and they have a certain level of consciousness to do so. Similarly, the other paradigm is a vibrotactile paradigm. As I mentioned before, it has the same features as the auditory paradigm, so it does not rely on the visual input. So as you can see here, we can place some tactile stimulators, for example, on the wrist. And for example, here you can see two tactile stimulators. And one is the target, the other one is the standard one. And of course, the patient just needs to focus on the target. And when they are able to do this, we could see a similar P300 potentials in the EEG signal. So we can use this tactile paradigm or the auditory paradigm to assess the functions of the patients. And we have already, previous studies have already shown that we can use these paradigms to assess brain functions of different groups of patients, like I showed before, the US and responsive wakefulness syndrome, or the MCS, like a minimal conscious status. So that's basically the basic concept behind Montbagel. And so this is basically the first part I want to introduce. And then for the second part, I'm going to present the whole concept of the Montbagel and the paradigms that have already included in the Montbagel system. So here is like an image showing kind of clinical settings that you may have with DOC patients. So as you can see here, for most cases, patient is lying in bed, and we can put on the EEG cap, so to record the brain signals, and we could place the earphones to deliver the auditory stimuli to the patients. And we could also place these vibrotactile stimulators on different body parts to deliver the tactile stimulations. And of course, for the like the medical staff, or you need to instruct the patient to follow these tasks, for example, to focus on the high tones in the auditory paradigm, or to focus on the tactile stimuli in the vibrotactile paradigm. And when the patient is able to do this, we could see their responses in real time. So you can see there we have the computer there, which receives and analyzes the brain signals like the EEG signal, and shows evoked potentials in real time. So in this way, we can know if the patient is following instruction, if the patient have a active consciousness in this status. So here is like a more detailed components that we have in the Montbagel system, as you can see here. So we have on the right side, we can see the cube. So we have the different sockets here to collect two different parts of the components. Like on the left side, you see we have the EEG part, so we can use the gelatinous 16-channel EEG system to record the brain signals. We have the headphones that we use to deliver the auditory stimuli, and we have 3 bands of vibro stings to deliver the vibrotactile stimulators. So in total, we can have like 7 vibrotactile stimulators at different locations. So that's the way we use in the in the Montbagel system. And for a typical setup for the Montbagel, as you can see here, usually the patient is lying in bed, and we have their headset connected to the cube to deliver the auditory stimuli. We have the vibrotactile stimulator collected to deliver the vibro stings. And of course, we have the wireless transmission of the EEG signal from the gelatinous to the computer. So that's whole setup of the Manbig system. And so this is the paradigms we have in the Manbigo. Now we have, you see there, these 6 different paradigms. The auditory evoked potential, that's the auditory paradigm I mentioned before. And we have 3 different vibrotactile paradigms with 2 stimulators, 3 stimulators, and 7 stimulators. We also have the motor imagery paradigm. So this is kind of similar to the recoverix. Probably you have already heard this before. So this is the, the patient is trying to imagine different like movements, and we see the response in the EEG signal. And the last one is a recent state EEG. So here is to record in the recent state EEG signal, and we can use that for the follow-up analysis. So, oh, maybe I should go back here. I don't know if you can see it clearly. On each of the paradigm, we have some markers here. So for the first one, it's, I don't know, maybe it's a bit small. So on the right top corner of each paradigm, you can see here, it's kind of a notebook there, which means this paradigm can be used for the assessment. So you see all 6 paradigms has such a function to assess the brain functions of the patient. And the second marker is this check marker, which means, for this check marker, we can use these 2 paradigms, like the auditory paradigm or the VT2 paradigm, to do a quick test. So quick test means it's just a online real-time like assessments. And for the other 3 paradigms, there's a small document here. So for the VT3, VT7, and the motor image paradigm, so this small document means a communication function. So we can use these paradigms to communicate with the patient. So we can ask simple questions, and the patient can answer yes or no by focusing on different stimuli, such as in the VT3 or VT7. For example, if they are focusing, for example, on the right hand stimuli, which means they're answering no, and for they mean they're focusing on the outside, which answering yes. So we can use these paradigms to build a communication channel to the patients. Of course, when we detect a consciousness in this patient, and then we can try to communicate with them. So I'm going to also explain a little bit of these functions of the Manbigo. So the first one is the assessment function. As you can see here, this is the interface. Later, I will also show you in the lab demo. So we have the EEG scope here, which shows EEG signal in real time. And meanwhile, we have also the EEG signal quantity indication here. So like in this case, of course, the signal is good, so it's nothing showing here. But if we, for any reason, some channel becomes bad, then this may becomes yellow, and this area may also becomes like in red when you know there's any like bad signals. And for the bottom part, you can see here that's the evoked potentials we could observe during this assessment, which shows the brain responses of the patient during like a paradigm. Here, I think it's a VT7 paradigm, for example. And the next function I want to show you is a quick test function. So it's kind of similar to their assessment in terms of the upper part. So we have the EEG scope here and also signal quantity. And for the bottom part, it looks a little bit different. There's no ERPs anymore, but there's a moving green bar here. So whenever the classification is done correctly, so when the patient's consciousness detected, then this green bar will moving from the left to right. And eventually, a quick test result will be dependent on the the whole assessment session. So in this way, we can have a very short but real-time assessment of the brain functions of the patient. And the next, probably also very important function I want to explain is the communication function. So here, once we have a successful assessment, we have a very high accuracy, so we know that the patient now is active and the patient has the active period, and the patient is able to follow instruction, so we can try to communicate with them. And here you can see on the bottom part we have the options to ask questions. For example, if you are in Austria, for example, then we could, for me, I should expect it's a yes answer. Then the patient should focus, for example, on here on the left side of the stimulation. That's a target. So if the patient can still understand the instructions, can still focus on the correct side of the stimuli, and we could get a correct answer. So that's a way we can ask questions to the patient and get responses from it with the communication function. So as a short summary about these few functions it's presented, and you see all these paradigms it's presented is using the audible paradigm. That's a P-strand paradigm. So we have different stimuli like the auditory or the vibrotactile stimuli. And so for the auditory paradigm, it lasts around, let's say, 5 minutes, a little bit less than 5 minutes. It's a little bit longer than the vibrotactile paradigms. So usually that's also why for the when we are testing with patients, usually we start with the vibrotactile paradigms, because it's shorter and it's more likely to catch their the active duration of these patients. As we know that these DOC patients, they are usually very easy to get tired or even falling asleep. So we need to really to repeat this probably several times to get to catch this small window when the patient is active. So usually we start with the vibrotactile paradigms. And of course, if possible, when we have the assessment successful, then we can try a quick test for a real-time assessment, and the quick test is much shorter. You can see here for the AEP, the auditory paradigm, the quick test is like 1.5 minutes, and for the other vibrotactile paradigms, the quick test is like a little bit over half a minute. So that's a very quick way to see if the patient is active or not. And of course, we can also try the communication if the assessment is successful. On the last kind of few thing I want to show you is the ERP results. So that's usually after the assessment what we want to see in this case. So we first want to see if there's a significant difference between the target stimuli and the non-target stimuli. That's the case here. So we see the blue and the green lines here. One is from the target, another one is from the standard. So whenever there's a significant difference detected, then it will mark as a green shadow here. So that's the way we want to see if we can get the correct response from the brain, and we can use this to assess the brain functions of the patients. So that's usually the way. And usually we can also see the accuracy plot. You see in the middle of this image. That's the way we want to give a number. So usually we would expect the patient would be able to communicate if they can reach accuracy above 70% or even higher. So I mean, usually for the DOC patient at the beginning, it's very difficult to get very high accuracy, but after some training, we could gradually increase performance, and eventually we can get high accuracy, and even we could able to communicate with these patients. The like the last paradigm I may also want to mention is this. We also have this like model image paradigm. That's the results from the model image paradigm. Probably you already see this from the recovery results. So yeah, that's the way we we can show the results were quite similar to the recovery. So for some of the STIOS patients, they have also a ability to imagine the hand movement, for example. Then we could also get this ERD or EIS, that's kind of event related simulation or event related simulation from the patient, and we can also use these to assess the brain functions of these patients. Of course, it is more challenging task compared to these audible paradigms, but this, of course, is really depends on the patient group. As we know, it is really the patient is quite different from one to each other. So yeah, we cannot expect this works for everyone, but we do see like good results from some of them. That's of course, I mean, that's the feature, let's say, of these patients. They are like severely impaired, and we couldn't usually need to work harder with this patient to get good results. So that's of course a challenging part of this Manbig system, but that's also something we would like to see with these patients. So then I will move to the live demo. I guess we can answer the questions afterwards. I mean, if any of you have any question, I think you can just write down and we can answer the questions after this live demo. So now I'm going to invite my colleague and we can start the live demo. Okay, I'm going to stop the screen sharing. So, and I can switch the camera so you can see hopefully better.
Yes, can see the
You can
wide
see,
angle camera angle.
yeah, you can see it. Okay, so I'm going to invite my colleague Vashani. Please take a seat.
Mashali, I envy you again. You can try the second time.
Yeah, maybe you sit on the other chair, it's better, I think. So yeah, I think this way would be. Oh, okay. So I hope you can see the whole Manbig system here. So this one is the laptop we use to receive the signal, and also you may see some like now it's just showing a few patients here. I already built
The reflection is a little bit like too strong, maybe like too lower a bit.
Maybe I try to switch on the lights. Wait a minute.
Thank you.
It's not getting better, or
a tiny little bit better. Maybe turn the control computer a little bit towards the wide angle camera.
But now it's getting better or worse?
Worse, it's all white now.
It's all white.
Yeah, I
now.
see the paradigm now.
Okay,
Yeah,
now, okay,
better.
okay, great.
Thank you, thank
So
you,
now,
thank you, thank you.
okay, now you can see better. So, yeah, I will start with preparing the EEG. So, as I mentioned, we're going to use the 16-channel G.tec, and I'm going to attach the EEG cap. So, Shirley, thank you. Okay. So let's see. I think I need to pull a little bit down here. Maybe also folded it here so it doesn't cover the eyes. Okay, I'm gonna make it a little bit tight. So, can I still make it a little bit tighter? Or maybe I put it on the chin instead of here. It's better. Okay, looks great. Yeah, anyway, I'm going to choose one for the paradigm, like we can start with the VT2 paradigm. It's shorter and easy to see. So can also see now the EEG signal, of course. Don't know, now it's getting white again, or
White.
yeah, it's tricky with this. Try to turn.
Yeah. Yeah, can see the EEG scope.
Okay, perfect.
Thank you, thank you.
Okay, so now I'm going to prepare the EEG signal, and hopefully you can see some clean EEG afterwards. So to attach the reference, is it okay? And the ground. So I think gradually you would be able to see some signals. As probably now you can see the signal quality indication. It's, I think, either red or yellow, so it means lots of channels are still bad because we did not play any JLs inside it yet. So after the preparation, we should expect a good signal on all channels. So probably if you observed that we have kind of same setup as the Recoverex. So we also have these 16 channels of EJ electrodes.
Are you going to demonstrate the motor imagination paradigm?
Let's see, depends on time. I will start with the vibrotactile paradigms because it's like easy and shorter. I mean, anyway, you already know very well about the Recoverex, the motor imagery in Recoverex. So I don't think I will go for that one.
Very good. Vibrotactile sounds very good.
Yes, so now if you can see, now we have like a very clean signal. That's great. And now I'm going to place the vibrotactile stimulators. So for the VT2, we have two vibrostings on the left and right wrist in this case. So the first, I will start with the right one. Yeah. Now I will just ask Vashani to hold the right vibrosting, and the other one is on the left. Okay, so you have both. Okay, perfect. And I will also put on the headphones. So this will give an auditory like instructions.
So Vishali is holding two fiber stem
Yeah,
in
I don't
her
know if
hands.
yes. So for the patients, usually we use a tape to
Yep.
fix it. So
Yep.
here, just you know, we can just ask her to hold it,
Okay.
and then we place the headphones. So let's see. Okay, it's good. Maybe I can put a little bit tight. It's good. So maybe you can place your hands a little bit aside from each other, or you can put those on your yeah, just to keep a bit relaxed. You can put maybe a bit aside from each other, so it's easy for you to focus on. Then okay, there will be an instruction that's there will be a like audio instruction telling the left wrist that's a task, the target that you need to focus. So focusing on the left wrist and ignore the other side. That's a task for Vashali. Also, that's the instruction we need to give for the patients when we're doing this demo. So, okay, ready? So we just start this session. So this session lasts like around 2.5 minutes. And now we can really see some evoked potentials from the left, I mean the target stimuli. So the green line here is from the target, and the blue line here is from the standard, that's a non-target. So actually we can see a huge P300 here. It's very nice sign of the brain responses.
Ren, can you maybe help us a little bit with the angle? Maybe you turn the Mind Beagle control computer towards the wide angle camera a little bit so we can see a bigger screen of the Mind Beagle control computer.
I can see if I can put the camera a little bit closer, maybe. No, let's see if this would be better.
The exposure. Okay, then you how to say 撳低啲按下一点那个荧幕，就是control computer monitor like tap it down a little bit because it become very
Bit
white.
down like this.
Yes, yeah.
So I think now it's better.
Yeah, now I can see two lines. The
Okay.
green and blue line. Okay. Yeah,
Okay,
I just want
great.
to make sure everyone can see two lines.
Okay, great. So yeah, thank you.
It's
Yeah,
the
so that's
real-time feedback.
yeah, that's very important. So nice. So you can see
report.
the accuracy. Yeah. So that's accuracy. You see it's 100%, and great job, Vashali.
Yeah, Vashali is famous for that.
Yeah, perfect. So you can see also very nice evoke potentials here. That's perfect. So we can just save the results, and we can also save the historical data in this case. And we can continue. So for the VT2 paradigm, as I explained before, we could do a quick test in this case. So it's kind of very short and real-time assessment. So it's just half a minute, and we can see the results from the quick time test. So the
because now it was kind of the calibration, and then now it's a quick test to make sure the calibration is all good.
Yeah, exactly. So that's a way. It's also a very good way to assess the status of the patients. Because for the DOC patients, the status could be very dynamic and could change from 1 minute to another. So before, especially, for example, if we want to do communication, before we doing that, we could just do a quick test beforehand. So we know, okay, if the quick test is successful, we are more confident, the patient is conscious, and it's more likely to get communication results with the patients. So the setup of the quick test is saying as assessment or as a calibration, let's say. On here, we can just continue with this one. So if Vashani, you're ready, and we could start with the quick test. It's just half a minute. So the same task to focus on the left. Okay, let's go. Yeah, so you can see the real-time feedback from the green moving bar, which means once each trial is classified correctly, then it will start moving. And depending on the whole processing of this half-minute data, the quick test will tell us if the result is if it's successful, if the quick test is successful or not. Yeah, so you see it's a successful quick test. Great job, Vashali. Yeah, so that's the way we usually start with the VT2 paradigm, because as you can see, it's very short and easy to get an idea of the responses of the patient. And we could also try the VT3 because here we can also not just do the quick test, but we can also do we can try communication. Of course, again, we need an assessment as a calibration run to get a the to get a classifier and which we can use later for the communication. So for the VT3 paradigm, it's a little bit more challenging, as I explained a little before. So we have not just a left or right stimuli, we also have additional vibrostim on the left ankle. So it's kind of distractor. So the tasks of patient would also be a little bit more complicated. So they need to actually to switch the target from left to right. So at the beginning, it will be the left, and then it will switch to the right. So now I'm going to check again. So if this is still to the right and the left, yeah, so the location of the right and left is the same as VT2, and I need additional vibrostim to place on the ankle. So that's I'm going to do it now. So I'm going to place additional vibrostim, on the left ankle. So I can just place it. Okay. Yeah, so now we have 3 vibrostings placed on the left right wrist and the left ankle. So just try it again. Can you feel it? Great. Now we can go for the assessment run, or you can also call it a calibration run. So now, Mashali, so the first you need to focus is the left, and then you need to switch on the right. So now, of course, Vashali will receive an auditory instruction at the beginning. It's the left wrist. Okay, let's go. So you may also observe here this bubble is like blinking, which also indicates the sides that the patient needs to follow. Okay, now Vaishali shift to the right side, focusing on the right. So it's similar to the VT2. We could already see a huge P200 evoked potentials here. And it is also very significant to their standard stimuli. So you can see this difference between the green line and the blue line. Okay, perfect. So you can see the accuracy again is 100%. Great job. So we could also take a little bit look into the results as we get here. See their like the evoked potentials, it's significant, and we can see the accuracy reaches 100% just after like a few trials. So again, we can just save the data here, and then we can continue with the communication. I think I still have a few minutes, so I will just maybe try 1 or 2 questions to see if we can communicate in this way. So for the communication setup, you can see it's basically the same as in the assessment. We still have the same locations of all the vibro stings, and the task now, of course, is depending on the question. So for example, if now I ask a question, are you in India? So there, I would expect it's a no answer, then you should focus on the right side, on the right wrist. So if I explain this to the patient, and then we can start. Okay, so now focusing on the right side if you want to answer no. Okay, let's go. Focusing on the right side. No, it's a wrong answer, but I guess Vaishali is really want to go back to Indian. But oh, you tried the wrong one. Okay, okay, that makes sense. Okay, so we just repeat another one. And now, do you like Manbigo? I would say I would expect a yes in this case. And now you just need to focusing on the left one, and we'll see. So now, okay, starting with the left one. Let's go. Okay, great. So now we got the correct answer. Yeah, thanks a lot, Vashali. I think we can stop here because of the time. Okay, let's move the
Thank you, Ren. Thank you,
platform.
Vashali, for the consistent 100% accuracy.
Yeah,
and
it's
even,
always percent. Should I? Yeah, great job,
and
Vashali.
even
Yeah,
demonstrated the yes no question.
Yeah.
That is an important feature because it is what makes MindBeagle fascinating.
Yeah,
That
thanks
we
a
are
lot,
able
Vashali.
to. Thank you,
Yeah.
Vashali. Thank you. Thank
Yeah.
you.
Okay, so I can switch the camera back.
Mhm.
Yeah, so
So, Ren, I heard that there is a kind of a new package of MindBeagle that is more of a MindBeagle Lite that without the AES component. Yeah, can you tell us more about that?
yeah, so as I just showed before, now we for the current or the previous one or the one I'm just showing is with a cube. So we put everything inside it, not just the auditory, the vibrosting. We also include like the FES, the functioning stimulator, and the test, and like the transcranial direct current stimulator there. And exactly, so now we are going to have a more compact one, let's say. So you see now the cube is with a relatively large size because we put everything there. And exactly, so now I think that we will have a kind of separate design. So we'll have an auditory box and a web like a WebRSteam box. So it's kind of G box. That's
Yeah, G everything.
yeah, G Xbox. Let's say.
G.
Yeah. I already saw the photos of this design, and now it's like the first like version is now I think testing in our Barcelona office together with the software part. So hopefully I would be able to show it soon, because it's more compact and of course it's more portable and of course more affordable in terms of the price.
Yes, definitely. I have sent quotation of the light version of the
Yeah.
G2 boxes
Yeah.
version already. They are like surprised. Oh, voila, so much
Yeah.
more cost effective now.
Yeah.
So yeah, it's a good news
Mm.
for the audience as well.
Yeah.
It is more affordable, so more hospital, more patients, family, or the healthcare
Yeah.
professional can communicate and to do brain assessment with the DOC patients as well. Thank you, Ren, for this very nice presentation to let us have a real-time live demo experience together. How does it look? The MindBeagle system looks like, and very nice interaction that we have. That question is not related for this session. Any participants have any questions about this MindBeagle BCI communication and brain assessment system for DOC patient? Please feel free to type it in the Q&A box. Yeah.
Yeah, I mean, if you have any question afterwards, you have my linking, you can just write to me, you have also my email address. You can also write me anytime. I'll be happy to answer the questions afterwards. Yeah.
Yes. So thank you very much, Ren. Like look forward to catching it when I'm in Austria in February, and then bring the good news back to Asia Pacific. And I know Hong Kong and Singapore also looking into adopting this MindBeagle. So this light version would be good news for them as well.
Yeah,
Yeah.
definitely.
Thank you for all your
Okay.
good work. Help me hug especially. Thank you for
Yeah.
her good performance.
Yeah,
It's a
as always.
real reaction, real phototactile paradigm. Thank you for all the nice work. And we keep in touch. Have a good day on your side.
Yeah, you too. Bye.
Thank you, Ren. Thank you. Nice. So here we go to our finale session. We have Mr. Will Wong from Hong Kong, the owner of MedMove Integrative Health Clinic in Hong Kong. He is our Recoverix franchise partner in Hong Kong, and he is also a physiotherapist professional. He is going to share with us the topic, empowering possibilities, envisioning a brighter future for stroke and multiple sclerosis patients in Hong Kong through Recoverix Clinic and outreach service. At noon time today, we kind of discussed a bit, is it possible to deploy more home-based neuro rehab treatment for stroke patients? And here we go. Mr. Will is already have some experience and some system, some stories to tell with his outreach service with Recoverix system. Thank you, Will, for your preparation for this presentation. I can see your screen nicely. Maybe you can try to put it full screen, because some people may be joining us with mobile phone. Then we put it full screen would be easier. It's a bit fuller.
Yeah.
It's a bit fuller, but not totally full. It's fine. Okay. So it is Will, the owner of MedMove Integrative Health Clinic, also our Recoverix franchise partner in Hong Kong. We have a good grand plan that in 3 to 5 years, we would grow Recoverix network into like 14 outlets, Will, and together with outreach services. So looking forward, please enjoy this session. Will, here you go. Stage is yours.
Friends Christy, and they're very fruitful today. Learn a lot, and I guess you may be a bit tired. Then yeah, I try to make it quick and try to share some experience and what I'm doing in Hong Kong. So good afternoon, everyone. Thank you for joining me. My name is Will Wong, registered physiotherapist in Hong Kong, and I'm running a MedMove Integrative Health Clinic and focusing on recovery X, because here we are lacking in neuro rehabilitation specialist clinic in private setting. Then today I would like to explore how we can empower stroke and multiple sclerosis patients in Hong Kong and envisions a brightened future for them through our recovery clinic and outreach services. Basically, MedMove Neuro Rehabilitation Clinic basically is a Hong Kong-based, internationally recognized private medical institution offering cutting-edge brain-computer interface and neuro rehabilitation. And our team, including experienced registered physiotherapists, I'm one of them, and neurologists specializing in treating stroke and multiple sclerosis patients, neurologists including those neurosurgeons, and those some of them are my advisors for stroke rehabilitation program. And also we are specializing in treating stroke and MS patients, help them to regain their function, like reduce their pain, spasticity, trauma, and improve things. Very important thing, as a physiotherapist, we all focus on mobility. That is a key for the patient and also their care. Then also we would like to increase their balance and gauge all those mobility, functioning ability. The introduction, then I would like to share the challenges and opportunity we are facing in Hong Kong with all of you coming from all over the world. To begin, then basically, I would like to give you the overall picture, have a brief overview of stroke and multiple sclerosis, and there's some statistic on prevalence in Hong Kong. Then let's begin. Let's understand the challenge phase today in Hong Kong. Then basically, we know that the stroke basically begin the fourth leading cause of death with around 3,000 fatalities each year. Then it's come to a major concern in government and also the community, because we all, if you ask someone if they are afraid of stroke, yeah, probably 100%, they will, they are really focusing on that health issue. And also, similarly, multiple sclerosis is on the rises, with its prevalence increasing by over 40% in recent years. These conditions not only impact patients, but also their families and communities. Then I think the situation in other places is similar, because, you know, stroke would be the big population, and, you know, people and government pay extra attention in the prevention or the interventions of stroke rehabilitation. Then here's come to the impact of stroke and MS. The impact of these conditions is profound. Then stroke can lead to various disability, including weakness in limb, maybe upper limb and lower limb, and also the speech difficulties, as we hear some prolonged professional sharing about the like how to make use of those BCI to improve the speech ability. Then it's actually is a breakthrough technology. Then for this impact and burden, I think you know, today is the focus. Then recovery is one of the key solutions for tackle those disability, including the weakness in upper limb and lower limb speech difficulties, and also the cognitive function. As a physiotherapist, we are not only focusing on the physical capability, but also we also are concerned about the cognitive function, because we all know that the physical motor performance actually can have a co-relationship and with the brain activities. Then patients often experience emotional challenge like anxiety and depression. Then mental health is also our concern, not only physical health, but also we focusing on taking care of their mental health issue. Like very common to see stroke patient or MS patient, they may have some mental health issue like anxiety and depression. And we know that from Christy's presentation today, there are lots of study ongoing for like antidepressant or like dementia, those cognitive function and mental health issue. Then it's hope that we can see the ongoing process. And moreover, caregiver also facing increased responsibility and stress. Then it's highlight the need for comprehensive support system for both patient and their family. In Hong Kong, you know, Hong Kong people are really hardworking. You can see Christy and me, really hardworking people. We all focusing on our work, like we work for more than like 12 hours a day. Then we, you know, we relied on the help from the maid, like for the like our family members, like children or elderly. Then you can imagine in case of those episode like the stroke patient, then there's a big burden on the family and the carer. Then in this case, we do need for comprehensive support system and for helping those people in need, especially those patients suffering from stroke or MS, which are the crossing their physical mobility problem. Then that means they lose of their abilities of the independent living. Then they have to rely on the carer. Then in this case, this is the great burden on the patient and the family, also the whole community. Then in this case, I think it's a situation similar in other places in Asian country or other European countries. Then that's why, as a physiotherapist, I think recovery is the breakthrough for what we are doing for many years, for more than a decade. As a physiotherapist, we do hope to help others to improve their mobility and enhance the quality of life through improving their mobility, ability, and the independent living. Then that's why here is the introduction of the recovery clinic in Hong Kong. I set up the recovery clinic more than half year. Yeah, it's very newbie, but I'm working very hard on promoting recovery clinic in Hong Kong, including, as the topic mentions, not only the clinic-based setting, but also the outreach services. Let's talk about recovery clinic. I would like to hear other experience after my presentation, to hear your experience in the recovery clinic in your country. Yeah, and we do have the regular monthly meeting to share our viewpoint and what we are doing and our experience with other Asian country regarding recovery clinic, the operation and the experience, like the current journey, all those aspects. Then our mission is to provide comprehensive rehabilitation services tailored specifically for stroke and MS patients. We integrate conventional medical practice with holistic approaches. Then you may hear the fantastic talk given by Dr. Vincent Trent. Then also talking about the integration of the conventional physiotherapy approaches with the low-flow neurorehabilitation technology in order to enhance and live up to the standard of our physiotherapy practice. Then those conventional medical practice with holistic approaches, including physiotherapy and like the consultation given by our medical team. Our objective is to enhance patient independence and improve their overall quality of life through personalized care plans. Is a very important point, because in the old day, maybe we do not have those technologies to help us, and then we have to use those conventional physiotherapy practice, like manipulation, will help the patient to move their affected limb passively. For those patients with chronic stroke patient, then sometimes we find difficult to help them by only the conventional way. But today, all the breakthrough of medical devices with the recovery devices, then we do find a new solution for them by integrating the conventional and low flow with neurorehabilitation technology. Then it did help a lot for the patient. Like you hear the sharing from Dr. Chen, then talk about the very effective and good result. Yeah, even though the sample size not that huge, but you already see the impact and the breakthrough with those integration of the holistic approaches. The benefit of our clinic, the benefits of our clinics are significant. We try to develop personalized treatment plans that catered to individual needs through a multidisciplinary team approaches. I think some of you from medical background and some medical professional here today, then we all promote interdisciplinary approaches in our medical setting. Then here is our clinic also strongly promote multidisciplinary team approaches. We all know that early rehabilitation is emphasized to maximize recovery potentials after a stroke or during multiple sclerosis like those progressions. We do provide continuous support from our healthcare professionals, ensure that our patient received comprehensive care throughout their recovery journey. Then you can see we provide personalized treatment plan, and also we emphasize multidisciplinary team approaches with the contribution from different prospects from different medical professionals like neurosurgeon, like nurses and physiotherapists. Then we collaborate with each other to provide a comprehensive solution for a patient. Then patient center is the key. Then patient center care model. Then we do hope to help the clients, stroke clients to go together throughout the recovery journey. It can be long. We hope to give them a little hope. Outreach service is one of the key components of recovery clinic in Hong Kong. Maybe you, some of you may not have ideas about Hong Kong, like you may never been to here, but I guess you may know that Hong Kong is a really small cities in the world. It's like a dot on the map, but actually then that's why it can be an advantage for us to deliver our services, because the small places, that means if I drive a car to somewhere like from the east to the west or from the north to the south, then basically less than 2 hours, then I can access to that point, even though it's far away from my destination. Then it's still it's like only it takes 2 hours. Then in this case, we can provide the home visits or home services to our client, especially those stroke client. That they have the great difficulties in mobility. Then some of them, even though they are backbound, then in this case, for them, it's not possible to come to my clinic, even though my clinic in the same district. Then they still, as I mentioned, a backbound or a chairbound. Then in this case, it's very difficult for them to move out of their bed. Then in this case, outreach service is one of the key for providing the comprehensive solutions for stroke client. In addition to our clinic services, we have a robust outreach program designed to extend our reach into the community. This includes community workshops focused on stroke awareness and prevention strategy, as well as home visits, as I mentioned, for personalized assessment and support for those unable to travel. Then I often got a call to ask about our surfaces, especially the recovery surfaces. Then they may wonder how if this is effective for the chronic stroke patient or a new stroke patient. Those questions, as a physiotherapist, I would love to give them the consultation and advices, but for them, it's better to give them physical contact, like the physical examination or assessment. In this case, I would love to provide outreach services like the outreach, like in-person physical assessment, and then to assess if they are suitable for the recovery treatment. As you know that at least some of them may be backbound, but they can still receive recovery treatment, because we focus on like their cognitive level. If they can understand the simple command or instruction, then basically then they can receive those new solution for them, especially for those chronic patients like with hopeless after suffering for stroke for more than a decade or 20 years. But outreach services give them a little hope. And for achieving the goals of outreach services, then we also are collaborating with local organizations which help us enhance resources availability for affected families. Like for some local organizations, especially those local stroke patient organization or stroke care organization, then they have the very strong network for the stroke or MS patient. Then in this case, we we would love to collaborate with them in order to spread out the concept and the ideas about the holistic approaches, and then they know that they can still, no matter they suffering for more than a decade, and they try more than 10 intervention, but it's then they can we can provide the new solution for them by the outreach services in order to break through all the barriers, geographical barriers. The community impact is one of the very important issue, because you can imagine like the stroke patient and their carer, some of them may not that they may be quite suffering from the mental issue, like they may be very anxiety or very depressed. They may not find the external support for them. Then in this case, we have to be proactive to provide the community services, like outreach service has made a significant impact on the community. We have numerous success stories that highlight improved patient outcome through our initiatives. Then testimonial from patient and family emphasized the importance of strong support networks. Furthermore, our partnership with local health authorities aim to raise awareness about stroke symptoms and improve MS management. As one of the executive committee members of Hong Kong Physiotherapy Association, which promote the physiotherapy education to our community and let them know any integrative approaches and solutions to deal with different diseases. Then we try to partnership with different authorities, then aim to raise the awareness about stroke, like education and like multiple sclerosis management. Then you know that recovery is not only effective for stroke client, but also is the unique interventions for tackling MS patient. Then it's a really amazing and effective news for all of us. Here with I would like to share a few cases, real life success story. You may think that success is this mean like the amazing result like shared with, by like Christy or other professional. Like you can see the livelihood pick test or 10 meters walking test. Then you can see the great improvement in the result, like from like 20 seconds to 10 seconds, something like that. But here is not that amazing result. But for me, I do think that it's still success story, because we are not only focusing on the result, but also we also look at the the improvement in different aspects throughout these cases. The first cases you can see, I provide the outreach services to this 65 man. He is a musician. Basically, he is a drummer. He's a very famous drummer. Used to be a drummer for like local famous band. Yeah, it's like he used to be the drummer for like the superstar. Then for him, it's really a disaster when he was suffering from stroke. Then he become another one. Like you can imagine, he used to be a very shining drummer on the stage, but after stroke, basically he hide himself and no motivation for everything at all, like waiting for the end of the day, like that. But his friends do want to help him to regain the motivation in life. Then that's why his friends Google it and find recoveries. Then he used to stay in mainland. Then their friends help to remove all the barrier and try to help him to come to Hong Kong and then to receive recovery intervention. Then you can see the picture. Basically, it's a hotel. Like I look like Uber driver, and for recovery treatment, basically it's very portable one. Then I just pick up a big suitcase, then go to the hotel, and I drive my car. It takes around like 30 minutes from my clinic to that designated hotel, and you can see the hotel environment like that. That's normal hotel, right? But you still have to think about how to set up the recovery devices. Then for him, basically, it's a tall man. It's like 1.85 meters. It's really a tall man, taller than me, and is quite a big man. Then that patient, stroke patient, is a chronic stroke patient. Then basically his mobility is very poor. Like he had to rely on two moderate assist from the carer to move from the bed, from lying to sitting. Then you can imagine I get there with only one main support, and then transfer the patient from the queen size bed to the picture, to the chair shown on the picture. Then I set up the recoveries there, then help him to take the recovery treatment. Then for that cases, And as I mentioned, it's a success story for me, but it's not like I'm not going to show you very amazing result like the 10 meters walking test. But for these cases, except the recovery treatment around 1 month, around less than 10 times. But within the 10 times treatment, because due to some reasons, because low care and some like accommodation courses, then finally he was delivered to the nursing home. But during their journey, I provide the recovery treatment for him, and the carer and his friends told me he regained the motivation and confidence for rehabilitation through our intensive outreach rehabilitation service, because he do find something different compared with the passive intervention, conventional treatment, because he can like imagine like the motor imaging and the visual feedback give him a little hope, and he find it maybe it can be the solution and help him to break through the barrier. Then I think the motivation and the confidence for rehabilitation is one also very important for like our physiotherapy treatment. Because that recovery is the active approaches, because the patient have to like follow the command and very focused on those command and look at the screen for getting the live stream neural feedback and visual feedback. And the second cases I would love to share is the 73 old ladies and stroke patient then who benefits from our integrative medicine approaches, combining recoveries with conventional physiotherapies. You can see the picture here. It's another setting. I also provide outreach services to the old lady who is living in the nursing home. Then you can see the nursing home is quite big, but it's quite privileged one compared with others like old people who may not afford those accommodation courses. But for her, they do not have like financial difficulties, then they can have those big room. Then in this case, I help them to set up the recovery treatment stage. Then you can see basically the carer often spend their time long working hours to help and to accompany with their family, and they already received various of conventional treatment from others like Leo team, like more than a few years already, but they find it's not that effective. Then they approach us and ask for our help. Then you can see this patient compared with the previous one. This one is like with like poor ability. Then I can say you can see the picture is a wide tube patient. Then that's mean for this patient compared with previous one, the big guys basically he's a wheelchair client. Then you can still try to help the patient to go to somewhere with the wheelchair. But for this patient, as I mentioned, is the backbound patient with the wide tube all the time, because she loses the abilities of the swallowing. Then she has to maintain the survival through the right tube for the like the nutrient intake. Then in this case, we can still help the patient to receive the recovery treatment, because she is kind of like a very sound patient. Then she can follow the simple instruction very well, basically. But you can imagine this chronic stroke patient suffering from stroke more than 20 years, already have like very spasticity, upper limb and lower limb. Then in this case, but she can follow the simple instruction. That's why we can still provide recovery treatment for helping her. Then, for this old lady, then after receiving like around 5 to 6 recovery treatment, then their family members share with me, they observe the patient have less spasticity over their upper limb and lower limb when they, observe the patients to have like the walking treatment. Then they can see the improvement of the less muscle tone over their upper limb and lower limb. Then and for me, and also they also share like they think that their family that stroke clients with better cognitive function, like before the recovery treatment, they think that the clients always like sleepy and extreme fatigued, just lie on the bed. But with the regular recovery treatment, I provide like 3 times a week recovery treatment, home visit. Then basically they think that the client with better cognitive function and less fatigue, that's kind of like energizing for her emotional state, leading to enhance the daily functioning, because she can still like have more motivations and less fatigue to like change the position from supine lying to like sit on the wheelchair, to like watch televisions with their family together, but not only lie on the bed with the new intervention. Okay, after sharing the 2 outreach services, the future directions I would love to share with you. Looking ahead, we envision an even brighter future for our patients. Our plans include expanding services to offer more community outreach programs that focus on prevention and education about stroke and multiple sclerosis. We are also developing new integrative treatment protocol specifically for multiple sclerosis patients to improve their symptoms management. Continuous collaboration with healthcare professionals will enhance service delivery and patient outcomes. Then is a new start for my clinic. Then in less than 1 year, integrate the new recovery treatment to our clinic and provide the integrative approaches for stroke and MS patient. Then I do have a great expectations on this recovery because it's really effective, no matter the sample size is not that huge at the moment, but we already see some like very encouraging result throughout the journey. Then as you see this picture before, recovery gym worldwide, then Hong Kong basically is a small dot on the world map. But I hope that like long-term goal, maybe it can be happened within 3 years. I hope to show you the Hong Kong map. Then we can be scaled up and provide many dots on Hong Kong map with the recovery clinic and help more clients to help them to improve throughout their recovery journey. Yeah, thanks for your attentions today. Yeah, it's a long day. If you have any query about our surface recovery clinic or would like to know more information, yeah, feel free to text me, email me. Yeah, do not hesitate to reach out. Yeah, you can find my contact information on like LinkedIn, or yeah, we can share later. Yeah, let's see if any questions.
Thank you, Will, for your nice presentation, and we can all definitely feel your passion in recovery. Thank you for trusting the technology, and thank you for all the groundwork, really like sweat groundwork in Hong Kong to penetrate the recovery surface to benefit the stroke patient, no matter they are in the hotel or in the nursing home. Just want to like make recovery reachable to them. That is, we highly appreciate on your effort and really pioneering this recovery outreach service in Hong Kong. So I know that in the participants group, there are some of you are considering bringing recovery to your country. Yeah, Hong Kong and also Bangkok is the very first territory that taken up by an investor like Will, and I have a partner in Bangkok. They really see the need of the stroke patient, then just become a franchisee and have a grand vision to make recovery more accessible to the stroke patient. So if you also see the need in your country, in your territory, let's say Australia, New Zealand, China, Taiwan, Indonesia, feel free to give me an email. We can discuss the return on investment, and we would make sure all the franchise partner get well trained to operate the recovery system properly and provide the standard protocol therapy to make sure the best patient outcome, to serve the patient, to give evidence-based hope to them. So yeah, any questions from the audience regarding this recovery clinic operation and also the outreach service? And while waiting for the question, I would like to take these few minutes to really thank you all for joining this very first Brain Computer Interface Neurotech Masterclass Hong Kong today. Yeah, I'm based in Hong Kong for this BCI company, and it's really very heartwarming to see something is start blooming. And there's a little milestone done, and then we leverage and further promote the adoption of BCI technology in benefit for the technology advancement and even the patient benefit. It's really meaningful, means a lot to us that all of you stayed with us the whole day and attend all the session to selflessly share knowledge and learn from the speakers. Thank you for your participation. Yeah. And Will, thank you for your preparation. And yeah, thank you for trusting me in this franchise business and your good work in renovating your clinic to make it more recovery focused. And your hard work in the outreach is also a touching story from Hong Kong already. And together we can penetrate the recovery service further into the community. And of course, Will is also welcoming to partner up with some let's say, chain clinic group, and also private hospitals, and also some NGO group that maybe you have a private clinic wing. Please feel free to partner up with Will to deploy the recovery service more into the community.
Yeah, thanks for your support, Christy. And for today, thanks for organizing such a wonderful sharing section with all the professional. And I already heard from some local expertise in that aspect and some old friends as well. Then it's already inspire me. Then we can do some collaboration with different setting like the university and also the NGO or hospital. Then Dr. Chen share with us then for the RCT, then basically I think we can collaborate with the private and community sectors and to try to scale up and try to recruit some patient and see the ongoing process of how far we can go.
Yeah, that sounds good also. The RCT, like of course joint force and multi-center, like different stroke type, all that better together. Yeah, it's all good opportunities. We just talk with people, reach out, talk, collaborate, and good things will happen. David, in the Q&A chat, he is based in Hangzhou, our BCI BCI distributor for Unicorn EEG headset. Saying a great thank you to all the keynote speakers. Really, thank you all for your contribution to this education event. And because from time to time, I will look at the statistics on Zoom, right? And I have hosted a different masterclass already. Masterclass Australia, Masterclass Malaysia, Masterclass Hong Kong for this time. And usually the online attendees is about 30 something, 40 something. And today, most of the time is like 57, 61, 54, like this average. I'm happy that you guys stay together. I'm very happy with these statistics. Okay, we have some question or some feedback in the Q&A. Someone says, thanks for wonderful masterclass you provide. Thank you for joining and all the session. And Sean is asking, what would be the limitation to implementing a BCI rehab system like Recovery in a public health system, like the NHS in the UK? What are the key publications that emphasize the evidence available? Oh, very nice question. Will, maybe I can answer that question. Right, public health system, usually they would more look into the, they want to have local evidence. Then from scratch, we won't have a local evidence because it is a Austria invented system, right? Then we usually share the already done clinical study that if you are interested in what are the papers, let me also quickly show you. Let me share screen, huh? Notice. Right, my g.tec paper folder. See, I have a lot of folder. Give me like 2 seconds. Here, this is my favorite recovery paper folder. And what I would say the most powerful clinical evidence, let me show you 3 papers now. This one published in Frontiers in Neuroscience. Yeah, BCI treatment for upper extremity for stroke patient. We use this Fugl-Meyer assessment as the primary endpoint, and you see the p-value is already catching eyeball, lower than 0.001. And the conclusion is even more, like the wording is quite definite, is quite rare as a clinical study use this definite wording, and because the evidence is so obvious that it is well supported to use this wording. You see, it can promote long-lasting functional improvement in upper extremity in stroke survivors, no matter how severe the impairment is. Functional improvement can be explained by improved neuroplasticity in the CNS. This is a good paper to show the efficacy in upper limb using Recoverex. The other one, of course, after upper limb, we have to see lower limb, right? And this is the paper, BCI for gait rehab in stroke patient. We also have mentioned the significant result in the walking speed. And this table in this paper is the most useful one. You see, we evaluate the patient outcome using all these assessment tools. And we can show this paper to healthcare professionals in the public healthcare system to go through the statistically significant assessment. A lot of them is lower than 0.00, lower than 0.05. And this one, these two, lower than 0.001, what are those? Time up and go test and 10-meter walk test. BCI gait rehab is meant for walking better. Then the time up and go test and 10-meter walk test is the walking test that's highly significant. So they say, also a powerful paper. Then what is the third paper that I want to show you? It is this one. It usually would answer the question about, hey, did you compare your Recoverex with traditional treatment? And I would always use this meta-analysis, which is in the clinical paper world. Meta-analysis is always a good reference, good evidence. Professor Kenneth Fong from Polytechnic University of Hong Kong, Department of Rehabilitation Sciences, same department as Dr. Jack Zheng, presented with us today. Professor Kenneth Fong also contributed in this paper. What is special about this? This meta-analysis paper compare those traditional physiotherapy, let's say FES, functional electrical stimulator. If we add a BCI component on the traditional therapy, how does it affect the patient outcome? And if we scroll to the result table for this, the result table and the conclusion, we see the traditional therapy added with BCI component, the patient outcome would be further enhanced. So BCI is important in neuro rehab. So if you want to get access to all these paper, of course, feel free to email me. My email is in my video window, lower right or left corner, li@g.tec.at. You can always find me on LinkedIn, Christy Lee, g.tec, then I'm here. And of course, the Recoverex website is also very useful for you to see the clinical results that we posted online. You can refer to it anytime, anywhere, share it with your friends, your colleagues, your potential business partner. Yeah, for the result. And sometimes words can be boring and sleepy. Then we have a lot of videos on our YouTube channel as well. Not only stroke and multiple sclerosis result, and we also have posted case studies. You see incomplete spinal cord injury, Guillain-Barré syndrome, Connors-Conder syndrome, long COVID and Parkinson's. All these, enjoy all your readings and evidence online and also offline with the downloaded paper. I hope I answered your question, Sean. So before having the local data, we always show them the Austria clinical study, then the public hospital can apply for a funding to buy in the system, then usually they would like to start a pilot study, small patient sample size to do a study, then scale it up like what Dr. Winston Chen from Haven of Hope Hospital has done and shared with us today. Right, we got another question. Will the video recording, it's a frequently asked question for all the master classes. Video recording, send me an email and I can share the link with you. Another question, I'm a student currently studying a course in neuroengineering, which includes learning about brain-computer interface system. While I understand the basic concepts, I find myself a bit confused when faced with scenario-based cases that require designing a BCI system. Although I am familiar with the simpler designs, the complexity of these scenarios poses a challenge. Could you please suggest ways to enhance my understanding and improve my skills in this area? Oh, then I'm not sure where are you based, and this weekend, next week I will be in Newcastle, Australia to conduct a BCI workshop in Newcastle in the ACNS conference, and early December I will be in the ICON-NIP conference in Auckland to have a BCI workshop as well. So more hands-on experience with BCI system would always work. And a lot of learning resources we provide. Upcoming would be of course, the workshop that I'm conducting in Australia and Auckland. But if you are based in other territory, this spring school, free of charge learning resources. Who doesn't like it? Free of charge 10-day spring school from 28th of April to May 7. Register for free. Secure your Zoom link, then 120 hours of talk on BCI system design and the research experience, selflessly sharing. So keep learning, and maybe also you can search for some scholar publications in Google Scholar or ResearchGate, search for BCI system or BCI study, then you can study the materials and the protocol design in the paper. Then you can learn from what has been designed already, then learn about what others have done, then try to be creative, then create something. That's usually the way to learn. I hope I answered your question properly. Sean, thank you. Great response. Oh, thank you, Sean, for asking question to make it more interactive. Ninu, how can I start the pilot study for motor imagery? How can I recruit participants? Start the pilot study for motor imagery depends on whether you are in a hospital or university research institute. I guess the procedure of applying, the procedure of submitting proposal and apply funding, get the grants for the research, the procedure could be a little different between university and hospital. Start the pilot study for motor imagery. Then you need a BCI system. Then you can start from contacting g.tec salesperson to get a quote of the BCI that is motor imagery experiment friendly system. Then you get the prelim idea of how much you need to pitch from your proposal. Then second question is, how can I recruit participants? So nowadays it is a lot of ways. Well, when I study in university like 17 years ago already, we always see some poster on those poster board. Then you can like 20 Hong Kong dollar as a as a gift after you have participated in the research. That is one way to post poster, then people come contact you, and then you give them some cash motivation. And nowadays, online and offline ways, there are a lot. Online recruitment, like using your first, second circle friendship connection to look for the subjects that you want to recruit, friends of friends, word of mouth, all that, a lot of methods. So the first step important is to start, and g.tec worldwide salesperson is available for you to prepare quotation for you. It's also one of your steps. Ninu also asked from where I can purchase the full kit. Would you mind to provide the link? If I provide the link, I would give you the g.tec website. And you can see in each product, in each system, you can see the start configurator icon, and then you can see some prelim pricing as well. Good. Thank you, Will, for staying with me for all these questions, not directly related to your session. And I feel less alone in this. And thank you all for the nice questions to make this event more interactive, more fun, or else would be me and speaker like keep talking like like a robot. Your presence, your participation is valuable, meaningful, heartwarming for all of us speakers. So thank you all for joining this very first Masterclass Hong Kong. More to come in Asia Pacific region, and for Masterclass Hong Kong, hopefully next year we also have it. So yeah, meanwhile, take care, keep in touch. Any questions, just email me, li@g.tec.at, and a lot of new innovation from g.tec will be announced online as well. We keep in touch, and Will, thank you again. Everyone, please take care and enjoy your late night, your evening, your afternoon. Thank you. Thank you. Have a good day.
Thank you.
Thank you, Will. Keep in touch. Thank you.
You should eat something.
Bye-bye.
Bye-bye.
Yes, I'm hungry. Thank you. Bye.
Bye-bye.
Bye-bye. Bye-bye. Bye-bye.